Good morning, everyone, and welcome here. It's a great pleasure to welcome you to the Senotherapeutics Revolution Forum, organized by the IPSA Foundation for Scientific Research, together with the IOR, Oncology Institute, and the IRB, Institute for Research in Biomedicine of University of della Svizzera Italiana. And the IFOM, Milano, FIRC Institute of Molecular Oncology. The IPSA Foundation was established in 2012 by the Swiss pharmaceutical company IPSA Institut Biochimique, based here in Lugano. From the beginning, the foundation has been committed to promoting an open, rigorous, and accessible scientific culture, encouraging the advancement of scientific knowledge, the sharing of ideas, and the dialogue between science and society. Our activities range from supporting research through fellowships, scholarships, to scientific outreach events that combine the humanities and science, such as this one today. This is our forum number 25. This forum brings together leading experts in the fields of cellular senescence, aging, and longevity, and we are proud to have curated a program that reflects the scientific excellence and innovative spirit that animate these emerging areas of research. A big expression of gratitude goes to the Foundation Scientific Committee of this foundation, represented here by Antonio Musarò, that is here today, with the Professor Alimonti and Professor Fabrizio D'Adda, has put together an incredible panel of international experts. I would also like to thank the team that reviewed and selected the abstract submitted for oral presentation and for the Best Poster Award, which will be announced at the end of today's forum. Thanks to Manuel Colucci, Miles Serial, and Fabrizio D'Adda again. We will conclude this exciting day with a special lecture at 6:30 by Professor Walter Rongo, a world-renowned Italian scientist now living in the United States, whose innovative research on longevity and ability to communicate science to the general public make him one of the most inspiring voices in this field. It will be a real honor for everybody to hear him speak this evening. Thank you all for being here. I wish you a day full of stimulating discussions, fruitful exchanges, and scientific inspiration. Before closing, I would like to extend a very special thank you to my colleagues Sara Comodo and Jacopo Crespi, along with the rest of my team and with the Professors Alimonti's team at IOR in Bellinzona, who have worked without rest over the past few months organizing this event. A big round of applause for them. Enjoy the forum, and now I will give over to Andrea Alimonti for an introduction to today's topic and the program we will be following. Thank you for your attention.
Well, thank you, Silvia. You just said everything. I prepared a speech, but essentially I don't know what to say now. So I really want to thank, therefore, Fabrizio. A few months ago, we received this short notice from IPSA to organize this meeting. And essentially, we really need to thank IPSA because they did everything. We just gave a list of speakers, but they've been tremendous in organizing this symposium. So, as you know, the topic of the symposium is about cellular senescence. And what is cellular senescence for people that may not know, right? So it's a state, a cellular state, which is characterized by a permanent growth arrest. This happens normally in aging, but it can also happen in cancer upon induction with different type of chemotherapy or any other targeted therapy intervention. And why we think this is important? We think that this can be an actionable target for novel therapeutics. We have named these therapeutics, many scientists here, Senotherapeutics. So with this, we mean not only drugs which can eliminate cellular senescence in an attempt to rejuvenate our organism or to defeat cancer, but also other type of therapy, like therapy which, for instance, can reprogram the SASP, or they can delay the occurrence of cellular senescence. So it's a really an interesting area, and what I really like most that in the recent years there has been an attempt to move this field forward. So there are already some clinical application. Industry is starting to get more interest in this Senotherapeutics. And so today we have invited here many speakers that are going to present also, I hope, data on new clinical study. But before that, we will have an introduction about cellular senescence from eminent speakers, which will really introduce us in the biology of senescence and will guide us in the understanding of this process. So I also have some communication. Remember that there will be a poster section. I think this is very important because here perhaps there will be the next generation of scientists. We are already very old for important discovery. So please don't forget to go to have a look at the poster section. Plus, very important, we will have this public session from Professor Longo. What does it mean, public session? That at a certain point in this room, I think around 6:30, we will also host not only scientists, but also people coming from Lugano. So citizen of Lugano. And I think this is very important because we need to disseminate the importance of work that had been done during the years by many of us. Very importantly, it was also told me that this symposium is accredited with 1 credit for from the Health Decay course for students. So don't forget to sign if you want to receive eventually a credit. Once again, I want to thank Fabrizio for organizing this, Silvia for generally supporting this initiative, this local scientific committee for the identification of the poster and oral presentation. And so we are ready to start. Thank you very much for coming, and let's start.
really very few words from me again. Thanks IPSA for having us here, and thanks Silvia for putting everything together, and then thanks Andrea for involving me. It's great to see this room, which is a big room full of so many people, including very young people. So Andrea has already alluded about the structure of the day, so we're going to have a morning which is going to be more basic science, and then in the afternoon we will discuss about the therapeutic potential, which I believe is more than a potential, in treating diseases associated, actually caused by the accumulation of senescent cells. So for the first morning, we have two chairs, Nina Cabezas, which I understand has recently become Swiss, or a Swiss scientist, and Luca Varani, which is a well-established Swiss scientist. And so they will be keeping the time for the speakers in the morning. Enjoy.
The first one is Peter Adams. Actually, if you can please come forward. He's the director of, I have to read this, of a director of a cancer genome and epigenetic program in San Diego. Thank you for coming over. And he will tell us about repurposing drug for aging and cancer, hopefully to beat them, not to favor them.
Great.
Please.
Okay, this on? Yeah. Thanks very much for the invitation to speak. It's really fantastic to be here, my first time in Lugano. And I also want to thank Silvia and the rest of the IPSA Foundation for supporting an event such as this, and also, support of science and public outreach in general. I think given certain things going on in certain parts of the world right now, this type of event is more important than ever. So thank you very much. So I'm going to talk. Does this? Yeah, okay. Yes, so I think I changed the title of my talk a little bit. I'm going to talk a little bit about cellular senescence in general, and then some studies ongoing in the lab, kind of preclinical, very early translational stuff that we have going on in the lab. So given that at least part of this meeting is a public meeting, I wanted to start kind of back at first principles as to, what are senotherapeutics and why are we interested in senotherapeutics. And as you can see here, as it says here, aging is the biggest single cause of loss of function, frailty, chronic disease, and death. Most of the common adult human diseases are diseases for which aging is the biggest single risk factor. Okay. We also know that aging is modifiable. Okay. So in other words, we can delay the onset of disease, and we can even extend lifespan. Okay. Now, opinions may vary as to which of those is most important, delaying chronic disease or preventing chronic disease or extending lifespan. Personally, I'm of the opinion that the primary goal is to delay or prevent the onset of chronic disease and improve the period of healthy lifespan. Now, senescent cells, the topic for today, these are a major cause of age-associated disease. We know that now. And these so-called senotherapeutics then are interventions, whether drugs or vaccines or CAR T cells, any type of intervention which can either modify the phenotype of senescent cells or kill senescent cells to prevent the deleterious disease-causing effects of these cells. Okay, so in other words, as you can see on the right, if we can target senescent cells with senotherapeutics, then we can delay or prevent the onset of age-associated disease. Okay, so that's the vision, and that's what we're going to be talking about today. So just a little bit more background then as to what senescent cells actually are. So senescent cells were first described as something that came about to proliferating cells, first described by Leonard Hayflick through in vitro studies on proliferating cells in culture. We now know that senescence is something that can also happen to post-mitotic cells. Okay, they can acquire a senescent-like phenotype. But anyway, when cells acquire various different types of molecular stress, whether that's shortened telomeres, activated oncogenes, DNA damage, that can initiate this very stable, perhaps irreversible exit from the cell cycle, and then importantly also associated with this inflammatory phenotype through the senescence-associated secretory phenotype, which is really a program whereby these senescent cells secrete various different inflammatory cytokines and chemokines, which can modify the behavior of neighboring cells and cause tissue inflammation and other things, okay? And also activation of interferon signaling through expression of these interferon-stimulated genes. So we know that senescence is has beneficial effects, okay? So it can be a potent tumor suppression mechanism, for example, by blocking cell proliferation, blocking cells progressing towards a cancer phenotype. And also the SASP, the inflammatory phenotype, can drive activation of the immune system to get rid of these senescent, potentially malignant cells. Okay, so senescence can be tumor suppressive, but we also know that senescence over the long term can also promote aging. Okay, and it does so, it's thought, by in part by blocking cell renewal, cell regeneration, and therefore tissue regeneration, but also this chronic inflammation that comes about as senescent cells accumulate over the long term within tissues. It can drive this chronic inflammation or inflammaging phenotype, and that chronic inflammation can in turn then go on to promote various different diseases of aging, including cancer. Okay, so it's a little bit perhaps paradoxical in the sense that in the short term, senescence is potently tumor suppressive, but over the long term, through this chronic inflammation phenotype, can also promote cancer. And so senescence has this dual role, okay? It has beneficial effects in terms of preventing cancer and also other things like promoting wound healing. But senescence also has these detrimental effects whereby it can promote tissue aging and disease. Okay, and so when we talk about Senotherapeutics, what we're really trying to do is to target these detrimental effects of senescent cells, to eliminate those detrimental effects of senescent cells to promote healthy tissue aging. So Senotherapeutics then can be thought of as, there's really two different classes, so-called xenomorphics. These can be drugs or other interventions which really modify the phenotype of senescent cells. So, for example, suppression of this inflammatory SASP phenotype to dampen down that chronic inflammation. Otherwise, there's also so-called Senolytics, which are actually drugs which kill senescent cells and therefore completely eliminate them from the system. Now, the challenge is that there's a number of challenges towards generating these Senotherapeutics. The first is that the senescent cells are quite rare, perhaps a few % of cells in aged tissues, and so therefore can be difficult to characterize. Secondly, senescent cells are not all identical. Okay, senescence is actually, we now think of it as really a spectrum of related phenotypes or so-called xenotypes. Okay, so a spectrum of phenotypes. And across that spectrum, some of those cells might be beneficial, as I've mentioned, and some of them might be detrimental. Okay, and so really what we need to try and do then to develop these so-called Senotherapeutics is to really define the beneficial and detrimental phenotypes of senescence, which are the good senescent cells and which are the bad senescent cells. And then we need to figure out ways to specifically target the detrimental senescent cells so we get rid of the bad effects, but without impairing some of the good effects. For example, as I mentioned, prevention of cancer and wound healing. So this is quite a challenge, and this is what we'll be hearing more about today. So I'm going to tell you a little bit about some of the things going on in my lab. First of all, in terms of, I'll talk a little bit to start with about what we're doing to really characterize senescent cells in young and old mouse tissues. Okay, and so what we're doing is, one thing that we're doing is trying to build up this atlas of senescent cells within aged mouse tissues. This is supported by the SenNet consortium in the US, which was a consortium put together by the NIH to support this really detailed characterization of senescent cells in old mouse and human and tissues. So as you can see on this slide, we're doing this in a few different mouse tissues, different mouse strains, different interventions. We're doing various different really state-of-the-art single-cell profiling technologies, so single-cell ATAC and RNA-seq, MERScope for spatial mapping, other single-cell mapping technologies. And then the goal is to put those together into a cell atlas together with the other members of the consortium, so that we can really figure out what do senescent cells look like in aged tissues, where are they, what's their molecular phenotype. And so just a little bit of the data that we've generated on young and old mouse livers. Here we've done 10x single-cell profiling. So this is ATAC-seq to profile the chromatin of the cells, and RNA-seq, so RNA and ATAC in the same single cells. And so using that, and these various different signatures of senescent cells here, for example, Sen-Mayo is a commonly used signature of senescent cells, and you can see that the abundance of these Sen-Mayo positive cells increases quite dramatically within these very old 30-month-old mice as compared to young 4-month-old mice. And these are just various other signatures of senescent cells that we work with in the lab. And you can see that we're seeing, by these various different signatures, an accumulation of senescent cells within the aged livers. This is looking specifically at the hepatocytes within the liver. Using MERScope, we can also do this spatially. Okay, so the 10x, which I just mentioned, depends upon dissociation of the cells, so we lose all the tissue architecture. But using MERScope, we can actually look at these cells in situ and see where they're localized within the liver. And what's interesting here is that what you can see is in the aged tissues, we see these clusters of senescent cells accumulating within the aged tissue. Okay, this is something that we've seen. Nicola Neretti, I think, was the first person to describe it, and I think Laura Niedenhofer's lab gave it the term sen spots. Okay, these spots or clusters of senescent cells which accumulate within aged tissues. So that's a little taste of some of the mapping that we're doing to really characterize these cells within aged tissues. I'm now going to talk a little bit about some of the interventions that we're taking in the lab to hopefully modify the phenotype of these cells to suppress their pro-aging qualities. And I'm going to talk about two interventions that we've been working on in the lab. Both of these are what we call drug repurposing studies. Okay, so these are taking existing drugs which are already either approved for use in humans or have been tested in humans, and we're trying to repurpose those drugs for application for healthy aging interventions. Okay, and so the two classes of drug I'm going to talk about are MDM2 inhibitors and cyclin D1 CDK6 inhibitors. And I'll start with the MDM2 inhibitors. So this is work that came out of some long-standing studies in the lab that we've had ongoing to really look at what is responsible for generation of this SASP, this inflammatory phenotype of senescent cells. And this goes back to work of Andrea Ivanov, a really fantastic postdoc in the lab when my lab was in Glasgow. And he described these so-called cytoplasmic chromatin fragments which accumulate within the cytoplasm of senescent cells. Okay, and he noticed that these were unusual because they are strongly positive for some markers of DNA damage, such as gamma H2AX, but not 53BP1. And he also showed that these fragments of chromatin come about by rupturing of chromatin through the nuclear envelope into the cytoplasm. Okay, and he called these cytoplasmic chromatin fragments. And so this has been an ongoing topic of investigation in the lab now for several years, a lot of it in collaboration with Shelley Berger's lab at UPenn. And so this is kind of the model that we have at the moment. This comes from a review that we wrote together with João Passos' lab. And basically, the idea is that dysfunctional mitochondria, through altered DNA damage signaling, can promote expulsion of fragments of damaged chromatin from the nucleus into the cytoplasm, these so-called CCF. And once in the cytoplasm, these CCF can react with the cGAS STING pathway, and so go on to drive expression of activation of NF-kappa B and the SASP and interferon stimulated genes. Okay, and so we think that the expulsion of these CCF into the cytoplasm has a number of consequences. One is, as I say, activation of SASP and ISGs. The other is as a way to remove damaged, potentially toxic DNA from the nucleus of the cell. And the other is as a vehicle for protein degradation in senescent cells. Okay, and so we've been interested in these for a while, as I say. So we did a whole genome screen to try and identify genes which were regulators of CCF formation in senescent cells. And we identified some genes which appear to inhibit formation of CCF, and then other genes which appear to promote formation of CCF. And turned out that two of these genes were highly related, P53 and MDM2. Okay, so if we inactivate P53, then we get many more CCF. If we inhibit MDM2, then we get many fewer CCF. And this was very interesting to us because there'd already been previous work published by Judy Campisi showing that MDM2 inhibitors, so basically small molecules which inhibit MDM2, are suppressors of the inflammatory phenotype, suppressors of the SASP. Okay, so we wondered do MDM2 inhibitors inhibit the SASP by blocking the formation of CCF? And so that was the idea that we went on to test. To do that, as I say, we started out using these MDM2 inhibitors. These are molecules which have already been tested in humans as potential cancer therapies. And so, as I say, we're interested in potentially repurposing these for healthy aging in humans. And so what these molecules do is that they obviously inhibit MDM2. MDM2 is in turn an inhibitor of P53. So these small molecule MDM2 inhibitors are well known to activate P53. And so this is work of Carl Miller in the lab, a really fabulous postdoc who's in the lab. And Carl went on to show that, yes, indeed, MDM2 inhibitors do block formation of CCF. They also potently inhibit expression of the SASP in senescent cells. Conversely, if we ectopically express P53, then that will prevent the formation of CCF. And if we do this by RNA-seq, we can see that there are a bunch of genes which are upregulated in senescent cells, these genes in red here, but then when we treat with the MDM2 inhibitors, those genes are downregulated. Okay, and so this cluster of genes is made up of many of these SASP cytokines and chemokines. And what's also interesting is that, as you can see here, knocking down MDM2, so getting rid of MDM2 will suppress formation of CCF in senescent cells. But if we do, treating with MDM2 inhibitors will block formation of CCF in senescent cells. But if we treat with MDM2 inhibitors in cells lacking P53, then the MDM2 inhibitors have no effect. Okay, so that tells us that the MDM2 inhibitors are going through P53. So this is an on-target activity of the MDM2 inhibitors. We went on to show that the MDM2 inhibitors work by promoting DNA repair, okay? So if we treat cells with MDM2 inhibitors, then we see a decrease in gamma H2AX, a well-known marker of DNA damage. Also, conversely, if we knock down P21, then you can see there's an increase in gamma H2AX. Interestingly, P53 also directly co-localizes at sites of DNA damage within the nuclei of cells. And more directly, if we inhibit MDM2 with the MDM2 inhibitor, then using the COMET assay, which directly measures DNA damage, then we can see that there's less DNA damage in cells where we treat with the MDM2 inhibitors. And also, if we use a reporter to measure DNA repair directly, this non-homologous end-joining reporter, we can see that MDM2 inhibitors enhance the efficiency of DNA repair in senescent cells. But perhaps the most direct illustration of this enhanced DNA repair is when we do single-cell genome sequencing. Okay, so here what we've done is to irradiate cells and then do single-cell genome sequencing. And you can see after irradiation that these senescent cells have a huge number of copy number gains and losses within their genome. I mean, their genome is essentially scrambled after irradiation. Okay, but if we irradiate and then treat with an MDM2 inhibitor, then their genome is largely normalized. Okay, so we think that the MDM2 inhibitor is activating P53, which is enhancing DNA repair and therefore maintaining integrity of the genome. And here you can see we've sequenced a bunch of cells, and in pretty much all cases except for this one outlier, the MDM2 inhibitors have returned the copy number of the cells back to the expected 2. Okay, so it's normalized the genome integrity of these cells. This is done in collaboration with Chong-Yi Chen at NCI. So then, moving in vivo, if we treat aged mice with MDM2 inhibitors, we can see activation of stabilization of P53 and upregulation of P21 as expected. Showing us that the MDM2 inhibitors are indeed activating P53 in vivo. And then when we do RNA-seq on those tissues, this is looking at mouse liver, you can see these are, what we see here is these are genes which are going up with age, but down when we treat with the MDM2 inhibitor. Okay, and conversely, genes that go down with age then go up with the MDM2 inhibitor. So this is an apparent rejuvenation of the transcriptome of these cells. And you can see this graphically here, okay? These are genes which are going up during aging, okay, but down when we treat with the MDM2 inhibitor, and conversely down with aging, but then up when we treat with the MDM2 inhibitor. So this appears to be a rejuvenation of the transcriptome of these cells. This is shown again here, more specifically looking at various different inflammatory markers. So inflammatory pathways upregulated with age and then dampened down by the MDM2 inhibitor. And also looking at specifically immune infiltration into the aged liver, we can see increased immune infiltration in the aged tissues, and then again that's dampened down when we treat with the MDM2 inhibitors. So suppression of inflammation and immune infiltration in the aged tissues. And then finally, this is quite preliminary, but we've also been treating mice with MDM2 inhibitors to see whether we can improve the function and suppress frailty of the old mice. You can see here that when that the old mice have relatively low function on the rotor rod. This is basically a measure of physical performance of these mice. The old mice compared to the young mice have low function on rotor rod, and it declines over the period of the experiment over 5 weeks. But you can see when we treat with the MDM2 inhibitors, then the function of those mice over that 5-week time span is maintained. And similarly, when we treat the aged mice with MDM2 inhibitors, we can prevent the increase in frailty of the aged mice. These are relatively modest effects at this point, but the experiment is ongoing, and we anticipate perhaps seeing more marked effects on longer-term experiments. So just to summarize then, I'll go through this quite quickly in the interest of time. we think that activation of p53 suppresses accumulation of CCF and therefore the SASP, the inflammatory phenotype of senescent cells. And we think it does so by promoting DNA repair and preventing the expulsion of this damaged chromatin into the cytoplasm of senescent cells. And if we treat with MDM2 inhibitors, then we can activate p53 and prevent the formation of CCF, prevent the inflammation, and indeed promote the function of these aged mice. Okay, so now moving on to cyclin D1 CDK6 inhibitors. This is another intervention that we've been exploring. I think you probably all know that cyclin D1 is best known for its role in promoting progression through the cell cycle. Okay, so it phosphorylates the RB protein, which promotes activation of E2F and then progression of cells through S phase of the cell cycle. Okay, that's the canonical, well-known function of cyclin D1 together with CDK4 or CDK6. So we were surprised when we noticed that cyclin D1 is actually markedly upregulated within non-proliferating senescent cells. Okay, so this is showing RNA-seq data. You can see that most proliferation-promoting genes go down in senescent cells, but there's this marked upregulation of cyclin D1 and cyclin D2. We can see that in other models of senescence by RNA-seq, so oncogene-induced senescence, replicative senescence, and we can also see it at the protein level by Western blot. Okay, so this was very paradoxical given that cyclin D1 is thought to promote progression through the cell cycle and senescent cells have exited the cell cycle. I should say that we are not the first people to note this, okay? This is an observation which is actually in the literature, even decades ago, but I think it's been kind of a long-standing mystery as to why cyclin D1 and D2 are upregulated in senescent cells. So Adarsh, a fabulous graduate student in the lab, set out to kind of investigate this and try and figure out why this might happen. And he took the approach of knocking down cyclin D1 and CDK4 and CDK6. Okay, and you can see the knockdowns here confirmed by Western blot, and then looked at what happens to senescent cells when he does that. And what he noticed was that if he knocks down cyclin D1 and CDK6, interestingly not CDK4, but knock down cyclin D1 and CDK6, there's a marked suppression of the SASP in these senescent cells, and also a marked suppression of various different ISGs, as shown here. We get the same thing when we treat with palbociclib, which is a drug which inhibits cyclin D1 CDK6. And you can see that here by RNA-seq analysis, a marked suppression of the ISGs and the SASP in senescent cells treated with palbociclib. And also a marked suppression of formation of CCF in senescent cells, which I've already told you are one of the triggers of SASP and ISGs in senescent cells. So I'm not going to go too much into the mechanism then of how we think cyclin D1 CDK6 is promoting formation of CCF in senescent cells. But what I'm going to talk about is some more of the in vivo studies that we've been doing to demonstrate the potential value of this. So I think you probably all know that ISGs are upregulated within aged tissues. This is a figure taken from a manuscript we have on bioRxiv at the moment, Aaron Havas in the lab. Many ISGs are upregulated within aged tissues. Cyclin D1 is also upregulated in aged tissues by both qPCR and Western blot. This is looking in old liver and also aged hepatocytes. And by spatial transcriptomics on the CosMx platform, it looks as if, here looking at this Sen-Mayo signature of genes, that these aged cyclin D1 positive hepatocytes are strongly positive for Sen-Mayo. So we think that cyclin D1 is upregulated in senescent cells in aged hepatocytes in vivo. So then we're interested in what happens if we inhibit this pathway in tissues in vivo. And so the first approach that we've taken here is using a genetic knockout. So we use an AAV8 with a TBG promoter. So this specifically infects the hepatocytes of aged liver. And then we have an SA-Cas9 and a guide RNA, and that allows us to do very efficiently a cyclin D1 knockout within the livers, within the hepatocytes of these young and old mice. And you can see here that when we do a cyclin D1 knockout in vivo, we do see marked suppression of expression of the ISGs in vivo. And same when we treat with palbociclib, and we get a marked suppression of the ISGs in vivo. And then finally, so then inhibiting cyclin D1 either genetically or pharmacologically is suppressing the expression of the interferon-stimulated genes in vivo. Again, we wanted to know, does that perhaps have any functional benefit in these aged mice? And so here what we've done is the same kind of experiment as before, where we take young mice and old mice, put them on a rotarod. The young mice perform well, the old mice don't perform so well, but we treat with the MDM2 inhibitor. Sorry, this is this is palbociclib, a cyclin D1 CDK6 inhibitor. You can see we treat with palbociclib, and we see improved function on the rotarod, and palbociclib also arrests the progression of frailty in these aged mice. Okay, so again, seems to be beneficial for function and frailty in aged mice. I should say that these are experiments which we've been taught how to do by João Passos at the Mayo Clinic. João is a good collaborator on many of the things that we do. Okay, so finally then what we think is happening here is that through this pathway, cyclin D1, CDK6, through mechanisms that I haven't really discussed, we think is promoting DNA damage, formation of CCS, activation of the cGAS-STING pathway, expression of ISGs, and so tissue damage. And what palbociclib does is it inhibits cyclin D1, CDK6 and blocks this pathway, and therefore prevents the accumulation of damage in aged tissues. And so just finally, then, I just want to summarize what I've said is that, this repurposing strategy where we can take small molecules which are already being tested or approved for use in humans and perhaps repurpose them through their ability to modulate other pathways specific to aging, we can suppress this chronic inflammation phenotype associated with aging and perhaps promote healthy tissue aging and suppression of disease. And so ultimately, we think these small molecule interventions can promote healthy aging, suppress the onset of chronic disease for substantial medical, social, and economic benefit in aged populations. And so just to give my acknowledgements then, the SENNET studies I mentioned, there's a large team of investigators involved in that, but myself and Bing Ren lead the San Diego part of the consortium. Alan Wong and Chuan Zhu are two key collaborators within San Diego on the SENNET consortium. Carl Miller led the work on P53 and MDM2 inhibitors that I described, and Adarsh and Aaron led the work on cyclin D1 CDK6 inhibitors. And so with that, and I also, again, want to really thank the funders and other institutions who support what we do. And so thank you very much, and I'll take questions if there are any.
Any question? Resu? Okay.
Hi, Peter. Very nice talk. So I have two questions. The first on the first part on the MDM2 inhibitors. So these cells look very healthy, genomically speaking. So why are they not re-entering cell cycle? If you remove the drug, then P21 probably goes down. So perhaps they might re-enter the cycle.
Yeah, I mean, we're specifically activating the P53 pathway. Mm-hmm. I mean, as you know, that in itself is actually going to promote cell cycle arrest anyway. But
after you remove it, I'm saying, because they're gonna.
Yeah, I guess it depends. I mean, I think there's more to senescence than simply a chronic DNA damage response. Okay, so I think I think there are mechanisms which are engaged, for example, in terms of perhaps chromatin epigenetic reprogramming, metabolic reprogramming, and these mechanisms can be stable and contribute to the stable cell cycle arrest. So I don't think just simply eliminating the DNA damage is going to be sufficient to get those cells back into the cell cycle. Although I know there's great work from some people who say that that can contribute, but I don't think DNA damage is the whole story.
And regarding your second story and connecting to the first one, so we believe Palbo actually is active only via p53. So I wonder whether you have checked if there is something that actually also connects your p53 story to the effect of Palbo and the reduction of CCFs.
Yeah, no, we haven't looked at that, and I don't know whether we haven't asked whether the effect of Palbo in senescent cells is dependent on p53 or not. That we should do that experiment. Thanks.
And over here. And I'll go with a general question while we wait for Magford to arrive. So when you treat cancer, you can be aggressive and don't care too much about side effects because alternative is cancer.
Yeah.
With senescence, I guess, it's not quite like it. So what can we do to take care of this problem?
Right. So as you say, the bar for this is one of the challenges in the field, because the bar for interventions to promote healthy aging is a lot higher than the bar for treating cancer. Okay, I mean you obviously in relatively healthy aged people, we cannot tolerate toxic side effects of any of these drugs. Okay, so that is it means that the drugs have to be specific and with minimal side effects. And we also have to be cautious about how we try to translate these drugs into the clinic. Okay, so I'm personally, I'm of the view that for these types of healthy aging interventions, we should start out, and this is the model taken by Jim Kirkland and others with Senolytics, is to first of all test these drugs on pre-existing pathologies of aging, chronic kidney disease, rheumatoid arthritis, etc. Okay, see whether they have any benefit in those models. Are the side effects, if any, tolerable before perhaps even thinking about rolling them out more generally.
Thank you. And oh yeah,
from the back. Wonderful talk.
Thank you.
I have a naive question. Has the level of histone acetylation any influence on CCF?
Good question. So we know that CCF are deficient in histone acetylation. So they're primarily enriched in so-called gene silencing modifications like H3K27 trimethylation and H3K9 trimethylation. They are depleted of histone acetylations. Whether or not by increasing histone acetylation with, for example, with histone deacetylate, and we do actually find that HDAC inhibitors, histone deacetylase inhibitors, HDAC inhibitors, which increase the level of acetylation, do block the formation of CCF. We don't know whether that's through their effect on histone acetylation. We have reported that it's working through effects on mitochondria function, which is a whole different story. So I don't know whether histone acetylation directly is affecting CCF formation, although it's possible.
Peter, can you tell us a bit more about the paradox of cyclin, D-type cyclins in senescence? What drives their upregulation? Are these functional D-type cyclins? Do they rely on stoichiometric levels of P16 not to re-enter the cycle? It's the chromatin context what prevents them to move into S phase. Can you tell us more?
Yeah, so we think that we haven't shown that senescent cells have cyclin D1 associated kinase activity, okay? But we do think it's a kinase dependent function, okay? Because it also depends on CDK6 and it's inhibited by palbociclib, which is a kinase inhibitor, okay? But we haven't formally shown that there is kinase activity there. On the other hand, RB is unphosphorylated in senescent cells. Okay, so we think that there is probably a substrate involved. So cyclin D1 CDK6 is phosphorylating something to inhibit DNA repair and therefore promote the accumulation of damage in CCF. So it's phosphorylating something, but it's not phosphorylating RB.
Okay.
So we think its substrate specificity is redirected in a senescent cell. In terms of what's upstream of cyclin D1, we don't know. We've done some kind of, mapping of enhancer promoter contacts, and it looks like in senescent cells, cyclin D1 has more potential promoter enhancer contacts, but we haven't functionally investigated any of that.
Yeah, we have time for only another question. Sorry, we are a bit late.
Sorry.
Yeah.
So, very nice talk.
Thank you.
So, in the past few years, there are some paper come out reported that except the CCF release, there's also the mitochondrial, even the mitochondrial DNA, even the mitochondrial RNA release to the cytosol. So, do you have any comment to relate to that? And another question is, instead of use the MDM inhibitor, do you think directly target the downstream of this nucleic acid synthesis pathway, or target or enhance the nucleic acid degradation machinery could be more effective way?
Okay.
Thank you.
I'll take. I didn't quite catch the second question, so in the interest of time, I'll just take the first question. I'm happy to chat about the other one in the coffee break. With regards to, as you point out, João Passos at Mayo has shown that in senescent cells, mitochondrial DNA also leaks into the cytoplasm. John Sedovy at Brown has shown that line 1 retrotransposons get expressed and cDNAs get into the cytoplasm. Okay, so we are very interested in the potential crosstalk between these pathways, and currently we've put together a collaborative grant proposal led by João Passos to try and investigate the specific crosstalks and mechanisms, interconnecting mechanisms. But I don't really have much more to add at the moment, other than we're very interested in that. Yeah, thanks.
Yeah, and thank you again, Peter, for a very interesting presentation. And
good morning, everyone, from my side too. So I'm Nina Cabezas-Walscheid, a new appointed professor at the ETH in Zurich. And it's my great pleasure to be chairing this session on basic biology of senescence. And our next speaker is Vassilis Gorgoulis from the University of Athens.
I would like to thank you for the invitation. To talk about senescence and targeted senolysis. The reagents I'm going to discuss are all under patent. Our group is particularly interested into how cells react to oncogenic stimuli in order to preserve homeostasis. Based on the fact that most cancers demonstrate genomic instability, we hypothesized that the DNA damage response pathway could play such a role, that of a reacting force against oncogenic threats. Based on a series of studies we conducted, along with backup applications from Fabrizio, we proposed together with the groups of Thanos Halazonidis and Giri Bartik the following model of oncogen-induced DNA damage for cancer development. According to these models, activated oncogens compromise proliferation, triggering replication stress, causing the double-strand breaks, which activate the DNA damage response pathway. This is the reacting force, setting in motion the anti-tumor barriers of apoptosis and senescence, eliminating incipient or neutralizing incipient cancer cells from the earliest stages of cancer development. However, as DNA damage accumulates, error-free repair is shifted to error-prone, fueling genomic instability, which eventually breaches the anti-tumor barriers, leading to cancer progression. Now, as a result, the DNA damage response, replication stress, and senescence now are considered hallmarks of cancer. Our model can explain the following things. First, emergence of genomic instability in cancer. Second, evasion of apoptosis by mutating key tumor suppressor genes like p53, early activation of senescence, and also bypass of senescence, which means blocking senescence entry. However, an important question emerges. Could it explain escape from senescence? In other words, once senescence is established, can it lead to its exit? Now, before addressing this question, we need to see two things. What is senescence and how to detect it accurately? Now, as Peter told, senescence is a stress response characterized by 4 key features: cell cycle arrest, deregulated metabolism, macromolecular damage, and a secretory phenotype called SASP. The senescent cells are resistant to apoptosis, and the question mark refers to data, including evidence from our lab, that under certain circumstances, senescent cells can re-enter the cell cycle, and this has important clinical repercussions, as I will discuss later. In brief, senescence has a bright and a dark side. In short term, it has a beneficial role preserving homeostasis. However, on a long-term basis, if not timely removed by the immune system, it has detrimental effects. Now, if not detected accurately, misleading conclusions can be drawn. This is just an example from a recent paper, publication in a very high impact factor journal, where the authors conducted a transcriptome analysis from CD4 T cells from their knockout model, and based on a series of transcripts which are commonly found in SASP, they decided to assign these cells as senescents. However, they failed to see in the lower part of their heat map that Ki67, a key proliferation marker, is high, which contradicts with the hallmarks of senescence. And this is just one example of misinformation published. Now, to avoid such cases, members of the senescence community, including ours, have proposed a 3-step multi-marker algorithm for accurate senescence detection. In step 1, screening, assessing S-beta-gal activity or detection of lipofuscin, the dark matter of the cell. In step 2, verification with additional markers, positivity for the cell-dependent kinase inhibitors P21 and P16, absence of proliferation markers, and low expression of lamin B1. Step 3, provisional, if staining with markers if the cell of origin is required. As you can see, lipofuscin is a key feature of this algorithm. What is lipofuscin, or else dark matter of the cell? Is a metabolic byproduct, an oxidized lipoprotein complex, and it reflects deregulated metabolism and macromolecular damage. Recently, in collaboration with Demaria, we showed that deficient translation participates in lipofuscin build-up. A key feature of lipofuscin is its autofluorescence. It is diluted only via cell division. And also, it's the only constant feature of senescence, irrespective of cell senescence type. Our group is particularly invested time in developing reagents to detect lipofuscin. We developed 2 reagents, GL13 and GLF16 in 2 variants. With GL13, the pro of GL13, it has the ability to detect senescent cells in fixed clinical tissues, which is the bulk material of pathology labs, a feature which is not possible with S-beta-gal, because S-beta-gal is applicable only in cell culture and frozen tissues. However, in inexperienced hands, because it's diluted in ethanol, it can lead to background. So we evolved GLF16, which has the property to be diluted in aqueous solutions, and when encapsulated in a nanocarrier, it has the ability to detect and isolate live senescent cells. By applying GL13, we and others demonstrated senescence in a wide spectrum of clinical to experimental settings. I will show you in the next few slides some examples. One of the first clinical entities to harbor senescent cells was Hodgkin lymphoma. As a molecular pathologist, I was convinced for many years that Hodgkin lymphoma was linked with senescence because the key histological feature of Hodgkin lymphoma, the red cervix cells, are reminiscent of senescence. They are large, multinucleated, and they are embedded in inflammatory milieu, which is reminiscent of SASP. However, at the time, I lacked the tools to prove it until we developed GL13, confirming our hypothesis. Moreover, when we analyzed a set of Hodgkin patients, we observed that the patients with higher percent of senescent cells had worse response and worse outcome, a finding which I will explain later. Another entity, COVID-19, we showed, exploiting antibodies against the virus that we developed in our lab, we showed, applying the algorithm, that a percentage of SARS-CoV-2 infected alveolar type 2 cells exhibited features of senescence. And here you can see, on the right panel, co-staining of GLF13 with P16. Confirmation of the infection was performed by electron microscopy, as you can see in the lower part of the panel. Moreover, we demonstrated that the infected cells that demonstrated senescence could be a source of SARS-CoV-2 mutagenesis because the senescent cells show increased expression of the ApoBec mutagenic system. Recently, we showed also that giant cell arteritis was linked with senescence. Giant cell arteritis is an autoimmune disorder affecting mainly women and the medium-sized arteries. And the source of senescence was fibroblasts, macrophages, and endothelial cells. And again, applying the algorithm, we saw that all GL13 positive cells were also positive for H21 and negative for H67. GLF16, our latest reagent, robustly identified senescence in vivo and in vitro. On the left, this is an inducible P21 senescence model where you can see clear colocalization between GLF16 and P16. And on the right, a palbocically melanoma senescence model where you can see clear mutual exclusive pattern between GLF16 and Ki67. One of the most important features of our new reagent is that it can allow flow cytometric analysis using multiple markers. Here is an example by applying GLF16 in an inducible P21 senescence model. You can see that gated GLF16 cells were co-stained for P16 and Ki67, and in the lower left part, you see that this population, GLF16 positive, P16 positive, and Ki67 negative, was subsequently analyzed at the transcriptome and chromatin level, clearly demonstrating a senescent signature. When encapsulated in a micellar nanocarrier, it can allow detection and isolation of live senescent cells. How the nanocarrier works, once it enters the senescent cells, the nanocarrier is dissolved by the low pH of the senescent cells, and then the released reagent binds to lipovskin, lighting the senescent cells. Subsequent sorting of the senescent cells, we can allow subculture of live senescent cells and subsequent downstream molecular analysis. Similar results were shown ex vivo using patient-derived live organoids, and you can clearly see where senescence was induced by hydrogen peroxide, and you can see in the non-senescent organoids the mutual exclusive pattern between GLF16 and Ki67, high Ki67, low GLF16, and once senescence is induced, the reverse. The most important feature of a GLF16 when encapsulated in the nanocarrier, it's its ability to enter senescent cells in live organisms, allowing real-time monitoring. Here you can see two examples. using two models, a palbociclib melanoma senescence model and a bleomycin fibrosis senescence model, where you can see intravenous administration of MGLF16 detects the senescence-rich tissues and lights them. Note that the liver in both cases is lighted because the liver is the route of excretion. Senescence was furthermore analyzed by performing a detailed histopathological analysis. So back to our initial model. Could it explain escape from senescence? Indeed, with the tools we developed, in 3 consecutive studies, we indeed showed that escape from senescence exists. And in the last study, we showed that genomic instability is at least one route which can lead to escape. Particularly, we used an oncogene-induced senescence model using a CDC6 as an oncogene. And once we activated, we observed that it led to 2 breaks in the short arm of chromosome 3, inverted the region, and activated the Kirklandian release factor BHLH40, which set in motion the escape route. So this is a video where you can see after a prolonged period of senescence, after 30 days, the first colonies in the red cycle, you see a senescent cell. And after a while, you can see in the red cycle, in the green cycle, the first colonies of escape cells dividing. So we updated our model, showing that genomic instability can lead to chromatin remodeling, unlocking cell cycle reentry programs leading to escape. Based on these results, we suggest that cancer therapeutics should be modified. Why? Because conventional therapeutic strategies target mainly the proliferating fraction of cancer. So we believe that patients could benefit if we add to the conventional therapeutic strategies senolytics. And thus, we target both populations, proliferating and the senescence. And this increases the probability of tumor eradication. As a proof of concept, conducting a pharmacological screen to find agents that could kill LKB1 null lung adenocarcinomas. LKB1 is an important metabolic activator. It's the 3rd most frequently mutated gene in lung adenocarcinomas. We identified 2 targets, tirfostin 23, which is a tyrosine kinase inhibitor, and pisenatol. Both these factors bind to the E3 ligase von Hippel-Lindau, setting in motion a HIF-1 leptin energy exhaustion pathway, leading to cell death. However, not all LKB1 null cells are died. There's a fraction of cells that acquire the senescence phenotype. This is a classical example of therapy-induced senescence. So if we leave this fraction untreated, we actually leave behind a source of relapse. So applying a very well-established senolytic scheme of dasatinib quercetin, we dramatically reduced this senescent population, reducing the risk of relapse. Notably, the senescent population was observed only in the LKB1 null cells. So, what is senolysis? Senolysis and senolytics is the selective elimination of senescent cells, and they mainly target the anti-apoptotic pathways which are activated in senescent cells. However, most senolytics are derived from drug repurposing procedures, and they are followed with a lot of side effects. So how can we achieve targeted senolysis? So we had the following idea. We exploited the lipofuscin binding domain of our reagent and connected it with the senolytic of our choice, linking it with a cleavable linker, and then encapsulating it in a nanocarrier, a stealth nanocarrier, which actually does not allow interaction with the the immunoglobulins and the albumin of the plasma. And following this procedure, we generated the senolytic MGL392. So this senolytic MGL392 binds specifically lipofuscin, and as I mentioned, lipofuscin autofluorescence. So by treating our models with MGL32, it led to quenching of lipofuscin, meaning that it binds specifically to lipofuscin. When we treated the cells with cytohalacin, which is a actin polymerization inhibitor, quenching was blocked. So we have specific binding of lipofuscin. Then we compared the effect of MGL392 with dasatinib. As you can see, the selectivity index of MGL392 was significantly higher than free dasatinib. Selectivity index is the ratio of toxic dose to effective dose, and it's at least 9 times higher, which means that although both targets, both reagents kill senescent cells, only MGL392 spares the non-senescent population. This is shown in vitro using 2 of our in vitro senescence models. Ex vivo, in patient-derived lung organoids, you can clearly see here that dasatinib kills non-senescent cells, which is and spares the MGL392 spares them. In vivo, similar results. Both reagents eliminate the senescent population. However, only MGL32 spares the non-senescent cancer cells. And in the low right panel, you can clearly see the effect of valbociclib. Finally, doing a histopathological analysis of sacrificing the animals treated with dasatinib and the animals treating with MGL392, we did not see any pathological lesions in the mouse group of mice treated with MGL392, whereas the mice treated with dasatinib demonstrated there was a clear evidence of myositis. So, how the drug works? Once it enters the senescent cells via endocytosis, the low pH of senescent cells disables the nanocarrier, the drug is released, it binds to lipofuscin, the enriched environment in esterases cleaves the esteric bond, releasing the drug, which binds to its targets, leading to senolysis. So, our future plans is to develop a radiotracer to detect senescent cells in humans, because our reagents, because they have fluorescent moieties, are suitable only for small animals. So we want to develop a radiotracer similar to fluorine dextroglucose, which is used in everyday clinical practice to detect tumors using PET-CT. Towards this direction, we already have developed a cold analog, which clearly can show that it can bind and quench lipofuscin in our models. So with this, I would like to thank all members of my group for their hard work and commitment, especially my 2 postdocs, Sofia Maguta and Dimitris Veroutis, which played a major role in developing the reagents in the drug, and all my collaborators. I will thank you very much for your attention, and I'm open to questions.
We have now time for some questions. So, Kiri, we have one. Here. The microphone is coming.
Just a second.
It's coming.
So my understanding is that lipofuscin is mostly in lysosomes, but lipofuscin is free?
Lipofuscin is in the cytoplasm. In senescent cells, it's always in the cytoplasm. In the senescent cells, we have disrupted lysosomes. This is why we have always pH low.
All right, interesting. So it kind of leaks out of lysosomes?
It does not only leak out, but it actually, lipofuscin is not an inert material, it's a bioactive material. It's a result of macromolecular damage it's a vicious cycle. It oxidizes proteins and lipids around and it aggregates. As a medical student, I read first time about lipofuscin in Robbins, it was discovered 3 centuries ago, and nobody paid attention. And it's the only constant and key characteristic of senescent cell. Any type of senescent cell that I activate, I always see repurposing. So it's not in the lysosome, it's in the cytoplasm.
Interesting. Thanks.
There is another question
sorry.
there. Please. Also there.
There is a question.
Here.
Yes, ma'am.
Vassilis, thanks for the nice talk. So, of course, when we talk about cancer cells, everything becomes more complex. Also, in terms of like lysosome, So, are cancer cells also having some leakage of lipofuscin? And second, would it not even be better to develop something that can target both senescent and non-senescent cancer cells, but spare non-cancer cells? Because that would be probably next level of like killing all the cancer lesion and separate. Because now you were saying, well, we have these cancer cells that when they're not senescent, they survive when I apply but then maybe it's even better to kill them, right? You were to spare the normal ones.
This is a very, very, very good question. However, in cancer, a lot of routes, all roads lead to Rome. So we have a lot of pathways. So if you block one pathway, something else comes. And this is very evident in the DNA damage response pathway. We block one pathway, another pathway comes out. And this is why I mentioned we use 2 strategies. Use the conventional and the development of the conventional pathways plus senolytics, or in certain cases, xenomorphics. For example, you don't want, we see a lot of times senescence in endothelial cells. We don't want to eliminate senescence endothelial cells because the proliferation rate of the endothelial cells is very low. So if you kill the senescent cells in various disorders like obstructive pulmonary disorders, you leave gaps. So you need to do a combination of traditional therapy, senolytics, xenomorphics, and all this comes down to precision medicine. So we need to treat not the disease, but the patient. So I agree, but...
What about lipofuscin released by lysosomes of cancer cells? So because, there is a lot of lysosomal stress in many cancer cells.
We always use rigorous controls, and we have seen no detection of this thing.
Vassilis, do you observe any overall reduction in the in vivo treatments of inflammation, for instance? Inflammation. Do you observe in vivo treatments an overall reduction of inflammation?
So now we are conducting the preclinical... Oh, sorry, yes. At this point, we are in the phase of doing the preclinical portfolio of the drug. Our preliminary results shows only with free dasatinib we have this myositis. We did not see in 8 weeks of treatment any effect, but this remains to be shown.
Okay, thank you very much. If there are no further questions, we would like to thank Vassilis again. Our next speaker is Jesus Hill from the Imperial College of London, and he's going to talk about cellular senescence as a therapeutic target.
So thank you. Thank you. I'm going to talk about specifically SMARCA4 and how it regulates NK-mediated killing of senescent cells. So first, I want to thank, of course, IPSA and the organizers for the invitation. And I want to also thank the previous speakers because they have introduced many of the key concepts of senescence that I need. But I'm just going to go about a couple of things. So one is very clearly senescent cells accumulate in many different diseases, not just in cancer or fibrosis, but in an increased list of diseases that are related with age. And we have this paradox that some of the previous speakers have talked about, that senescence, despite having this obvious detrimental and pathological role, has also a protective role in cancer, fibrosis, and in other diseases. How we put together this is just depicted in this model that is adapted from work from basically from Daniel Muñoz and Manuel Serrano. And what we see is that when there is damage in the tissue, so there is initially this damage causes senescence, and the senescence, what it does is a double response. On one hand, intrinsically arrests these damaged cells. On the other, causes this SASP, this inflammatory immunomodulatory phenotype that recruits the immune system and is able to eliminate these senescent cells. So extrinsically also eliminate these damaged cells. So then the tissue can regenerate, and this transient induction of senescence in general is positive, contribute to normal development, proper wound healing, tissue regeneration, tumor suppression, and limit fibrosis. And one of the things that makes senescent cell detrimental is the persistence of senescent cells, when rather than this transient effect on senescence, so we have this accumulation of senescent cells, and this accumulation of senescent cells is detrimental. So it causes tissue dysfunction through SASP-mediated chronic inflammation that disrupt the tissue and contribute to age-related pathologies, including, paradoxically, for example, tumorigenesis. And of course, there are differences between the detrimental and the beneficial senescent cells, but one of the key things is this persistence. And knowing that senescent cells, when they accumulate and they persist, are detrimental, so we want to see how we target it. And that's what we are just dealing with today in this Senotherapeutics. And as the previous speaker mentioned, there are many ways that we can just do that. So for example, we can delay the entry into senescence, and that can be used in some settings, or rather, like the SASP contribute to this chronic mediated inflammation, to this detrimental effect, we can just also inhibit this SASP. So I'm going to show you just a couple of examples of my lab. So in my lab, we use like functional screens to identify using drugs or genetic screening way to modulate senescence. So for example, this is from a study in which we look into ways to try to delay the entry into senescence, and we identify a drug, 3DA, that is able to delay the entry into senescence, and we thought that can be used, for example, to treat ex vivo cells that we are going to engraft, and then increase engraftments by delaying by decreasing senescence that limit this engraftment potential. And this can be used, for example, for the hematological transplants and others. Other of the things that we have studied in the lab is ways to try to target these inflammatory cells, or what we know as xenomorphic. So we have performed different screens, and we have identified regulators specifically of the inflammatory SASP that then maintain other aspects of the senescence phenotype, and that they can be used to try to target these detrimental effects of inflammation in senescence. But of course, the way in which most people are targeting senescent cells and is most attractive for different reasons are senolytics. And senolytics are, as introduced, drugs that can basically selectively eliminate senescent cells. And some of the advantages that they have is that they can be used intermittently. They have been shown as proof of principle with genetic models to improve many different pathologies, including increasing lifespan and health span, and being able to treat multimorbidities. So our lab has tried to look into senolytics in different ways, and I'm just going to show you like very quickly here like 3 of the examples of things that we have tried to do. So for example, we have gone through use hypothesis-driven approaches, and this is something similar to what Vassilis was telling before. So we know that senescent cells have high levels of this beta-galactosidase activity, and we hypothesize, as others, that if we just generate galactose-derived prodrugs that are basically inactive until the point that galactose is cleaved from these drugs will be selectively cleaving senescent cells and will selectively kill the senescent cells. So that's for example, one approach, hypothesis-driven approaches. We have also done functional screens, and we have both done screens in which we have repurposed drugs, and we have, for example, identified cardiac glycosides, or genetic screens in which we have sampled the full genome or the druggable genome and identify vulnerabilities of senescent cells that can be targeted specifically. But one thing that we see is that for whichever disease, and the same for senolytic discovery, so we have the whole genome, or more clearly even the whole proteome, and there are a subset of genes or proteins that are involved in the disease. If we go for a hypothesis-related or a hypothesis-based approach, we're going to just be able to target a very small subset of this, and yeah, this is as good as hypothesis you can generate, but probably it's just like a bunch of those. You can just go and try to repurpose drugs, and this has the advantage that are drugs that are like tested, that we know that which are the side effects, and that there are many patients that have used these drugs. But still, there is just a subset of these drugs that are probably in the 1,000, and they're just probably just targeting hundreds of targets. And this is just a small subset of what we can do. On top of that, druggable targets are receptors, enzyme, ionic channels, something that have an active motif that we can just target. But there are many of the other disease-related proteins that are classically undruggable. They lack a pocket that we can just target with some of these classic drugs. Basically, they function through protein-protein interactions. They have active sites that are highly conserved and that are difficult to target specifically, or we don't know even the 3D structures. And one thing that we can do, of course, is just to do CRISPR or RNAi and try to target the whole genome, and we can identify some of these targets, but still it's difficult to go from there to try to have drugs. So we think that a potential sweet spot for try to identify more senolytic targets is try to exploit covalent inhibitors. And reversible inhibitors, they are binding in a reversible way, they have to fit a pocket, and are some of these classic drugs that we talked before. Covalent inhibitors, on the other hand, they have a part of the molecule that we call the warhead that can form a covalent bond with the molecule, with a target molecule, can bind anywhere in the molecule with some restrictions. For example, there are some of these that are just targeting cysteines, and they can target many different kinds of proteins. When they bind, they don't need to target a enzymatic pocket, but they can also destabilize protein-protein interactions and do a few other things. And one classic example of covalent inhibitors expanding the druggable proteome are like the Ras G12C covalent inhibitors. And these covalent inhibitors have been known for centuries, but they have a resurgence now that is exemplified, for example, by these Ras G12C covalent inhibitors. So one thing that we have been trying to do in the lab, and I'm not going to talk about this now, but there is a poster and a short talk by Maria in my lab, is try to use a library of covalent compounds to try to identify novel senolytics. And one thing that this also allows us, beyond expanding into the undruggable proteins, is to do co-discovery of starting chemical matters and new targets. And I think that this is very exciting, and Maria will show a poster and we'll talk about this later today. But what I'm going to talk in the rest of the talk is about another modality of trying to target senescence. We talk about xenomorphics, we talk about senolytics, but the other way that we can try to target senescence is by enhancing this immune-mediated killing of senescent cells. That is the natural mechanism that the body uses to try to eliminate these senescent cells. And the immune system eliminates senescent cells in many settings. For example, in the context of anti-tumor therapies. So there are like different immune populations that basically target are targeting these senescent cells. There is also immune cells that target, for example, premalignant senescent cells, also initiating fibrotic cells, and in the context of aging. And one common immune population between all of that are natural killer cells. So natural killer cells are able to eliminate or contribute to the elimination of all of these senescence populations. So what we know is that, for example, when a cell enters into senescence, they start secreting the SASP, and something that makes them a target of the immune system is a combination of these immunomodulatory chemoattractant chemokines, but also, for example, in the context of NK cell, the induction of activated ligands that are going to recruit these NK cells. And for example, in this context, NK cells and also other immune cells are attracted, and they are able to eliminate these senescent cells. So that's something that naturally happens. And what we thought is that if we can transiently enhance this chemoattractant SASP, we could enhance this elimination of this attraction of NK cells and improve the clearance of senescent cells. And I want to highlight that we want to use this in a similar way to senolytics, intermittently, so in a way that we can use it transiently, and then this is sufficient to attract the immune system and eliminate the senescent cells. So we don't have to do it persistently. So for doing that, we thought that we could do it in different ways. So first, we could basically conduct screenings to identify siRNAs that are able to superinduce the SASP. Then amongst those, try to use the ones that are able to enhance the NK-mediated elimination of senescent cells using models of co-culture of senescent cells and immune cells. And eventually, we will want to test whether these compounds or these strategies can enhance the NK-mediated clearance of senescent cells as anti-cancer therapies. So I'm going to go like a step by step into what we did. So initially, as we mentioned, we were interested in seeing whether we can identify ways in which we can enhance the SASP, superinduce the SASP. So for doing that, so we conducted a screening in which we use as readout IL-6 and IL-8 that are induced, in this case, in a model of oncogene-induced senescence. And we have a couple of controls here, this ALPAR2 and SIRT1, that when we knock them down, we superinduce the SASP. And this is basically depicted here. So this is the non-senescent cells, the IL-6 levels. Once we induce senescence, this is increased. And with our positive controls, this is what we call superinduce. So we basically conducted a screening for druggable, an siRNA screening for druggable targets that could superinduce the SASP. We identify a few that commonly upregulate the expression of IL-8 and IL-6. So we deconvoluted this screening by basically using 4 independent siRNAs against it. And we used also 4 different readouts, not just IL-8 and IL-6, but also CCL2 and IL-1 beta. So we have an idea of how each one of these hits are regulating the different SASP ligands. So this is, for example, the furin-1, that is one of these 3 hits that when we knock it down, we superinduce the expression of these 4 SASP ligands. And we can see that, indeed, when we use like 2 different siRNAs against furin, we see increased levels of IL-6, IL-8, IL-1 beta, and CCL2. So what we did is we took basically siRNAs against things that were inducing at least 2 or 3 of these ligands, and also we took the ones that were inducing CCL2. So we took a total of, I think that was like 76 or 78 of these, and then we used those to conduct a screening in which specifically we asked the question using co-culture models or whether knocking down these genes in senescent cells resulted in an enhanced mediated clearance of these senescent cells by NK cells. So for that, we have to establish a system in which we have like basically a co-culture of senescence or non-senescent cells or senescent cells in the presence of different amounts of these natural killer cells. So what we can see is that when we increase the effector to target ratio of the NK natural killer cells, they kill the senescent cells specifically. We can quantify that. And they specifically kill the senescent cells in an effector to target ratio manner and spare the non-senescent cells. So from that, what we selected is an effector to target ratio 2, so that we could see an increase when we knock down some of our genes. And we conducted this screening, and basically these are the results of some of the things that we saw that enhanced the NK-mediated killing of senescent cells. So we identified 3 genes here. So one was furin, the other was a subunit of polymerase 1, and the other was SMARCA4, that when we knock them down, selectively enhanced the NK-mediated killing of senescent cells. And we concentrated in SMARCA4. So what we thought that SMARCA4 is interesting because it's a component of Swyer-Smith complex. So it's a chromatin remodeling complex that is commonly mutated in cancer. And importantly, also, there are like efficient drugs, including PROTACs that degrade SMARCA4 that can be used to target SMARCA4. So this is, for example, the result that we achieved using a SMARCA4 PROTAC. So I want to mention a couple of things. So one is that when we use this PROTAC, we don't enhance the NK-mediated killing on non-senescent cells. So basically, despite of adding the PROTAC and adding NK cells, the senescent cells are spared. But we increase, and this is quantified here, so this is the killing of the senescent cells by NK cells, and this is the increase that we achieve once that we treat these cells with this PROTAC that targets SMARCA4. So one thing that I want to mention is that this NK-mediated killing is specific of senescent cells. The other thing that I want to mention is that this elimination of senescent cells depends on the presence of natural killer cells. So this SMARCA4 does not have a senolytic or not a strong senolytic effect per se, but it needs of the presence of NK cells in order to eliminate the senescent cells. So this I saw you using a NK cell line, so NK92, that is an immortalized cell line, and IMR90 ERA cells, but it also works in other models of senescence. So, for example, inducing senescence with chemotherapy, inducing senescence in models of ovarian cancer, and also not just with this NK immortalized cell line, but also with primary NK cells, both derived from cord blood or derived from peripheral blood. So how does it work? So SMARCA4 inhibition, as we expected, what it does is superinducing the SASP. So we mentioned before that there are two things that senescent cells do in order to be targeted by NK cells. One is upregulate NK-activated ligands. So we can see between control and senescent cells, NK-activated ligands are upregulated, but there is no further upregulation when we treat with SMARCA4. But what we see is that the SASP, these chemoattractant chemokines that are needed for attracting these NK cells, are upregulated in senescence and superinduced once that we inhibited SMARCA4. So we think that SMARCA4 inhibition superinduces the SASP in order to enhance this NK-mediated killing of senescent cells. Importantly, the other thing that we saw is that and this might explain why these effects are selective of senescent cells, is that SMARCA4 is induced at the protein level in senescent cells. So this, for example, is like the slides derived from the liver of mice in which we have expressed RAS, and this RAS induced senescence in hepatocytes, and these senescence hepatocytes have also increased levels of SMARCA4. Similarly, and this will be relevant, so you cannot see very well the immunofluorescence, but you can see the quantification. Similarly, this will be relevant later on in the talk. So this is a model of ovarian cancer xenografts that we treat with cisplatin, and this induces senescence. And when we do so, SMARCA4 levels also increase. So we believe that this upregulation of SMARCA4 might explain the selectivity of this superinduction of the SASP in senescent cells. And mechanistically, what we see is that when we inhibit SMARCA4, we increase cGAS-STING signaling. So we can just, again, have here like the model of oncogene-induced senescence. So we treat cells with a PROTAC, and we see that there are like increased levels of total cGAS, but also phosphorylated STING, that is a marker of activation. And downstream of cGAS-STING activation, there is activation of TBK1 and IRF3, that is signaled by the increase of expression of these phosphoforms. So moreover, if we use cGAS-STING inhibitors, so we can prevent this SMARCA4 superinduction of the SASP. And this is shown here with two STING inhibitors and a cGAS inhibitor. And the question is how cGAS-STING is activated. So we mentioned, we talked before about CCFs, about mitochondrial DNA, but also retrotransposons, mobilization of retrotransposons is a form of cytoplasmic DNA that can activate the cGAS-STING, and this has been described before. So we look into the fact that SMARCA4 inhibition has on the expression of retrotransposon and other transposable elemental subfamilies. So we see that there are like some retrotransposons that are activated when we treat with SMARCA4. Importantly, when we look more generally into transposable elements, we see that satellites that are not to be distant on senescence seems to be expressed preferentially when we treat with SMARCA4 in senescent cells. And overall, what we see is that there is an increase of this accumulation of cytoplasmic DNA, double-stranded DNA, that is what activates cGAS-STING. And for example, this is also one of the proteins that is encoded by line 1, so this transposon that is also mobilized and that is expressed. So we think that, at least to some extent, inhibition of SMARCA4 results in mobilization of transposons that might activate cGAS-STING. So this can be tested. We can use retrotransposons inhibitors to try to see the effect that it has on the expression of this SASP in response to SMARCA4. And in fact, retrotransposons inhibitors prevent the SASP superinduction caused by SMARCA4 inhibition. So the model that we have is that that SMARCA4 is somehow repressing the expression of transposons and repetitive elements. And in general, the presence of cytoplasmic nucleic acids. So this, when we inhibit it, basically activate the SIGA-16 pathway. And I have not shown data, but there is also like increase of RNA in the cytoplasm that activate the RIG1 MAPS pathway. So overall, all of that result in increase of the SASP, and that contribute to the natural killer cell recruitment. So once that we know that we can use SMARCA4 to try to superinduce the SASP and try to increase this immune-mediated elimination of senescent cells, we wonder if this can work in the context of anti-cancer therapies. And we have introduced before already to this combination of chemotherapies and senolytics, so what is called the one-two-punch approach. And what we want to use is this in the context of ovarian cancer. And why in the context of ovarian cancer? So ovarian cancer is a tumor that of an unmet need in which there is the current treatment standard of care is cisplatin. Cisplatin is known to induce senescence, and it is also known that ovarian cancer survival correlates with immune infiltration. So what we thought is that if we could, rather than use senolytics in this case, use our SMARCA4 inhibitor to basically enhance this immune-mediated surveillance of senescent cells, this might cooperate or synergize with cisplatin in order to clear these ovarian cancers. So we went into testing that, and this is a work from Maria in the lab, and what we use is as a xenograph model of ovarian cancer that is immunocompetent, in which we have like 4 different groups: cisplatin standard of care, the SMARCA4 PROTAC, or a combination. And what we can see is that when we treat with this SMARCA4 inhibitor that is well tolerated, non-toxic, so we see an increase, and this synergizes with cisplatin of expression of SAS mediators, including chemoattractants that are known to be necessary to attract NK cells. So in fact, there is an increase of infiltrated NK cells and T cells in the tumor, and those include mature active NK cells. When we look into what's happening with the tumor size, so we see that tumor size decrease, and we can just see this both by looking into the average, but also we just filter for the tumor size. The bigger tumor size is underrepresented in the group that we have treated with cisplatin and these SMARCA4 inhibitors. And just to finalize, so what the last thing that I want to show is that, in part at least, so this effect is due to natural killer cells. When we just deplete natural killer cells using anti-NK antibodies, so we can rescue this tumor size. So signaling that at least part of this anti-tumoral response is NK-mediated. There might be still some T-cell responses that we have not evaluated, but we see increased T-cell infiltration. Just to conclude, different xenotherapies include senolytics, can be used to treat cancer and age-related disease. So SMARCA4 inhibition superinduces the SASP, and we can use that to potentiate the NK-mediated elimination of senescent cells. And I showed you some data on how we can just combine cisplatin, the standard of care, with SMARCA4 targeting PROTACs in order to eliminate ovarian cancer tumors. So, and just to conclude, so these are the people in my lab. So I want to highlight specifically both Virinder and Maria Antonietta that had conducted experiments in the NK project. Aya Magnis is one of our key collaborators, our funders, and of course, everybody else. So thank you so much.
Thank you very much, Jesus, for this very exciting talk. Questions there? Do we have?
Fantastic talk. Just curious, in your ovarian cancer experiment, if you've tried that in an aged environment. I assume you used younger mice, but in an aged environment with chronic sterile inflammation, could there be consequences to superinducing the SASP?
Yeah, so that's a very interesting question. We haven't done the experiment. So we have been, in fact, awarded very recently a grant to try to work more in ovarian cancer, senescence, and immune responses. And this is one of the things that we want to do in that context.
Over there.
Great talk, Jesus. So if you do a co-culture experiment of senescent cells and non-senescent cells, and then treat with the SMARCA4 inhibitor, is the killing still specific to the senescent cells over the non-senescent cells?
We haven't done that experiment, so I think that that's a good idea. So that's something that we can try relatively easily. Yeah.
Okay, thanks.
Jesus, have you just proven the xenomorphic approach wrong with your experiments?
No. So I don't think so. I think that what I want. So we are today talking about Senotherapeutics, and I think that includes many modalities, right? So I think that, of course, for example, in the context of aging and also in the context of cancer, inflammation is driven a lot of the damage. So xenomorphics, and we have done that in the past, molecules that dampen this inflammation are gonna cause effects. What we are thinking is that another way of trying to do it, and I didn't talk of other ways to try to potentiate immune-mediated killing that include CAR T cells, vaccines, immune checkpoint ligands, but another way to do it is a transient recruitment of these immune cells. And I want to highlight the transient because we don't want to have like this super induction of the SASP all the time, because that probably is gonna potentially cause an inflammatory microenvironment that might be like tumor suppressive. But we think that if we do a transient induction, that might mimic senolytics in the sense that it's transient and intermittent and might recruit the immune system and eliminate the senescent cells. But of course, don't disprove xenomorphics at all.
Yes, Andrea?
Yeah, so yes, so we have worked a lot on NK cell. We published a paper recently, right? So, and by discussing with the relevant NK biologists, particularly from Marseille, where they are making the revolution in NK, so they told me that the recruitment doesn't explain if really you have an NK effect, because indeed they can be recruited, but after they can die. So I don't know if you have looked at the proliferation. So maybe your SASP can render them much more proliferative. And after, I mean, that's an advice, is you should look also at the inhibitory ligand, because perhaps the inhibitory ligand are down, and so you have much more activation. And what they told me is actually that you always have to confirm by protein level this ligand, because often there is no correspondence between. So I suspect that since you have a very good effect, you also have more proliferation and maybe more activation through downregulation of NK ligand. But I don't know, that's a suggestion.
Yeah, no, good point. So for example, we see in the tumor that there is increase on the granzyme positive NK cells, so there is increased killing. So we have looked at least in vitro into the inhibitory ligands. I don't think that we see like an effect on inhibitory ligands, but one thing that we also look is whether, for example, some of these SMARCA4 inhibitors can have intrinsic effects, not in the senescent cells, but in the immune cells. And I think that potentially there might be also some. So I agree that there might be more things than just the recruitment itself, yeah.
Over there?
Hey, just a very nice talk. So related to this, actually, two challenges/questions. So the first is whether you can make the senescent cells that might not be sensitive to NK cell now resensitized just by inhibiting SMARCA4. And if you try that, the ones that are resistant normally. And the second is, of course, NK cells activity also change over time. So in an aged environment, it might not just be sufficient to recruit them because they might just not be active. So I wonder whether you also looked at whether, a young environment is much better, at clearing the senescent cells that after the treatment with SMARCA4 inhibitors.
Yeah, so the first question I think is a very good question. So we haven't looked specifically into that, but I think that, of course, that will be like something that therapeutically will be very relevant, whether we can target now cells that we couldn't target before. We haven't looked into that. There might be other ways that we can try to achieve that, but I think that is a very good question. The second relates to what was asked before, right? The young versus old environment and potentially whether the NK cells there. So I think that's absolutely is a question to consider. We haven't tested in an old environment and tried to see whether we can also reactivate some of these NK cells, but I think that these are some of the things that we are trying to do. potentially there might be other ways in which we have to tackle those questions, but I think that combination between, for example, this is marker 4 and some other things that we can do might be valuable. Yeah.
Jesus, one question. So you have this nicely way of transiently amplifying the inflammation, but what about the potential feedback loop to the bone marrow that then you produce more immune cells, and at a certain point you can even generate a more fibrotic tissue or collagen deposition and so on? Have you thought about that?
So we haven't looked into that in detail. Of course, I think that there are all the kind of things that we have to look. I mean, in the context of the experiment that we did in vivo, in the cancer, they were like relatively short term. And we look there in the infiltration and in the tumor, but we also look into the spleen and we look into peripheral blood in some cases. And I think that we didn't observe anything else. But again, those experiments were relatively short term. So I think that there are all sorts of things that we need to consider.
Awesome. Thank you. Okay, if there are no further questions, we would like to thank again Jesus and all the speakers of the morning.
We'll start with Fabrizio. Fabrizio Dadda di Fagagna is coming from the IFOM in Milan. He will tell us about telomeres and their role in aging.
Thank you, Luca. Will not thank the organizers, obviously, being one of them, but I will thank you for being here. It's really, again, a pleasure to see so many people attending today. Let's see if... Yes, so we'll be talking about telomere biology and its relevance in aging and different diseases, including cancer. Aging is complex, but probably a sufficiently broad definition of the process of aging is that it's the result of the loss of homeostasis. And it's probably sufficient to look at the skin of a young person, how different it becomes with time. And this will immediately gives you a message of a lack of equilibrium, a loss of homeostasis. Importantly, aging is not just about wrinkles. Aging is the largest risk factor for several human diseases, including cancer, cardiovascular disorders, and neurodegeneration, as Peter alluded before in his introductory lectures. Aging is complex, yes, but I think, especially given this audience and the number of speakers in this room today, we think that we actually know what makes us age. And we age because our cells age. It's a reductionist approach, but I think it's an approach that works. And senescent cells are aged cells, and we know they have lost the ability to proliferate, so they cannot contribute to the physiological turnover of dying cells. And in addition, as you have heard in the previous presentation, they have this nasty habit of secreting toxic molecules. So a single cell problem eventually becomes an organ problem, a system problem, and a full organism problem. So the question then becomes, okay, if we age because our cells age, what makes a cell age? Because in principle, cells have all the means to keep themselves in perfect conditions. Everything that breaks down in a cell can be replaced. There is one single component in our cells, however, that if damaged, cannot be replaced. And of course, this is our genome. And yes, there is very good correlation between the accumulation of DNA damage with time and aging and the processes that are associated with degeneration. So yes, more damage, more aging, and more diseases of aging. However, DNA damage is repairable. Senescence and aging are irreversible events. And so how can a reversible event like DNA damage, how can it lead to an irreversible event such as aging? So in the past, we tried to address this question by performing a very simple experiment. We take normal cells, we expose them to random DNA damage by ionizing radiations, and this indeed generates DNA damage throughout the genome. This induces a prompt response by the cells. We call it the DNA damage response, which promotes DNA repair. However, we discovered that those few lesions that happen to occur within the ends of the chromosomes, the telomeres, those lesions cannot be repaired, or they can be repaired much less efficiently. And so with time, telomeres accumulate persistent lesions that chronically activate the DNA damage response. And probably telomeres are not, it's not a surprise to involve telomere biology in the context of aging, because you may be familiar with the concept that cells, when they proliferate, telomeres get shorter and shorter and shorter, and eventually they become too short and they trigger a DNA damage response. So when we were seeking to and to address the question, what is that irreversible event that causes cells to become senescent, we are prone to propose that it's the loss of telomere integrity, call it a long but damaged telomere, which is probably more relevant in non-proliferating cells, or a critically short telomere, most likely impactful in proliferating tissues, as that irreversible event that triggers that irreversible condition known as cellular senescence. Therefore, telomeres are effectively our Achilles heel because they are the irreparable portion of that irreplaceable component, which is our DNA. Importantly, however, it would be wrong to think that DNA damage causes cellular senescence, although it's tempting. It's the activation of the DNA damage response pathways that mediates all the consequences of DNA damage generation. And so if you can intervene and you can modulate the DNA damage response signaling, you may have an impact on all the consequences downstream of DNA damage generation. And this is what we attempted to do in recent years. And here I'm really summarizing a large body of results from our lab. So what we discover is that when telomeres become either too short or damaged, or somehow anyway, they engage the DNA damage response machinery, and so they are directly recognized by these DNA damage response factors. Well, this set of events is strictly dependent on their transcription. In other words, telomeres get damaged, they get transcribed, these RNA stay associated with the chromatin, and this helps recruiting these factors that signal the presence of DNA damage, and this mediates cellular senescence. And when we discover that, we realize this provides us with an opportunity to target not the proteins, but the RNA, which are necessary to activate these proteins. So we design antisense oligonucleotides targeting this telomeric RNA, and we discovered that this antisense oligonucleotide dismantle, inactivate, dissolve this DNA damage response complexes, and effectively we have developed the first selective inhibitor, selective, specific, only for telomeric DNA damage response activation. So this is not an inhibitor of DDR throughout the genome. It will selectively inhibit events only occurring at the telomeres. And therefore, with this tool, we can uncouple DNA damage from its consequences, the DNA damage response. So we can check whether a certain phenotype or a certain disease is the telomeric DNA damage response activation contributing to the effects that I'm studying. Now we have an inhibitor, we can apply the inhibitor, and we can see whether anything gets modified. And with this tool, we validated in a number of settings, in vitro, but also in vivo. Usually they were acute settings, including, for perhaps more relevant here, is a mouse model of an accelerated aging syndrome, Hutchinson-Gilford-Progeria syndrome, which is a very rare accelerated aging syndrome. Luckily, it is rare, but we have been scavenging the literature, and in this review, what we tried to do is to systematize or structure a bit what was evident. And what we realized, there were a number of human conditions associated with telomere dysfunction, and some of them actually you could really say they are caused by telomere dysfunction. And today I will be discussing some of them and how we can impact on them. And I will start with IPF. It's called idiopathic pulmonary fibrosis, but honestly, there is not much idiopathic in IPF because it is there is a very well causal link between telomere shortening and this disease. I will probably not bore you by reading or summarizing all the evidence, but again, the message is very simple. IPF patients tend to have short telomeres, they have DNA damage response marker, they have plenty of senescence markers. There are patients that have germline mutations in telomere biology genes, such as telomerase. Telomerase is the enzymes that maintain long telomeres. Without telomerase, your telomeres get shorter and shorter. And so you have the familiar forms of IPF with the telomerase mutations and also some sporadic forms. So Clearly, this is a telomere biology condition, and so we thought that we should model it by adopting a telomerase knockout mouse, and so this is the one that we used. So the RNA component of telomerase was inactivated in this mouse model that many others have characterized. Mice, for some funny reason, start with very long telomeres, so you have to breed them for a number of generations until the phenotypes arise. So we used the third generation of these knockout mice, and we treated them. So what we did was to inject them, as you can see here, when they were relatively young with our antisense oligonucleotide. We used two different ones. Telomeres are made of TTA-GGG sequences, so we use an anti-G and anti-C RNA ASO against this RNA, or a control antisense oligonucleotides. And we treat them systemically, so these are intraperitoneal injections, and then we monitor efficacy at different time points. Today, for lack of time, and just show you the results at 12 months of age. And what we observed, first thing first, do these mice accumulate DNA damage to their telomeres? Do they activate the DNA damage response? And is our approach effective in reducing this DNA damage response signaling? And the answer is yes. You can see by high here. These are the stainings of the lungs of these mice, and this is the results of the same stainings in mice treated with this antisense oligonucleotide, and the quantification is here. Each circle is one mouse. So we see reduction of DDR, we see an impact on the histology of the lungs. You can see by eye, again, you probably don't need to be a pathologist to see the difference between a normal lung and a very sick, compromised telomerase knockout lung, and how this treatment is improving them. So our pathologist, Claudio Trapodo, has compiled a number of observations, quantitative, that he has compiled in what we call the pathological score, and also here you can clearly see there is an increase in the pathological phenotypes that are reduced in this, upon these treatments, but most importantly, IPF patients die of fibrosis. So can we actually make an impact also on fibrosis in these lungs? And indeed, if you stain lung sections by Masson 3' staining, which is a way to highlight fibrosis, you can see there is a very fibrotic lung, which becomes much less so when mice were treated. So we have been, of course, characterizing these mice in many different ways, and RNA-seq, of course, is a powerful tool to dig into the different pathways. One point that we really wanted to make, to address, was whether these mice were recapitulating physiological aging, perhaps in an accelerated manner. And this set of results really makes the point. As you can see, these are the results of an RNA-seq experiments. The genes that go up and down in normal aging also go up and down in the same direction in our model. So in an accelerated way, we are recapitulating physiological events, but most importantly, these changes are brought back, are reduced, are rescued upon these treatments. So we think that yes, these results are relevant for this telomerase IPF model, but maybe relevant also in physiological aging. You have heard before about a lot of signatures of aging. We adopted also some of them, the Mayo signature, and indeed we observe, perhaps not surprisingly, that senescent signature is elevated in these knockouts and reduced upon these treatments. However, that was mice. Are we really recapitulating what is happening in humans, in IPF patients? So this is the results that supports that. So what we did was to download a data set of RNA-seq from two independent reports of the data of gene expression changes in IPF patients compared to the normal population. As you can see, that those changes are to a very good extent recapitulated in our knockout mice, and to a good extent, not completely, but to a good extent, they are rescued upon treatment. So we think that really bodes well for their potential use also in human settings. Finally, of course, there are many more data that we have squeezed out of these mice, that here they are synthesizing this PCA analysis that clearly shows very, very simply that you have the wild-type mice here, the knockout mice here, and the knockouts that have been treated, and so they get much closer to the wild-type situation. However, you may say, Fabrizio, you like to win easy, meaning you are treating mice when they are young, when fibrosis is not well developed. Can you actually make a difference when fibrosis is already well established? So this is what we did. So these are much older mice. So they are 10 months old mice. So we treat them in the same way, and then we monitor efficacies at different time points, again, including also when they are fairly old, 20 months of age. And however, also here, fibrosis is reduced when fibrosis was well established. So again, this suggests that this treatment, we may make a difference not only in preventing the condition, but also to an extent either stopping the condition or even reversing it. So to summarize this part, basically we believe that in this setup, the telomeric shortening or telomere dysfunction activates a DNA damage response that enforces cellular senescence, and by driving inflammation, this causes lung fibrosis, and by blunting the initial step, so the DNA damage response signaling, we can make a long-lasting impact on the lungs. So that was about the lung condition. However, there are there is a lot of evidence there are human conditions that, upon telomere shortening, telomere dysfunction, impact on the hematopoietic system. And by the way, I should have mentioned this before, we are not impacting on the telomere length. So we are reducing DDR signaling without lengthening the telomeres. This is a very crucial point that I almost forgot to mention. But as I was saying, yes, lung is one, but there are also a number of conditions that we have summarized them here as you may call them maybe more broadly bone marrow failure syndrome. So we look at the bone marrow and the spleen. Here there are too many data to go through. The message is fairly simple. In all the cell types that we check, there is a dysfunction that to a very good extent is rescued by these treatments. But when I am not an immunologist, and when I saw this result, I thought that since we see such a broad effect, maybe we were able to impact the hematopoietic stem cell function. And that's why this impact then reverberates in all the different cell subtypes. So we perform an assay, a fairly simple assay that measures the fitness of hematopoietic stem cells. It's a colony formation assay. And this revealed that, perhaps not surprisingly, the knockout mice are less able, less fit to make colonies in this in vitro assay. However, when you take the bone marrow and you plate the same number of cells of wild type, knockout, either untreated or treated with a control agent or treated with these two independent antisense oligonucleotide, you rescue their ability to form colonies nearly to wild type levels. And this is a persistent response because it can last up to 9 months after the treatment. This was, again, for us enough, but then we were told, no, that's not enough. You have to prove this in vivo by a transplantation experiment. So this is what we did. We performed a competitive transplantation experiment in which you irradiate some mice, so now they don't have a functional bone marrow, and there you transplant either a wild type and a competitor challenging bone marrow. So this could be either another wild type or our knockouts treated with our antisense oligonucleotide. And here we observe that, of course, the wild type is able to repopulate this empty niche, the knockouts is not. And one of the two, surprisingly, only one of the two is much more efficient to rescue to repopulate this bone marrow. And now we are analyzing one is much better, well, is significantly better than the other. And what is emerging is that in all the previous analyses that, again, most of them I skipped, one of the two A's was actually better than the other. So it's very likely when you compound these small differences, when you sum them up, clearly when you challenge and you push these cells to repopulate an entire empty niche, that clearly one is better than the other. And just to complement this approach, we also use a completely different mouse model. In this case, mice do express telomerase they do express telomerase, but by some genetic tricks, again, spare the details, basically the bone marrow is somehow forced to do a number of cell divisions because many of the cells are dying, so the mice develop anemia. And when the mice are already anemic, we treat them with the antisense oligonucleotide, and then we monitor different parameters. Here I'm showing you survival, and as you can see, the Treated mice survive much better than the untreated mice, and testosterone in this case is our active positive comparator. So we think that there is an opportunity for interventions also in the hematopoietic system. However, again, these were telomerase knockout mice, a special system. Can you actually make a difference in a wild type situation? So this is still very much ongoing, and again, this is unpublished, so we did this honestly silly experiment of treating mice very, very, very old. So these are 26-month-old mice. We treated them with our antisense oligonucleotide, and 2 months later, 3 months later, we sacrificed them and we monitored their ability to perform colony formation analysis, because we think this is giving us a quantitative readout and a functional readout. And as you can see, we are able to improve their ability to make a colony. So this means that also in a wild type situation, this treatment may be effective. But again, these are mice. How about humans? And here, Raffaella Di Micco came to the rescue. So with her, we performed some experiment. We exposed the CD34 positive, so purified hematopoietic stem cells from humans, which were healthy, just a bit elderly. We treated them with these 2 independent antisense oligonucleotides. She's more sophisticated than us, so she distinguished red colonies from white colonies. And as you can see, there is a hint of efficacy, especially for the red colonies, and to an extent also for the white myeloid colonies, white colonies. So it seems that we can be efficacious also in wild-type settings, both in mice and perhaps in humans, at least ex vivo. Very last point, I mentioned so far tissues that to an extent are proliferating, maybe not much, but certainly the immune system and the hematopoietic system is. But there is also some evidence that non-proliferating cells, like brain cells, like neurons, may accumulate DNA damage, and this may be occurring in more so in some disease contexts, such as neurodegeneration. And indeed, there are reports highlighting links between Alzheimer's disease and DNA damage generation. So we wanted to have a look into this ourselves. And so we adopted a mouse model of Alzheimer's disease that expresses 3 mutations which recapitulate human conditions. We check and we stain their brains, both in the cortex and the hippocampus, for DNA damage response markers. And as you can see here, we do see an increased levels of DNA damage response markers in these neurons. This is associated with markers of cellular senescence such as P21 and an induction of pro-apoptotic genes. And perhaps not surprisingly, not for us, but maybe we're biased, we observed that this DNA damage was preferentially accumulating at the telomeres, as you can see here, quantified in different manners, but not at other equally repetitive, equally large and structurally relevant portions of our chromosomes, such as the centromeres. What we then did was to try to establish a model to study these events in a more convenient in vitro system. So what we decided to do was to expose primary mouse neurons to oligomers of the A-beta-42, which is a peptide, which is a component of the amyloid plaques, and to an extent, this may recapitulate in the pathogenesis of the disease, although there is, of course, a lot of ongoing discussions whether the plaques may be causative or consequential in the disease. However, what we observe is that if you take healthy neurons, you just expose them to A-beta-42, you observe the accumulation of DNA damage. Again, this DNA damage occurs at the telomeres. And therefore, immediately what we did was to treat these neurons exposed to A-beta-42 that had DNA damage of the telomeres to our telomeric antisense oligonucleotides, and we observed that we can reduce the DNA damage response activation of the telomeres in this in vitro system, and most importantly, we can reduce also some of the consequences of DDR activation, such as, again, P21, the pre-apoptotic gene set, and to extend the cell viability. So we think that we are, once again, by attacking a fairly upstream event, we are now modulating many of the consequences of this DNA damage response activation. And of course, we worked out the mechanism that, again, unfortunately today I don't have the time to discuss here, but of course they involve altered calcium influx and increased levels of ROS, which eventually impact on telomeres that we know are very much sensitive to DNA damage, oxidative DNA damage accumulation, and as I mentioned before, when damaged, they are also less effective in being repaired. Now, I haven't been speaking about cancer. Aging and cancer are linked in many different ways, and today, recently we have tried to assemble a coherent picture that I co-authored with Lucrezia, which is sitting on there, and she's the co-author of this review, and therefore since being a DNA damage lab, we are certainly interested in the aging aspects. You have heard about the neurodegenerative diseases, but also we are interested in cancer. And so can, with our understanding of the DNA damage response, including the telomeres, make an impact on cancer biology too? And yes, perhaps, because there is a fraction of tumors that to gain immortality, rather than re-expressing telomerase and therefore elongate their telomeres, essentially what they do, they keep reshuffling their damaged telomeres. And this is an alternative approach to telomerase re-expression. So that's why these telomeres are called ALT, for alternative lengthening of the telomeres. Typically, they are sarcomas or brain tumors. And consider that in this case, the DDR is helping the survival of these tumors, because DDR here is necessary to favor the recombination, therefore the lengthening of these chronically damaged telomeres. So we thought, how about we impede such a set of events by treating these ALT tumors with our antisense oligonucleotide approach. And here I will be going very, very quickly, just a single slide. What we observed that when we treat these tumor types, these ALT cell lines with an antisense oligonucleotide, we are, again, not surprisingly, reducing the recruitment of known DNA damage response factors. This is what we have observed also in all other settings. What happens here in this cancer setting is the cells try to fight back, and they recruit additional factors in order to favor a different kind of DNA repair activities. This is not efficient, this is not effective, eventually leads to a further accumulation of DNA damage response activation markers, and eventually this leads to the death of the cells. So in this way, by targeting the DNA damage response activation, we are not favoring survival or rejuvenation as in the aging setting, but rather here we are killing selectively cells that need the DNA damage response. So I'm stopping here. I think that, again, we try to make the point of the relevance of the telomere biology, and of course the fact that we have developed this research tool that we believe they've demonstrated some efficacy in different settings of aging-related diseases and cancer too. Most important slide, this is not my group, these are different generations of my group that gathered in Ortigia a few years back to celebrate 20 years in the lab. On the right, you see all the people that are presently now contributing to this, to the efforts in the lab. And last but not least, we have open positions for a junior PI interested in working at the interface between cancer and aging. So if you are interested, that is the QR code, or you just talk to me. Happy to take your questions.
Thank you very much, Fabio, also for being on time. So we save a little bit. Questions over here, one and one over there.
Beautiful talk, really great. So I was wondering, clonal hematopoiesis, which is a condition that is very frequent, I would say, in the elderly, right? This is associated with the increased risk of cancer and cardiovascular disease, etc. So do you have evidence that, for instance, TET2 mutation, since they are associated to promotion of DNA damage in the hematopoietic stem cell, is this DNA damage at the telomere? Can you prevent with your Tasso.
Yeah, you're reading my mind because we did that experiment in collaboration with people way more expert than us in clonal hematopoiesis. Turns out that in this model, clonal hematopoiesis, it's a fairly rare event, too rare to be studied in our setting, meaning you would really need dozens of mice to see a phenotype and the rescue of the phenotype. So unfortunately, I don't have a question, but definitely we were interested in addressing this because, of course, it's a relevant one.
Okay.
Marco.
Hi, Fabrizio. Very nice talk. So, especially because of your Turk knockout model, I was wondering whether the response and the sensitivity to the antisense oligos is dependent on the amount of also damage they have at telomeres. Because I imagine that these Turk knockout mice have shorter telomeres, so that just by chance, they would have less damage at telomeres than like more healthy, premature senescent cells that are maybe young and have long telomeres. So did you make any, comparisons to see whether more damage at telomeres or at different sites in the telomeres make a difference on the response to the
what I can say is that we used also different mouse models in which, for instance, we removed TRF2, and that means that suddenly all the telomeres now are signaling, they are all uncapped, and there the DNA damage response is brutally activated, way more than in this setting, and also there we can make an impact with our antisense oligonucleotides. However, in that setting, it is again so brutal that mice die within 10 days. Actually, we are characterizing these events, so we cannot follow them in time. But what I can say that in that window of time in which mice are still alive, we can still make a difference with this antisense oligonucleotide approach. Yes.
Yeah. Fabrizio.
Raffaella.
Thank you so much. Really great talk. In your in vivo experiments, when you use the oligos in the mice, so can it be that what you are seeing is a consequence of a rejuvenation of the immune system, that now it's much more active in eradicating senescent cells? Did you look at
I keep telling my postdocs.
Cells,
absolutely.
example.
So a contribution of the immune system in mediating events outside of the immune system, it is definitely a possibility. It is hard to address because either you have a transgenic mouse model in which, so it's complicated. We are thinking about transplantation experiments, which are also complicated. So you're reading my mind again. So definitely this is what we are thinking, but addressing it experimentally may be challenging. But definitely this is something we are considering.
Fabrizio, over here.
Clemens.
Very simple question. How do centenarians, as compared to 70, 60 years old age people, cope with their telomeric damage? The telomeric damage should simply be a function of cell turnover divisions over life. And if you live 30 years longer, you should have much more telomeric damage. Is the answer also the immune system? What is the inter-individual explanation that not everyone has the same telomeric damage associated aging phenotype?
Well, actually, there is difference. I think there are reports that healthy old people, I'm not sure centenarians, may have less DNA damage response markers.
But why?
Why? Because, of course, they may have been exposed to different challenges. And they may
divide their cells at the same pace. They have way more cells turned over.
Yes, but consider also that, of course, we are not all born with the same telomere length, and so this may have also make an impact.
Is that known?
Yeah, no, there is certainly, there are
the difference that I start with a 30 years difference to my classmates, so to say.
But your point is well taken. I think the fundamental question that the field hasn't addressed yet is how short is a critically short telomere to signal. Embarrassingly, we still do not know. When we will know that, we will be able to map those. Now, nanopore sequencing of the telomere is available. I think that is the basis of my next grants, if somebody gives me money.
Fabrizio, here. So very nice talk. So I was wondering, you transplant experiments when you rescued. So it is known that upon aging, you have a myeloid bias production, right? So do you see now a balanced production, or did you check that?
I think we did check because we did sort cells. I should talk to my post, but I think that is a very relevant question. And of course, the hope is that we rescue also the myeloid skewing. I will double check and let you know, but it's a good point. Yeah.
Thank you.
Thank you. So we can stop here so we recover some minutes. Thank you again, Fabrizio. Now we move to a couple of short talks selected from the abstract. They're only 10 minutes, but not less interesting. And we start with Federica Marasca from the INGM in Milan. She will tell us about RNA targeting. Very curious to read about it, hear about it. Please. Also, there will be no time for questions at the end. You're more than welcome to grab the speakers when they finish and bother them.
So,
Please.
good morning, everyone, and thank you to the organizer for selecting my abstract for this beautiful meeting. Well, I'm a research scientist in the lab of Professor Beatrice Bodega, and actually one of the major interests of our lab is to unveil the function of the dark side of our genome. So I'm moving to a little bit different topic. When I speak about dark side of our genome, I'm referring to repetitive elements, and in particular, our focus are retrotransposable elements that are mobile genetic interspersed elements that alone cover the 50% of our genome. Among them, line 1 are obviously the most famous retrotransposons, and actually is the, let's say, more represented subfamily of retrotransposons. And it is present in our genome with 500,000 of copies, and despite what we learn from the textbook, actually, really, the vast minority of these elements, the 0.1%, has the ability to retrotranspose. The vast majority of these elements is composed by these actually short elements that are everywhere in our genome, that we have simply inherited them through evolution, short fossils unable to retrotranspose. Actually, we have inherited these elements because they have been co-opted for novel regulatory function. It is now accepted that these elements, both the DNA and the RNA that is transcribed from these elements, could play regulatory function. And actually, within this specific frame, we were the first to assess a function for a line 1 transcript in the regulation of the immune system, and in particular in the regulation of T-cell quiescence and T-cell dysfunction. Well, to cut a very long story short, actually, we demonstrated that the nuclei of quiescent naive T cells is fully enriched of the signal of these line 1 elements. And actually, we demonstrated that they are promptly downregulated during the process of cellular activation. But given the fact that standard tools like bioinformatic pipelines, experimental approach, or reference annotation simply discard these elements, in order to understand precisely which elements were expressed in our cells and which is the structure of the transcript that were expressed, actually, we leverage on an RNA sequencing of the chromatin-associated RNA, with long and short read sequencing coupled with de novo transcriptome reconstruction. And actually, this approach allows us to find that these short intronic line 1 elements that are evolutionary old line 1 elements are expressed in our system, as you can see here, and basically they are spliced into novel transcript variants. We are speaking about novel splicing variants. We demonstrated that these splicing variants remain associated to the chromatin of quiescent T cells, sustaining the quiescence of the cells by simply repressing the expression of genes required for cellular activation. And we demonstrated that the activation of the T cells induced the downregulation of this line 1 transcript by a pathway that is dependent from mTORC1 activity and the splicing suppressor that is BTBP1. But more interestingly, we found that in dysfunctional T cells, exhausted T cells, tumor-infiltrating T cells, actually this mechanism is hampered. And this leads to the aberrant reaccumulation of line 1 transcript in the nuclei of tumor-infiltrating lymphocytes. And this is the most, let's say, relevant data of the story. Actually, we found that by targeting line 1 transcript in tumor-infiltrating lymphocytes by means of antisense oligonucleotides, we were able to restore proper immune anti-tumor function, as you can see by the increase of effector cytokine secretion alongside with increase of anti-tumor killing abilities. Well, given the relevance of these findings, we decided to investigate whether this line-1 transcript could regulate human T cell function also across lifespan, because actually the immune system changes a lot in composition and function during lifespan. For example, at birth, in neonates, actually neonatal T cells are immature and hyperreactive, while in elderly individuals, these cells are dysfunctional with sign of immune senescence. So what we found in this study was that actually neonatal naive T cells, despite being quiescent, do not express at all line-1 transcript. We demonstrated that this is something dependent to a tonic TCR and mTORC1 activation that happen in these cells. But more intriguingly, we measure line-1 expression in circulating T cells isolated from different individuals at different life stages. And we found that line-1 expression actually progressively increases during the process of T cell maturations, picking up in elderly T cells, both naive and memory T cells. But in this study, we were really much interested in understanding specifically the biological processes that were regulated by the absence of line-1 transcript, and in particular, those biological processes that get reactivated by the absence of line-1 transcript or lower level of line-1 transcript in T cells. Therefore, we leverage on this transcriptomic approach in which we perform multiple comparisons. I will not enter in the detail of that, but basically we found a core set of genes that are actually always upregulated whenever line-1 is absent or present at lower level. And we are speaking about genes related to cell cycle and more predominantly to protein synthesis, that is a really important process for proper immunological effector function. Of course, we validated these results, and we found that actually neonatal naive T cells display a higher protein synthesis rate in respect to adult T cells. As you can see, these anti-correlate with line-1 expression, and neonatal T cells get faster in the cell cycle progression. And similarly, we obtain the same results by knocking down line-1 transcript in adult T cells. What about elderly T cells? Well, at first, we found that actually these elderly T cells, both naive and memory, actually display lower protein synthesis rate in respect to adult T cells. And once more, by knocking down our line-1 transcript, we were able to restore protein synthesis level. So, given, let's say, these results that clearly suggest that line-1 transcript regulate immune function by modulating protein synthesis in lifespan, we asked whether this could be the same also in the tumor microenvironment in exhausted T cells. Well, at first, we analyzed the public available data sets of tumor-infiltrating lymphocytes, as you can see, multiple tumor types, several patients, thousands of cells. And actually, we analyzed specifically the expression of line-1 through these tools, iRescue, that we recently developed in the lab to measure specifically the expression of repetitive elements in 10x data. And actually, we end up finding that line-1 expression actually correlate quite nicely with T cell exhaustion, but intriguingly, it anti-correlates with protein synthesis. And once more, if we knock down the expression of line-1 transcript in exhausted T cells, as you can see here, actually we were able to rescue protein synthesis levels in, let's say, to levels that are similar to those displayed by effector T cells. So, in conclusion, we found this novel mechanism mediated by line-1 transcript that regulate immune T cell function in aging and cancer. This mechanism is actually passed through the regulation of protein synthesis, and actually we really believe that the targeting of line-1 transcript could be a novel immunotherapeutic approach both for cancer and aging. But related to this specific point, I really would like to underline the importance to understand the precise structure of the line-1 transcript that we want to target in a precise cellular condition. Because this, of course, could improve a lot the specificity of our approach. And in order to do that, actually in the lab, we are exploiting this strategy of single-cell RNA sequencing of tumor-infiltrating lymphocytes in which we pass through both long-reads RNA sequencing and full-length transcriptome using SMART-seq technologies, of course followed by de novo transcriptome assembly. And actually we end up with a catalog of 4,000 of line 1 transcripts expressed in tumor-infiltrating lymphocytes, while differential expression analysis among the clusters allow us to reveal a specific signature of line 1 transcripts that are specifically expressed in exhausted and pre-exhausted T cells. Actually, this is something that is running in the lab, also in partnership with our company, T1 Therapeutics. So really to conclude, I really do hope that I convince you that by looking to this dark matter of our genome, we can discover novel mechanisms and therapeutic opportunities. So the last slide, I would like to convey my biggest thanks to Beatrice, that of course followed me during this scientific journey, to my colleague Valeria and Rebecca, and of course to our collaborators and all of you for your attention.
Thank you very much, Federica, also for being on time. Sorry there is no time for questions, but now we have Marco Bollis with another selected talk. He's from the Institute of Oncology Research here in Bellinzona.
Okay. So first of all, thank you very much for selecting our abstract for a short presentation. And in the last few presentations, we heard about some of the most crucial hallmarks of aging, which are telomere shortening and DNA damage. But of course, there is another hallmark of aging, which is not, that has not yet been cited, which is the chromatin, the alterations of the epigenome. And the alterations of the...
Marco, sorry, can you speak up a little bit?
Yes, of course. Can you hear me? Great. So the alterations of the epigenome are really, really crucial because they are really predictive of age of individuals. And DNA methylation, for example, it has been used as a proxy of epigenetic changes that occur throughout aging to generate what we call epigenetic clocks. But, you know, the field of epigenetic clocks is a bit debated because it's not yet clear whether these alterations that occur across all different tissues and cell types at the epigenetic level, they are just like some kind of the representation of some kind of random epigenetic drift, or if it's just some kind of stress response to DNA damage or other factors. So we decided to investigate whether it is possible that there are some genes or some regulators, some mechanism that could regulate the speed at which epigenetic age and epigenetic modifications occur with aging. And our first hypothesis was, okay, if there is some kind of genes that regulate epigenetic age, then we must find some clues in the gene expression profiling. Of course, it's much more difficult to look at gene expression because it's much more less associated to aging, but we decided to start with it. And our criteria would be that we need to find some genes that are, if they are regulators of epigenetic age, they have to be increased with aging in all tissues, and we want that they contribute to the aging phenotype, and possibly, if we target them, we can somehow slow down or revert epigenetic aging. So we started at the very beginning by collecting from different sources and databases a lot of gene expression profiling matched with age of individuals, and we tried to identify what were the genes that most correlated with aging in all of the tissues. And of course, we identified a lot of genes. Some of them make a lot of sense because we're associated to pathways that are very well known to increase with aging, such as p53 signaling or DNA damage repair, p53 signaling, and so on. And so, of course, it was particularly difficult to identify whether within this set of genes there is something that could be a regulator of epigenetic age. But for the moment, we decided to create a gene set out of this by selecting the top genes that behave, that mostly increase with aging across all different tissues, that we named gene set A, and we put it at the side for the moment, and we go back to it in a while. So, of course, we have to find a way to narrow this list down, because if there is something interesting within this list of genes that increase with aging, we need the possibility to further dig into this list to find candidate targets. And we thought, well, maybe if our genes of interest are important for epigenetic aging, they have to be it's possible that they have to be silenced or repressed when you have rejuvenation. And when do you have rejuvenation? Reset of the epigenetic age. So basically, it occurs in two conditions. One is physiological, and the physiological one is during early phases of embryo development and during zygotic reprogramming. Because what happens during zygotic reprogramming is that the new organism, the zygote, is born from an ovocyte and sperm cell, but the ovocyte is not young. If you look at the epigenetic clocks of ovocytes, they're not young. And what you see that the epigenetic age is reset, or not immediately, but all the way from the two-cell stage to the blastocysts. So something happens in this 4 days that somehow let's say that coexists with the zygotic genome activation that resets the epigenetic clock to a young state. And therefore, we collected many different data and analyzed all the gene expression changes that occur throughout this process. So we analyzed this like as you would do for single-cell sequencing. So we created a trajectory, we analyzed pseudotime, and determined all the genes that are shut down once you have epigenetic rejuvenation. And we have another gene set. So we select, there are thousands of genes again, 500 genes again, and we select them, and this is another gene set, and we put it to the side again. And then we look at the third condition, but different condition, where you have epigenetic rejuvenation, which is not physiologically, but is artificial, of course, and it is when you have cellular reprogramming. So when you reprogram the cells either to full reprogramming or partial reprogramming using Yamanaka factors. So what happens during reprogramming is, of course, that at a certain point in time, you have epigenetic rejuvenation, but it does not occur immediately, and it occurs in a specific time window that, depending on the experiment, can be between 11 and 15 days from reprogramming before the cells has completely lost its identity. And so we decided to understand what genes are repressed and shut down at the moment at the rejuvenation window. So when you have epigenetic rejuvenation, what happens, what is shut down? And we created another gene set here, which we call C. So what happens if we have the genes that are increased with aging, which are thousands, the genes that are downregulated, suppressed when you have rejuvenation, physiological rejuvenation, and if you match them, intersect them with what happens when you have reprogramming. And at the end, from different thousands of genes, we ended up with just one gene in common. And this gene was a non-coding RNA. It's link-pinned. And I have to confess that the first time we saw that that was one gene in common, that was one non-coding RNA, the first reaction was like, oh my God, we're never going to make sense out of this, because this is a bit like what I was about to think when I saw a non-coding RNA. But in fact, we're a bit more lucky, because this non-coding RNA is in fact a precursor of a microRNA, which is very well known because this microRNA has been previously defined as an EP microRNA, because it targets, among the top targets, it targets the many elements of the epigenetic machinery. I reported some of them here, but most interestingly, it targets both components to target DNA methylation and DNA demethylation, which of course are very important if you think about epigenetic clocks, which are based on the DNA methylation. And of course, this is a bit also slightly difficult to understand. Why would a microRNA target both and families of enzymes that would do opposite things, methylation and demethylation? But we wanted to see that maybe, we thought that maybe this was an important gene to further investigate. And we know that it increases with age, not just in human, but it's a top candidate in increasing in aging in all species, at least in mice and rats and other species. And it's not a marker of pluripotency. So it means that it's not that if you do not have cells that do not have microRNA 29 are not necessarily dedifferentiated. And of course, we know that causation and correlation does not imply any causation. So we just said, okay, so what happened if we shut down using antagomers, or if we overexpress microRNA 29 in fibroblasts, in primary fibroblasts, and also in other cell types? So what happens that we performed 3 treatments, and we did not see any toxicity. We did not, both with the mimic and the inhibitor, we did not observe toxicity. The cell growth was barely the same. But when a lot of things were going on at the transcriptional level and at the epigenetic level. And of course, we looked at epigenetic clocks, and it was great because all of the clocks we had tested, we could clearly see that there was a downregulation of the epigenetic age, there was a consistent downregulation of the epigenetic age. And given that in the previous talk we talked about retrotransposons and so on, this is particularly interesting for us because one of the clocks we have tested is what they call retro-age clock, which is really based on regions of the genome which are where the retrotransposons reside and long terminal repeats and so on. And one of the features we see when look at gene expression following inhibition of microRNA 29, you will see a total shutdown of these repeats. So we use several bioinformatics approaches and analysis to understand that repetitive elements are shut down in this context following inhibition. And of course, the rejuvenation using multiple epigenetic clocks is also supported by the data that we see at the transcriptomic level. So if we look at transcriptional signatures of aging, of course, there are the transcriptomic clocks, but if you were not want to believe at a clock, you could look at gene expression, you could look at pathways that are perturbed, and we see that following treatment, we do upregulate and downregulate many signatures of agings that are reverted, so genes that...
1 minute.
Oh, 1 minute, okay, here we are. So it's important to say that it regulates in a certain way, it regulates DNA methylation, and it regulates, oops, sorry, I just... Whoa.
You got a spoiler.
No, this is another presentation. I don't know what happened. So it's. Can we go?
You lost your minute.
Well, anyhow, this is not my presentation, so I don't know exactly what happened, but it's another file. But I can conclude anyhow. So the point is that this microRNA is particularly associated. It regulates epigenetics, and it's particularly effective in, let's say, in controlling what can be written to the epigenome. When you upregulate microRNA 29, you downregulate the epigenetic modifiers, and the cell is in a state where it can no longer write to the epigenome. In contrast, when you inhibit microRNA 29, you are in a condition when the epigenetic modifiers are raised, and it's very easy for the cells to write to the epigenome. And the epigenetic changes that occur following inhibition of microRNA 29 are much more pronounced. And of course, this is particularly of interest in the context of cancer, because microRNA 29 is one of the most downregulated microRNAs in cancer. And this is particularly important, it's last slide. It is important because we know and we have seen that overexpression of microRNA 29 inhibits plasticity, and in several cancers, such as prostate cancer, epigenetic rewiring is particularly important to develop resistance to pharmaceutical agents and androgen deprivation. And we have some evidence that overexpressing microRNA in this context prevents plastic adaptation in these tumors and also triggers senescence in this context. Okay, so it was really a flash talk, and I didn't manage to explain all I wanted, but we have a very beautiful poster outside, and if anyone is interested to swing by, I would be happy to further, let's say, detail the results we got.
Thank you, Marco. I know it's difficult to show a lot of data in so short 10 minutes.
It is my great pleasure to introduce the last speaker of this very exciting session, Raffaella Di Micco, who is going to talk about cellular responses to DNA damage in the metabolic system, from senescence to immunity. Raffaella.
So thank you, Nina, and thank you, the organizer, for giving me the opportunity of presenting today what we have been doing in the lab in the last few years in the context of senescence and acute myeloid leukemia. So just a few words on acute myeloid leukemia. Is the first talk on this. So just to introduce the disease, this is a very heterogeneous blood cancer that is characterized by aberrant proliferation of myeloid cells. And it mainly affects elderly people. Its incidence is above 55, 60 years of age. Current treatment, despite all the advances, is still based on high-dose chemotherapy. In particular, there is this 7+3 protocol that consists of a combination of Ara-C and daunorubicin. And most of the patients after this round of intensive chemotherapy then undergo allogeneic transplantation. So a few years back, we were really trying to understand why, in the context of AML, there are still patients that do not respond to chemotherapy, and the very early events that are occurring immediately after chemotherapy were not really fully understood. So we decided really to go more deep into this mechanism of therapy failure and, of course, trying to find vulnerabilities that will make these patients respond better to chemotherapy or even respond to alternative to the chemotherapy regimens. And, of course, coming from the senescence field, this was something not really clear in the context of AML, but it was very much studied in the context of solid tumors. We do know that chemotherapy or other genotoxic agents can induce this phenomenon of therapy-induced senescence. So tumors can trigger the cell cycle arrest that is characterized by induction of P16, P21, DNA damage, beta-galactosidase, and the famous senescence-associated secretory phenotype. This was described as an early response in several tumors. Apologies for studies that are not cited here, but there were work from Scott Lowe, from Manuel Celano, from Kremen Smith, also highlighting not only this cell-intrinsic response of cell cycle arrest, but also potential interplay between senescent cells and the immune system. And most studies identified, as we heard before, this interplay between senescent cancer cells and innate immunity. And more recently also, there was this discovery that these cells can even talk to the adaptive immune system, although this crosstalk was not investigated in the context of the acute myeloid leukemia. So the questions we were trying to address is there any senescence starting with in the context of AML, especially upon chemotherapy? And if any, is senescence having any role in rewiring immunological responses against acute myeloid leukemia? So we started years back, we teamed up with the lab of Clement Schmidt to trying to model these effects of chemotherapy ex vivo. So whatever I will show you today for the first part of the talk was done with primary tumor samples that we could collect through our biobank at San Raffaele Hospital. So we take patient-derived primary AM blast, and then we put them ex vivo. We administer chemotherapy. We use, in this case, RC. And after a few days of treatment, we investigated whether or not there was induction of cell senescence by several means. We did FACS analysis, we did RNA sequencing. And what was immediately clear is that despite the fact that the samples were responding to chemotherapy, if you looked only at the cell cycle arrest elicited by the drugs, immediately what we found is that we could stratify the patients into two groups that at that time we defined as senescence low or senescence high. Here are the stainings that we did with the fluorescent beta-gal. So it looks like not all the samples are able to elicit this senescence response. And when we did transcriptomic analysis of senescence high, control RSC versus senescence low patients, what we notice is that, of course, we do see in these patients activation of the p53 pathway of the DNA damage response, higher in the senescence high versus the low. But importantly, we had a lot of categories that were belonging to inflammatory responses. And in particular, what caught our attention was these categories of interferon alpha and interferon gamma response. And because we saw this interferon gamma response, we thought that maybe these cells are able to talk to the adaptive immune system and to the T cells specifically. We also looked more deeply into the mechanisms that are responsible for antigen processing and presentation in the cells. This is a very complex series of proteolytic events. And we did see that in the senescence high group that you see here, we have seen upregulation of most of the proteins that were involved in antigen processing and presentation. And if we look at now the HLA molecules, both class 1 and class 2, we do see that the senescence senescence eye have a stronger upregulation of these molecules in their surface. This was RNA sequencing. Of course, we validated this by FACS analysis to look at the protein level. And again, it was clear that the senescence eye upon ARA have this induction of class 1 that we could see by intensity, and also higher percentage of cells expressing the HLA-DR that is instead HLA class 2 and is more responsible for the interplay between AML and the CD4 positive T cells. So having addressed that there is this rewiring at the immunological level, we also tried to set up a series of quite complex experiments in which we co-cultured the leukemic cells from Senlo or Senai with CD4 or CD8 positive T cells, and we looked at some functional readouts in the T cells, in this case mainly proliferation. And again, when we do this co-culture, we could see that in the context of the Senai that were co-cultured with the blasts exposed to chemo, we do see higher proliferation, again as a surrogate of their activation, and this was true for both CD4 and CD8 positive T cells. In contrast, in the context of the Senlo, we were not able to rescue or to activate this immune response. And importantly, we also did a series of experiments that showed that this activation of T cells was dependent on HLA, because we also developed knockout for beta-2M or blocking antibodies of the class 2, and we could see that we could rescue this T cell activation when HLA are inactivated. So I showed you that we have more presentation in this subgroup of senescence eye patients. Is this associated with more presentation of peptides? So to do so, we teamed up with the group of Vincenzo Cerullo in Helsinki. And what we did from 3 patients of our senescence low group and 3 patients from our senescence high group, we did mass spectrometry. So we pulled down HLA-1 bound peptides, and then we started characterizing these peptides. So here I'm showing you the data that we retrieved from these patients. Again, we did this at the diagnosis, therapy naive, and ex vivo treated by RSC. You can immediately appreciate that the Senai patients, even in the control UT, we retrieved at least in 2 out of 3 more peptides, so suggesting that in this case they are already starting with before chemo more able to present peptides. But we also saw that upon chemo, the peptides that we retrieved were increased, specifically in the senescence high cohort and not in the senescence low. We also did some gene ontology analysis to check whether these peptides, if they are associated with specific processes. And it was interesting for us to see that we have many more of these in the Senai group, as you would expect. And most of these belong to pathways that are involved in the antigen processing and presentation, immune crosstalk. We also saw some viral mimicry that is also something very important in dictating activation of the adaptive immunity. And again, these responses were much stronger in the senescence high than in the senescence low. So, of course, we did our modeling ex vivo because the idea was to have a reductionist but still valid model to test our hypothesis. But we also wanted to see whether or not this immune stimulation would occur in vivo. And to do so, what we did was to engraft with primary leukemia immunocompromised mice. And after 2 months of engraftment, then we started modeling chemotherapy in vivo. In this case, we used RSC. After 8 days, we modeled senescence HLA activation. And as you can see here, this is a pool of experiments that we did, 4 independent experiments. When we use the chemotherapy, we are able to reduce the burden of leukemia. We do see upregulation of HL1. We do see induction of senescence in our model. And then what we asked was whether now these cells will better be recognized by the adaptive immune system. So what we did was to infuse our mice that were control treated or chemotherapy treated with autologous T cells coming from the same patients, and these cells were previously expanded according to a clinical protocol for 3 weeks and then infused in the animals. We also decided this time to apply a checkpoint inhibitor, nivolumab, just because from data that I didn't have time to show, we do see upregulation also PD-L1 on senescence plus, so we thought that they may synergize with ICB. And what we found was that, of course, chemotherapy reduces the burden. This burden is further reduced when we co-infuse, we co-treat RSC treated mice with T cells. And these lead to complete eradication of the mice when we combine the chemotherapy, T-cell infusion, and nivolumab. And here is another representation of the data in which we could really stratify the mice in mice that had or not leukemia burden by putting a threshold of number of blasts in the bone marrow. And you can see that this combination, RSC, T-cells, nivolumab, resulted in all the mice without leukemia. So it's really synergizing and potentiating this senescence induction with immunotherapy. Okay. Of course, what we don't know yet and what we were trying to understand is why some of the patients are competent in inducing senescence and some other are actually not competent. And we looked at the genetic alterations. Leukemia is very complex. There are different mutations, different karyotypes. So we did an NGS panel for 106 leukemia-associated genes. We also looked at karyotypes. Even if our cohort is not a huge cohort, and this may be a caveat because we only had 6 samples that were from the Cellular and more samples from the Senai, but we couldn't really find with this number any correlation between the genetic makeup of the tumor and this senescence competence. So we decided to look into possible alterations at the level of the epigenetics and the epigenome. And the reason was that we had some evidence that some of these changes in the senescence competence may really be dictated by histone post-translational modification. So together with Tiziana Bonaldi at IO, we did proteomic analysis, nuclear proteomic analysis to really quantitatively assess the levels of modification on histones. We assessed by mass spectrometry more than 30 post-translational modification, and then we looked in this Cellular versus Senai, these are therapy naive, so at diagnosis, if there were differences in terms of their epigenetic landscape. This was not the way it looks, but I hope you can appreciate that we could cluster the patients into 2 groups. So what we found is that the senescence high patients have an increased level of markers that are associated with active transcription. We do see increase in H3K4 trimethyl, H3K36 trimethyl. On the other hand, we do see that the senescence low have increased the level of repressive chromatin marks. In particular, one of them caught our attention, and this is the H3K27 trimethyl. And you can see across samples with this quantification that indeed we have less of the K27 trimethyl in the senescence high and more in the senescence low samples. This is a modification that is associated with polycomb repressor complex 2, and the reason why we were excited about it, first of all, there is a link for years now between PRC2 regulation and the senescence program, and also a few years back we could show that PRC2 is also repressing and controlling the expression of HLA molecules of leukemic cells in the context of relapse post-transplant. So we thought that maybe what happens here is that the presence of these polycomb repressive marks on class 1 and class 2 and possibly interferon genes might make these samples less ready to undergo, less prone to undergo senescence. So there is a drug that inhibits the catalytic activity of PRC2, that is tazemetostat. So we tried to test whether tazemetostat could rescue the immunogenicity of these samples. So we focused on the senescence low. We treat them ex vivo We did RNA-seq analysis in which we do see that most of the categories of the senescence were rescued by this treatment in these senescence low samples, together with a lot of categories related to inflammation and interferon. We do see upregulation of class 2, and although not significant, a trend towards upregulation of class 1. But most importantly, if now we do our co-culture with senescence low samples that were pre-treated with tazemetostat, we could rescue the activity of T cells to proliferate in this context. So suggesting that this could be a valid alternative to rescue the senescence-related immunogenicity in patients that are not competent in the context of chemotherapy. So to summarize what I showed you here, we showed that ex vivo chemotherapy triggers this senescence response in a subgroup of leukemic patients. We saw upregulation of HLA and co-stimulatory molecules on senescent blasts. We do see this rewiring of the immune peptidome that is so far descriptive, but very interesting because it means that these cells are able to present peptides that are related to processes that regards inflammation or senescence itself. We do see an activation of HLA mediated of CD4 and CD8 positive T cells, and somehow PRC2 inhibition seems to enhance the immunogenicity of these senescence low samples. And we also could show that there is a synergy between senescence-inducing therapy and T cell infusion and checkpoint blockade in the context of acute myeloid leukemia. So all of this was done with human samples. We just assessed and checked the activation of T cells, but there is such a complexity in the bone marrow, and there are so many cell types that could play a role, especially when administering chemotherapy in vivo, that we could not really assess in this study. So to do so, we moved to the mouse model again, because we thought that if we do our study in an immunocompetent mouse model of leukemia, now we can better ask this type of question and really address the complexity of the leukemic microenvironment in the context of senescence-inducing therapies. And so far, we developed 3 mouse models of leukemia with different mutations, NPM1, FLT3, MLL-F9, MLL-F9, and RAS. And the idea is really to investigate how this senescence induction, if any, can rewire the immune microenvironment, and if we can find vulnerabilities to really potentiate immune-mediated eradication of these senescent cells. So we started with the MLL-F9. This is a widely used leukemia model with pros and cons. It's driven by this translocation that affects the interaction of the MLL gene with other proteins. So it has some epigenetic regulation and activation of pro-oncogenic programs. It's still relevant because 5–10% of human patients, they have this type of translocation, and it's generally associated with poor prognosis. So we took these leukemic cells and we transplanted them into recipients. So in this context, we can really ask what is the contribution of the leukemic cells and what is the contribution of the host immune system. We model in the mice chemotherapy exactly like we did in the human setting, so by combination of Ara-C and daunorubicin. And what we notice is that in this model, chemotherapy is effective in reducing the burden of leukemia. However, when we tried to see whether or not there was induction of senescence in this context, we could not see upregulation of senescence-associated beta-gal. It's also, we didn't stop there. We retrieved the tumor cells from the mouse and we applied single-cell RNA sequencing to really check the transcriptomic engagement of the cells upon chemotherapy. To our surprise, these tumors were also highly heterogeneous. This is not something we would expect. We could divide it in 5 different clusters based on their transcriptional features and differentiation capacity. This is the composition of the cluster. You see how these clusters are changing upon chemotherapy administration. We do see reduction of the monocytes, inflammatory clusters, and some rewiring of cluster 2, the GM2 cluster. But then we digged into the transcriptional changes, and what we found is that Just this small cluster of monocytes at GMP1 were able to upregulate p53. Instead, the other clusters were not engaging a productive p53 DNA damage response. And importantly, what I have highlighted here is that we do see upregulation of MYC targets in these clusters. And to our surprise, when you looked at the inflammatory gene category, the interferon category, these were even downregulated in these clusters upon chemotherapy compared to the other two. So suggesting that we may mimic an immune suppressive microenvironment with this mouse model that has nothing to do with the positive effects of senescence induction and immune rewiring. We looked at CDKN1A, P21, and P16 levels. They are very lowly expressed across the different clusters. We looked at signatures of SASP that were coming from mouse studies, and you can see that actually there are some clusters that are more inflammatory starting with, but then the more you go into the cluster analysis, the less inflammation you get. But especially if you look at the effect of chemotherapy, we did not see increased inflammation, as was shown by the GSEA that I showed you before. So suggesting that in this model so far, we cannot capture senescence, we have reduced the inflammation. So why is that? It's still pretty much ongoing, but because in the context of the senescence low patients, we uncovered this repressive chromatin. We looked at distant markers in the context of leukemic cells in this mouse model. And it was nice to see that in the two clusters in which we do see this slight p53 activation, we also see activation of this enrichment of genes marked by K27 or K9 repressive markers, but they had a completely opposite trend when you looked at the other clusters that showed reduced inflammation. And this was nicely correlating with the level of EZH2 itself that was higher in the context, as you can see, in these clusters of leukemic cells in which we do see this repressor, a block of inflammatory response, suggesting that even in this case, this could be a vulnerability that we are going to test. We also looked at the immune system, of course, because the idea was to look at the rewiring of the immune system. We did the transcriptomic analysis on the immune microenvironment. We could identify several clusters, 21 in total, so trying to capture different cell types and different states across cell type. And just to walk you through some of the data, if you looked at the T cell or NK, you have a reduction upon chemotherapy of this fraction. No interferon gamma, not any inflammation that would be suggestive of a T cell activation, again, in line with the fact that in this context, we are having an immune suppressive macroenvironment. We do see also reduction in the B cells in number and in the programs that they are activating. So again, it looks like in this context, we are not really capturing this immune-mediated activation. We looked at the monomacrophage population. And here some things come of interest because there are some subsets of macrophages that do not change in number that were characterized by reduction in interferon alpha response. So again, suggesting that they may have features of immune suppressive macrophage that may block T cell activity. And we also looked at neutrophils because this is the subset that we found increased in the leukemic bearing mice upon chemotherapy. And we are now really trying to understand what is the role of these different subsets and whether there is a gene network that we could target to really potentiate and make again now the immune system capable of seeing and eradicating these leukemic cells. So to conclude, this is my last slide. We show that in this mouse model, we found that leukemia is very heterogeneous. We do see different clusters of leukemic cells that may have different response to chemotherapy. We do see that there is some reshaping of the immune microenvironment. It's more towards the immune suppression than the immune activation. And we are trying to understand whether or not, even in this context, epigenetic mechanisms that are entailed by the tumor cells may now dictate this immune suppressive microenvironment, and whether we can now modulate this in order to rescue the ability of the immune system to fully eradicate leukemic cells. So if you want to know more about the two stories, Simona and Alessia are having a poster over lunch, so you can go and get more details. And last but most important slide, people that did work. I acknowledge Simona and Alessia. Antonella is also the postdoc driving the mouse model story in AML. Lot of people, collaborators, funding agencies, and of course I'll thank you for the attention, and I am happy to take questions.
Thank you very much, Raffaella, for this very exciting talk. There is one question here.
That's really exciting, Raffaella. Good job. So do you know whether levels of H3K27 trimethylation in human AML blasts predict response to therapy, whether to genotoxic therapy or, for example, to venetoclax and azacitidine, which is also quite wide used?
We are looking into this. We are trying to see whether or not the levels are really... What we know from our data is that the senescence law, that at least by the ex vivo treatment are not able to activate senescence and do not trigger the immune system, this might be a limiting factor. So we didn't look at the patients, but this is something very interesting that we want to do. We want to look at patients that are also resistant to venetoclax. So it's a different cohort of patients. So we're trying to get with clinicians the hands on these type of samples to also ask this very relevant point. No one looked so far at the epigenetic and venetoclax. Well, not no one, because there is an accompanying paper to the one that I showed from Clemens that has more clinical correlates that are important, no? The response to therapy. And in that setting, we contributed by showing that the senescence eye are also the one that respond better to chemotherapy. So of course, we also looked at azacitidine. Maybe Clemens can give you more details on this. There is some good, usually we do see that senescence induction correlates with better response. But what you are asking, I think it's very relevant, because now in leukemia, especially the resistance to venetoclax and also giving a biological rationale for using a venetoclax is not really clear to me. Clinicians give these combination therapies of aza, venetoclax, but they not really grasp the biological why. Like it's more like is the best option to treat patients. We believe that studies like this can inform better clinicians in the choice, because already by this ex vivo treatment, we could predict which one will respond better. So it would be great to do the same for the venetoclax.
Clemens.
Raffaella, congratulations for getting the samples to run experiments in an autologous setting, which is, from a clinical perspective, really difficult to obtain. So you demonstrated that the ability to induce senescence, TIS, travels with a better T-cell response and better outcome. But the same T-cells or T-cells were obtained at the time of primary diagnosis, though these T-cells have seen the drug-naive disease, the AML of the patients. Can you help us to disentangle by your immunopeptidome analysis whether you found rather anti-leukemic peptides or anti-senescence peptides? And to spin the wheel further, you refer to allogenic transplant later on, where the clinicians believe they target the normal hematopoiesis and graft versus hematopoiesis effect, which helps controlling the disease. Would you be claiming that allotransplant is an anti-senescence treatment on the T-cell basis?
So many questions. So I start for the first one, that is the one of the autologous context and the fact that we could mimic this response by using T-cell diagnosis. So at the beginning, we were also a little bit surprised by this data, because you would expect that if this is an antigen-specific response, the clones of T-cells presented diagnosis are very few, or they may be generated upon chemo, as you would expect. So in vivo, they've seen the chemo chemo, they've seen the activation of the senescence. So if you take T cells later on from the chemo, you will get better responses. So what we did and what we think is that more than enough, this goes back to your peptidome questions. So we do see a lot of senescence-related peptides. These are not neoantigens. These are self-peptides that are just more expressed by the senescence blast on the surface of HLA molecules. We do see some leukemia. It's a combination of three aspects. We do see senescence. We do see AML, related peptides, but again, more towards the higher expressed in leukemia than new antigen. And we do see also this viral mimicry stuff, so a lot of things that are related to viral cycles or infection that might be also calling for these retro elements, reactivation. This is something we didn't check in the studies, but could be also a relevant point. The question of the transplant, I would say that as allogeneic transplant is to me the very first form of immunotherapy. I'm not a clinician, but I think that in these graft versus leukemia effects, you are just giving an immune system that is very potent in eradicate the cells. I would expect that the senescent cells will be I recognize more, so that the senescent competent patients will also respond better to transplant. We tried to do this type of correlation. The number of patients was very limited, but maybe in a bigger cohort, we can also try to see whether senescence competence correlates with a better response to the allo.
Thanks. Another question there.
I have much simpler question. So initially you had a patient grouped into sen high and sen low groups, and later you showed us this nice mouse model, MLL-AF mouse model. And then I was wondering, and then you also mentioned that this mouse model have a heterogeneous response.
Yeah.
And I was wondering, can you also mimic or like recapitulate this sen high and sen low group of patient in your mouse model, or does it just belong to one of these two groups?
This is another great question. So I think what we looked only at this mouse model with the MLL-F9, so it could still be that the MLL-F9 model is recapitulating the senescence law that we saw in the primary samples. I have to say also that we had some interesting data that we need to follow up, is that it's also, this response may also depend on the burden of leukemia in the bone marrow and the time of treatment. So if we treat the mice when the burden is particularly high, then we are not able to activate senescence, we are not able to engage the immune system. Instead, if we treat the mice that have a lower burden in the same mouse model, we are able to see some of these responses. So we really want to see if also the type of number of blasts, persistence in the bone marrow can make a difference. And of course, we also want to test other genetic alterations to see whether or not we recapitulate the same findings of the immune suppressive environment.
So meanwhile, Raffaella, one question. So you show very nicely that blast cells high and low from human-derived samples, right? So which is super cool. They show different responses, right? So I was wondering, have you taken into consideration at any point HSPCs in that? So
you mean like in terms to look at the normal, let's say normal leukemic counterparts? We didn't do. There are some studies though of chemotherapy response in vivo where they looked also at the stem cell compartment. There are studies in which, you mean stem cell of the leukemia yes. Stem cells. So we data mined some single cell RNA sequencing that were previously published in our institute to check whether or not this response could be dictated by different percentage of leukemic stem cells in the two patient groups. And to be honest, when we try to deconvolute our RNA-seq data based on the single cell that was published, we did not see that the senescence law had strikingly higher fraction of leukemic stem cells. Mm-hmm. We do see 5-10% of these early progenitors, but the majority of the senescence gets into the myeloblast compartment. Mm-hmm. So, and in that term, the composition was not that different. So I don't think that this is the key. in the early response, but it could be the key for the relapse. Could be that this senescence low have also this fraction of leukemic stem cells and then relapse more. So in the later time point, this could make a difference. In the early response, we couldn't really find the link between stem cells and blasts.
Okay, cool. Thank you. Last question.
I had a question, but I think you just answered to that question. So I was wondering if the mouse data could inform you about differences that you see in human data. Because what you saw in the mouse data is that there is actually a state, perhaps a stage of differentiation or you know, a dynamic state in the myeloid differentiation that appears to be more sensitive, right, to senescence. So I was wondering whether, you know, regardless of the stem cells.
We tried to see whether this response was informed more by the differentiation state. It's also something you would expect because it's known that maybe patients with a more differentiated leukemia, but we couldn't really find it. So it seems that it's more a general response that you get in a patient's cohort, but not in the other. And we're still trying to see whether instead it's more the epigenetic that dictates this response. But it's a good point.
Thank you very much, Raffaella, for this great talk. And I would like to also thank all the speakers from this morning session.
Please, can you take a seat? So to chair this section, which is more translational and clinical, we have the pleasure to have two chairmen. One is Alessandro Ceschi. Alessandro is the director of AFRI, which is the department devoted to research and education at the local hospital of Ticino, which is called EOC. He is also the president of the Swiss Clinical Trial Organization, so he's just been elected. The other one is, on the contrary, a businessman, also with a previous past in science. So Giovanni Rizzo is the founder of a venture capitalist fund, which is called Indago, which is one of the biggest in Italy. So thank you guys for accepting to chair this section.
Thank you.
Thank you, Andrea. Welcome everybody to this afternoon session. We're starting the Translation to Clinic session, the session 2. And the first speaker, I have the pleasure and honor to introduce James Kirkland, who is a specialist in internal medicine, geriatrics, and endocrinology. He's the director of the Center for Advanced Therapeutics at Cedars-Sinai in Los Angeles, an emeritus professor of medicine at Mayo Clinic, and the principal investigator of the NIH Translational Geroscience Network. Thank you.
Thank you very much. And I wanted to thank you for chairing this session, the organizers, and also Sarah Komodo for the fantastic job she did in organizing my flight. Unfortunately, because of that, I'm jet lagged, so I'll probably make no sense. People in my lab will tell you I don't make any sense anyway, but this is going to be worse. So we've already heard definitions of what senescence is and what can cause this cell fate. I'll just spend a second, though, reiterating a couple of things. First of all, it is a cell fate. It appears to be able to occur in almost any kind of cell at any point during life, including in oocytes, before they're even fertilized. It occurs across the vertebrates, can occur in many invertebrates as well. And there are many things that can induce a cell to become senescent. I may come back to some of these things as there's time, but some but not all senescent cells have an increase in P16 and or P21, but there appear to be other transcription factor cascades that can enforce the process. Most, if not all, senescent cells develop a senescence-associated secretory phenotype, but this is highly variable. It depends on the cell type that became senescent, how long it was senescent, what induced senescence, and very importantly, the microenvironment. I'll come back to that in the context of cancer. And senescent cells are resistant to dying. As we've heard, they're normally only removed by the immune system. I'll come back to that, but they're also protected from dying in hostile microenvironments by senescent cell anti-apoptotic pathways. So if you selectively eliminate senescent cells, and this is the case with very highly P21 expressing cells, very highly. Every cell expresses some P21, but senescent cells certain subsets of them tend to have extremely high levels. If you inducibly, selectively remove these cells, you can get some degree of extension of lifespan, perhaps, an extension of health span, but it appears that animals with this kind of mutation die later from virtually every disease that mice naturally die from, including most of the cancers that mice die from. Conversely, if you transplant small numbers of senescent cells, you can induce, these are autologous ear fibroblasts made senescent either by radiation or chemotherapy and reimplanted into the mouse that they came from with a luciferase construct embedded so you can see where the cells are. They tend to track to the peritoneum, they stay there. But the animals that are transplanted after a few months develop a frailty syndrome, provided you transplant enough senescent cells, and they also, after a lag period, die early of basically all diseases that mice naturally die from earlier, including most cancers. So we're fulfilling Koch's postulates, basically, that if they're, senescent cells appear to be linked with cancer, you reduce their abundance, you delay onset of most forms of cancer, you increase their abundance, and it occurs earlier. Now, the other thing is that senescence spreads from cell to cell, not only locally but systemically, and that can be shown in these transplant models. There's a threshold effect when you're looking at whole body situations. So if you transplant 1 million senescent cells into a middle-aged mouse, such that 1 in 10,000 cells in that mouse is a senescent cell, that's sufficient to cause that middle-aged mouse to develop the frailty syndrome that I mentioned and to die earlier of age-related diseases. If you transplant, though, 500,000 senescent cells, almost nothing happens. If you transplant 500,000 senescent cells, though, into an old mouse, they get this syndrome. Or if you make a mouse obese, which is a condition that can drive endogenous senescence, they will develop this syndrome. So the total of transplanted plus endogenous senescent cells appears to do things after a threshold is reached. And there's some thought that this may be because senescence spreads, as I mentioned before, and it may be that at a tipping point, the rate of spread of senescence exceeds the ability of the immune system to clear senescent cells. And then after that, senescent cells start interfering with immune clearance of themselves. And being a Canadian, I think about hockey sticks. And I'm living in California now, the 11th province of Canada. But like a hockey stick, there's an accelerating incidence of multiple disorders over time. And this appears to be associated with accelerated onset of multiple fundamental aging processes, including cellular senescence. And one theory is that senescence, one of the hallmarks or pillars of aging, is linked to most, if not all, the other hallmarks. And if you target senescent cells, you affect the other hallmarks. Conversely, if you target another hallmark, you tend to affect senescence, usually in the same direction, so-called unitary theory. So we've heard before, I'm not going to belabor it, that senescent cells are normally removed by the immune system. And there are many, many immune cell types that can target and remove senescent cells. Some of the understudied ones include tissue-resident immune cells, like dendritic cells and tissue-resident macrophages. And arguably, senescent cells can be considered part of the immune system, the third arm, as it were, of the immune system, in addition to adaptive and innate immunity. So back in 2004, when we were in Boston, we started trying to figure out ways to eliminate senescent cells. And that was based on Ned Sharpless's work. He became director of the National Cancer Institute, but he published a very important paper in Journal of Clinical Investigation in 2004 that led us to think this way. And what he found after Serrano's findings about P16 and senescence was that senescent cell the rate of senescent cell burden increase could be delayed by interventions that arguably, under certain ideal conditions, might extend lifespan in mice under very artificial conditions like caloric restriction. Caloric restriction would kill any normal mouse, be the first one to be eaten by a cat. But in the interventions testing program, where they're highly protected, they tend to live longer than other mice, but they don't live that well. You know, they're sleeping 20 hours a day, they don't have sex, etc. But they do live longer. And the same with rapamycin and so forth. So that correlation between senescent cell burden and interventions that can extend, arguably, lifespan led us to ask if that was causal or an association. So we started trying to develop Senolytics, and we started by trying to get antibodies that Jim Smith had developed and couple them to diphtheria toxin fusion proteins. That didn't work. Then we tried high-throughput screening with Scripps in San Diego. We did get some molecules. They looked like they were senolytic back around 2009, 2010. We didn't pursue them very far, though, because they seemed toxic. And then it hit us that there are at least some senescent cells that appear to kill the cells around them, but they themselves don't die, much like what you see with certain kinds of hematopoietic, you see with certain kinds of leukemias and lymphomas, where the cancer cells survive despite the fact they're killing cells around them. So we used proteomic and mass spec data that Izu and our group had. And we went to University of Oklahoma and Jonathan Wren there, who actually invented text mining back in the early 90s. He had 7 Cray computers in a row. And we did some early probing of these mass spec and proteomic data. And what we found that indeed there appeared to be upregulation of anti-apoptotic pathways in senescent cells. We queried that with small interfering RNA analyses, and this is the network that we found using the computing approaches. But we queried this with small interfering RNA approaches, and we found that different kinds of human senescent cells depend on different elements of these pathways that are interconnected in a network to survive. So targeting some of these particular gene products would kill human preadipsites, or MSCs, but not endothelial cells, HUVECs, whereas other ones would kill endothelial cells, but not preadipsites. So it seems that different components of this network are utilized by different kinds of senescent cells to defend themselves against dying. In the case of preadipsites, it was mainly ephrin-dependent kinases, SARC kinases, that were important. Ephrin receptors are like other dependence receptors. They detect cell-cell contact. If they're not occupied, they will kill the cell. They're one of the 17 dependence receptors, much like testosterone or estrogen receptors. If they don't have the sex steroid bound to them, they will kill the cell. This is a basis for treating prostatic or breast cancer, for example. Whereas HUVECs did not depend on these pathways. They depended more on BCL2 related pathways, especially BCL extra large. We used programs at the Broad Institute to look for agents that would target these pathways. And one of the few tyrosine kinase inhibitors, one of the few that targets things like ABL and so forth, that also targets ephrin-dependent SARC kinases is dasatinib. Other general tyrosine kinase inhibitors or ABL inhibitors like imatinib do not, and they're not senolytic. So whereas dasatinib is in the case of preadipsites, and we feel that's because of this effect on the ephrin-dependent kinase. We predicted it would kill senescent preadipsites. It killed the 30 to 70% that are pro-apoptotic and pro-inflammatory. The other 30 to 70% are not, what we call ticking time bomb senescent cells. They have a very quiescent SASP. Quercetin, one of the 4 naturally occurring flavonols, which is senolytic, we predicted based on these early programs would, or early data, would kill senescent human endothelial cells, but not preadipocytes, and that turned out to be the case. The combination killed both. We found around 40 drugs using this initial approach. They all turned out to be senolytic, but we decided to work initially on these agents because dasatinib has been in clinical use since 2004 and is off patent now, but still very expensive. And quercetin is a natural product. They both can be given orally, and they both have, importantly, very short elimination half-lives. So dasatinib's elimination half-life is around 3 hours. Fizetinib's first-pass elimination half-life is under 5 minutes. Its total elimination half-life is around 2.5 hours. And I'll come back to why we felt that was important. Using that original mechanism-based approach, we and others found many other agents that are senolytic. And these include not only things that target pro-apoptotic pathways, but also things that target the Warburg shift that senescent cells have. So senescent cells depend on glucose, much like cancer cells. They can't use fatty acids. So fatty acids of some types will kill senescent cells, and interfering with glucose metabolism, like with SGLT2 agonists, which cause glucose to be exported from cells, will also kill senescent cells. So it turns out that the SGLT2 inhibitors are partly senolytic and partly senomorphic. That was work done by people, by a person in our lab when he was with Toru Minamino, Dr. Suda, before he joined our group. Zoledronate, we worked with the group in Oxford who, over about 30 years, found that zoledronate, an injected bisphosphonate, is associated with, in long-term human studies, actually increased survival, increased health span, and decreased cancers. We looked at it with the Oxford group, and we found indeed it's partly senolytic and partly senomorphic. So there's some randomly identified agents that turn out to be senolytic or senomorphic. Other approaches, I'll talk about immunomodulators briefly. CAR-T approaches have been developed. Masayoshi Suda, who's now in our group, discovered the first vaccine against senescent cells. And since we've discovered a number more. There are SA beta-gal activated toxins. There's some microRNAs that are senolytic, some of which are about to be published. There are nanoparticle toxins. There are an interesting group of agents that are photodynamic senolytic inducer molecules, the trilock PDSIs, that are activated by light. And this looks like it's going to be very interesting in respect to liver fibrosis, because the light signals that are used to do this will penetrate the body, but you're not giving radiation. So this may be a way of getting rid of senescent cells in a particular organ. Senescent cells they're larger than normal cells. They are where most of the increase in mTOR that occurs with aging is. They produce misfolded proteins, and senescence can in turn be induced by misfolded or aggregated proteins like amyloid, tau, or synuclein. But if you've looked at them under the microscope, you've probably noticed that they've got a really weird cell membrane. They've got a lot of blebs on them. They're producing exosomes like crazy, but they're very fragile. That's why it's hard to use cytoph to isolate senescent cells. So there's some interesting work going on looking at using particular vibratory frequencies to kill senescent cells. And this can be done by using Doppler methods. And it may be possible using 3D Doppler vibrational methods to kill senescent cells in particular organs, much the way that I talked about with the trilocked PDSIs. Electrical fields, senescent cells are susceptible to particular kinds of high-frequency electrical impulses. And there's a trial going on now at Cedars of using this kind of approach to kill senescent cells in the duodenum. And it appears, don't know what the final results will be, but it appears that it may benefit type 2 diabetes. So there are a lot of innovative approaches coming along other than antibody approaches, small molecules, and so forth. The classical senolytics can act very quickly. So it takes a few minutes exposure to dasatinib or quercetin, for example, or fisetin, to initiate apoptosis in human senescent cells, in this case, taking senescent cells from obese diabetic individuals. And that apoptosis process takes 18 hours or so to complete, but a very brief exposure is sufficient to do this. It looks dasatinib and quercetin will clear around 30 to 70% of senescent cells in vivo as well as in vitro, but remember there are 30 to 70% of senescent cells that are resistant to these kinds of senolytics. Because senescent cells generally don't divide, although as we heard earlier, if they do divide, they tend to divide as cancer cells. If there's been an induction of senescence, say by focal radiation to a leg, and that's what we did in these mice a couple of months before this picture was taken, the hair on the leg goes gray and the mouse has trouble running on a treadmill. If you give a single dose of senolytics in that mouse's lifetime, that can partially restore, at least, their ability to run on a treadmill. That's because senescent cells, unlike cancer cells, don't come back unless there's a reason for them to come back, unless there's an impetus for new senescent cells to form. By the way, it typically takes a week to 6 weeks for senescent cells to form in vivo, using some of the models that we have, with the exception of iron-induced senescence, which can be much faster. So this means that if there's continued impetus to develop new senescent cells, you can arguably give these drugs in a hit-and-run fashion. They act very quickly, within a matter of minutes. We're using drugs with short elimination half-lives, and senescent cells take a long time to return, if there's a reason for them to return, like continued obesity, for example. So in the clinical trials, these drugs are being given intermittently, and some of the groups, including a group at Mayo working with us, found that intermittent administration under some circumstances is as effective or more effective than continuous administration. This is positive because these agents have very short elimination half-lives. So this is a way of reducing off-target side effects, potential side effects of these agents. Because if you're giving them once a month and their elimination half-life is 3 hours, that means they're gone within 9 hours. So the rest of the time, the drug is not there to cause adverse effects. Frailty after whole body radiation is alleviated by senolytics. I think Nathan Brasser will probably speak more about this aspect of things. Senolytics alleviate physical dysfunction in old mice. If you transplant senescent cells into middle-aged mice, as I talked about before, you can alleviate that syndrome they develop by treating them with senolytics for a 5-day course. And likewise with organ transplants that we did with Stefan Tullius, who's head of transplant surgery at the Brigham in Boston. If you transplant hearts from old mice to young mice, you cause dysfunction in the recipients. If you treat the donor, the heart itself, or the recipient with senolytics, you greatly reduce the mortality that occurs after transplanting old hearts compared to young hearts. So clinical trials are beginning. They're only at the preclinical phase at the moment using organs that are being discarded. We're currently throwing away, for example, 24,000 kidneys a year in the US, just because they're from older donors. So these agents act quickly so they can, and they also act with cold as well as warm perfusion. And so they can be given while the organs are on life port machines or other things. So these trials are commenced, they're beginning at that stage with kidneys. Cedars has the largest heart transplant program in the world, so we're beginning this for heart transplants there. And then there's a trial for liver transplants in Europe. It turns out that the combination of dasatinib and quercetin do not target P16 expressing human macrophages. We don't know with other immune cell types, though. Senescent cells, as we've heard about before, have upregulation of checkpoint inhibitors, much like cancer cells, except the checkpoint inhibitors are different. One of the more important checkpoint inhibitors in senescent cells is PDL2, not PDL1. PDL2 inhibitors act on the same target as PD-L1 inhibitors, but they also cross-link to another target that I can't go into right now. And we do not see the autoimmune effects that you see with PD-L1 inhibitors. You know, with PD-L1 inhibitors, people are getting diabetes and all autoimmune diabetes and all kinds of other things because of the PD-L1 inhibitors. We don't see that to the same extent with PD-L2 inhibitors. And there are other checkpoint inhibitors that senescent cells use that are a bit different from what cancer cells use. Manuel Serrano did this with Salim Tribe, working with them, and we were involved in this paper as well, looking at combining chemotherapy and PD-L2 blockade in the case of mice that develop mammary tumors at around 9 weeks of age. They develop an adenoma, and at 13 weeks they develop a breast cancer. And combining doxorubicin with PD-L2 inhibitors resulted in improved cancer regression. This also appeared to be the case in human cultured cells for human breast cancer. So trials are hopefully not too far off looking at this particular situation. Apigenin is another flavonoid. It's not a flavonol. It's xenomorphic. It's not a senolytic, but it attenuates SAS-promoted malignancy and reduces chemoresistance in the case of a mouse model of prostatic cancer. Birinapant is another interesting molecule. It targets the cellular inhibitor of apoptosis protein 2, and it's looking like it may be promising with respect to glioblastoma in the case of orthotopic brain tumors. So there's emerging evidence for benefits of senolytics on a range of conditions. One of the things that has been established and has just been renewed is the Translational Geroscience Network. And it includes 8 centers across the US. There's a sister network that just started in Canada 5 months ago. And there are discussions about setting up a network like this in the United Kingdom and also the GCC. So what this network does is it assists with study design and dealing with regulators. In the case of the US, the FDA. In the case of Europe, some of the trials are going on in Europe, the EMA, and getting the same language into the consent protocols and trying to harmonize protocols as much as possible so that the same kinds of testing are done irrespective of the indication of the agent, what the agent is, and what the outcomes are. Part of this is a facility for geroscience analysis, which is takes various samples from these trials, as I'll explain in a moment, and analyzes data coming from them in one place, in one way, and then biobanking of samples, which are about to be stored at the biobank in the National Institutes of Health in Frederick, Maryland. And then statistical and data management are done centrally. So currently there are 87 clinical trials underway that look at interventions related to fundamental aging processes. Some of these are lifestyle interventions like various diets and that kind of thing. Some are of xenomorphics like metformin, rapalogs, JAK-STAT inhibitors, envisaged doing this with some others, anti-inflammatories, and then also looking at NAD precursors and other kinds of things. Around between 25 and 30 of the trials are with Senolytics. And again, these are for a range of conditions, right from Down syndrome, which turns out to be a senescence-driven disorder that's associated with hematologic cancers, but lowered risk, actually, of solid cancers, to various other kinds of cancers, but also things like space travel, trying to grow better wool from sheep, using senolytic shampoos and argyle sheep, to post-COVID syndrome, to persons living with HIV, including looking at cancer incidence in those people, frailty, and a whole long list of other things that I don't have time to go into. One of the first trials, it was a phase 1 trial with all the problems these have. They're open label. There's a lot of bias involved in these things. It was a very small trial. It was partly based on Nathan Lebrasser's, who's going to be speaking, model of bleomycin-induced pulmonary fibrosis. And this trial was headed by Jamie Justice. And for what it's worth, the phase 1 trial, it did show that, and people with pulmonary fibrosis, as you're aware, they get shortness of breath eventually, but the real thing they get is frailty, and that's what tends to kill them. So when I see IPF patients, the thing that is the most bothersome for them is frailty. But for what it's worth, there appeared to be an improvement in 6-minute walk, 4-meter gait speed, chair stands, and short physical performance battery score. So we're trying to get to the point of doing a phase 2A trial and looking at trying to get funding for that. It appears that Senolytics will clear at least some senescent cells from obese diabetic, younger female patients with early renal dysfunction from their adipose tissue. And based on this, David Allison came up with the first biomarker score that responds to interventions based on that small trial. And the factors in it appear to be holding across a number of the other trials. So biomarkers of fundamental aging processes are going to be important for these trials. Mere biological clocks aren't enough. They'll be okay as predictive things, but some of these biological clocks actually get worse with interventions. What we need is things that get better with interventions, predict which intervention to use, and predict also if the patient's going to respond to that intervention, and tell the physician which intervention to prescribe next and in what sequence. And we need to meet FDA and EMA criteria for that they specify for surrogate endpoints. So we're trying to do that across all the trials by measuring the same things in parallel. So these are some of the protein factors, but we're also measuring reactive oxygen species, NAD indicators of mTOR activity, bioactive small molecules, including sphingolipids that look important because they can reflect remyelination. We're planning to include some microbiome-related factors that get into the blood and seem to track with fundamental aging processes. Also, non-coding nucleotides, especially some microRNAs and mitochondrial DNA, which looks especially important. And we're also measuring parameters across the trials in urine, in saliva, in CSF where we can get it, in superior nasal swabs, and in biopsies. So the Senolytic clinical trials for cancer treatment or their complications that are underway or planned, including one in hematopoietic stem cell transplant survivors, that trial is complete now and the data are under analysis, and that's looking mainly at frailty in these people. They develop a frailty-like syndrome if they don't die of graft versus host disease or chronic rejection or return of their cancer. Many of them will develop a frailty syndrome within 3 to 5 years. There's a similar trial, it takes longer to develop though, in patients who get frailty after breast cancer treatment. That trial is being run out of UCLA. Sensitive trial I'll talk about in a moment, run by St. Jude, glioblastoma trial, and there are planned trials for melanoma, myeloma, and triple-negative breast cancer. So childhood cancer survivors, they're followed at St. Jude for life. If these people have had chemotherapy or radiation for their cancers before the age of 10. And some of them, they've got about 800 patients like this, develop an accelerated aging-like state when they're between 30 and 45, and they're dying of Alzheimer's disease at age 40 and that kind of thing. They're frail, they look like they're 70, they've got diabetes, they get second unrelated cancers, etc. Now, not all people get this. It's the ones who have senescent cell burden detected in their skin that seem to be predisposed to getting this syndrome. So in this trial, which is about halfway through now, that's being conducted at St. Jude, it's a comparison between dasatinib and quercetin or fisetin with placebo. They're being looked at with respect to frailty parameters. With cancers, the National Cancer Institute proposed a 1-2 punch approach back in 2021 in a couple of position papers, where they proposed that there should be trials somewhere along the line of chemotherapy and radiation, followed by senolytics, followed by chemotherapy and radiation, followed by senolytics in cycles. And some early evidence for that helped back this up was Terry Burns' work in glioblastoma using BCL-XL inhibitors. So one of the trials that's underway is for stage 4 and 5. It's for latent glioblastoma after temozolomide and radiation treatment. And these are patients who have a myocisterns in with a catheter in the tumor and then a bubble on the skull so that fluid can be analyzed from the tumor bed. And this is an adaptive design, which is an entirely new kind of design where the FDA is allowing us to select the drugs to give next based on what we find in the CSF in these patients who are in the transient remission that these people tend to get. They're almost all dead at 18 months, but some of them will get a transient remission. And it's to try to prevent recurrence. We've developed a new class of drugs we call senesensitizers. I mentioned that 30 to 70% of senescent cells only are sensitive to the classic senolytics. It turns out that you can induce them to become sensitive to senolytics using this new group of drugs, which we're still working on. And this shows what happens with triple-negative breast cancer. So it looks like alternating chemo and radiation, then senolytics, then senesensitizers, then senolytics may work. It may also be the case that giving senesensitizers before chemo and radiation has a beneficial effect. So in conclusion, persisting senescent cells, we've heard before that it's the persisting cells that are dangerous, may give rise to and accelerate most, if not all, cancers, while rapid induction of senescence and rapid clearance of senescence may reduce cancers, for example, in elephants or right whales. These are huge animals. They develop from one sperm and one egg. They live much longer than we do, and they rarely die of cancers. So it's of interest that they do develop senescence very, very quickly. What we don't understand is how their immune system gets rid of those senescent cells once they've occurred quickly. So that'll be interesting to study. It appears that targeting senescent cells may prevent or delay cancer development. A situation where this is being tested is in Parkinson's, which is closely associated with melanoma, and vice versa. Adult Down syndrome with acute leukemia, so we're beginning a trial soon for adult Down syndrome, and persons living with HIV. Targeting senescent cells may increase or with either senolytics or senomorphics, may increase the doses of radiation and cytotoxins that can be given. This has been shown with methotrexate now. Some drugs, though, have specific side effects, like you would not want to give zoledronate before giving focal radiation because that'll cause osteonecrosis. Since therapy-induced senescent cells can continue to mutate and escape senescence, as we heard before, checkpoint antibodies, vaccines may help to accelerate cancer cell elimination by the immune system. 1, 2, 3, 4 punch approach might be even more effective. Targeting senescent cells may reduce cancer acute and chronic treatment complications, at least in preclinical models. And targeting senescent cells may decrease cancer treatment long-term adverse effects, like in the childhood cancer survivors. But we don't know until these trials are done. I spend all my time telling people not to take these drugs, not to prescribe them, not to buy them from Amazon. They're dangerous until we've done clinical trials that are regulatory agency approved and those clinical trials are replicated. Anything that sounds too good to be true is. I started off a few years ago thinking these drugs would have a 0.001% chance of making it through clinical trials. Now I give it maybe a 30 to 35% chance, but there's a high chance I think that they'll fail. Some of the early trials are looking promising. 1 or 2 others will fail. Again, we've got to have patience about this. I think it's important to do multiple trials in parallel rather than in series, so we can test this hypothesis quickly. And some other brief points, and I'll stop in a minute. There's the argument of good versus bad. So senescence can prevent cancer development and spread. And if you interfere, as you all know, with P16 or P21, that'll increase cancers. As I mentioned, long-lived large mammals that have decreased cancers have early development of senescent cells. And interestingly, very elderly individuals tend to have decreased aggressiveness as cancers. As a geriatrician, we see this all the time. People die of a stroke or something, you find 5 or 10 different cancers in that patient at autopsy that were subclinical. Or if they do get a cancer, their time to get metastases can be slow and the metastases can be slowly developing. And that could be because they've got bad soil for the cancer seed to try to survive on. So we need to think of that. At cancer sites, though, senescence can be bad. So therapy-induced senescent cells resist apoptosis and immune clearance. They can continue to mutate. They can come back as therapy-resistant cancer cells. Senescent cells in and near cancers can add to immune evasion because they can poison the immune system. IL-6, for example, will impair macrophage diapedesis. Mitochondrial DNA greatly impairs or alters dendritic cell function and therefore will affect the adaptive immune system. The dendritic cells are in the innate immune system, but they enable the adaptive immune system. They can cause hypovascularity, which can reduce immune access to cancers, and they can cause a fibrous capsule to develop around cancers that can prevent the immune system from getting at cancers. Systemically, senescence can be bad, as in the transplant studies. Senescent cells can interfere with innate and adaptive immune function and reduce cancer clearance. And so we have to think of new regimens that are going to be personalized. For this, we'll need the right biomarkers. We'll have to have the right drugs, and we'll probably have to sequence these things with known interventions for various cancers. Rather than giving just one kind of agent, we might have to think of personalized intervention strategies based on biomarkers. So I'll conclude by thanking a lot of people. Tamara Ciccone and Izu were instrumental in helping to develop Senolytics, and many other people, including people in this room, have helped with all of this, and we're grateful to our funding agencies. Thanks.
Thank you so much, Dr. Kirkland, for this excellent and very comprehensive talk. I'm sure there are some questions. Maybe I will start with a brief one. You mentioned a number of possible compounds with effects on aging. And can you please comment on GLP-1 receptor agonists that have been shown to have a potential to slow down cellular senescence? What is their role?
I don't know. I think they could be a double-edged sword. So we just don't know yet. They are showing some promise in the acute and semi-acute period in improving cognitive function and all kinds of other things. But there's some downsides to them too. They, the clinical downsides are that they can affect the brain quite a bit and addictive behaviors, including work addiction, exercise addiction, and good addictions, as well as bad addictions. But one of the worries with them is if there's any residual glucose in someone, then what GLP-1 agonists will do, because they're produced by glucagon-like peptide 1 is produced by the L cells in the duodenum, it'll greatly activate insulin production. So some of these people, if they've got any residual glucose, if they've got peripheral insulin resistance and any residual glucose around, they can have sky-high insulin levels, high enough to act through IGF-1 and growth, the IGF-1 growth hormone pathway. So we've rushed into going into these drugs. We don't know what the long-term effects are. They could be good, they could be bad. We certainly know there are some acute bad effects. There's this yo-yo phenomenon. People can lose weight so quickly they can have their retina detach. But I think it remains to be seen. I think the SGLT2 agonists, there's a better track record, and for both, and also SGLT1 agonists, because they're in every kind of cell, not just the cells that in the urinary tract. Every kind of cell has to have sodium-glucose exchange. And it turns out in some cell types they're senolytic and in others they're senomorphic. So they could be, and they've got a longer history.
Thank you so much. Questions?
Well, I might continue with another question while the other thing are thinking to make the question. So based on the difference in responding to the senescent therapy, senescence, and also the target population of patients, that they can respond in a different way based on the dosage of the drug, what do you envisage is the best clinical trials that you can make in a cancer patient?
The clinical trials we're doing are for very serious conditions for which there are no good current treatments, or where there's a need to add something, because we're worried about risk-benefit ratios. We're not interested in doing clinical trials in completely healthy individuals. If they don't have senescent cells, why would you give a senolytic? And why would you take the risk of having a side effect? I mean, even metformin is tricky as far as primary trials go, because people can get ketoacidosis. They climb one of these mountains and get dehydrated. A young person can get ketoacidosis, and that means going into the ICU and only a 40% chance of coming out. It's 1 in 50,000. But why would you do a primary prevention trial? That's why the TAME trial is secondary prevention. That's reasonable. But I think with Senolytics, there are some secondary prevention trials beginning, like with Down syndrome, like with relatives of type 2 diabetes, where we know that they're going to get something. So there's a type 2 diabetes trial beginning in Sweden. But I think most other situations, it's Alzheimer's disease, where a couple of the trials have shown early promise, including the trial that just came out from Harvard, and an earlier trial that came out from Children's, and one that's underway at Mayo. I mentioned there's an osteoporosis trial that Sandeep Khosla did in age-related osteoporosis that had very nice outcomes, but it's only an early phase 2 trial. There are a few others coming along like that, but it's very early days at this point.
Okay, thank you.
Thanks. Thank you.
Thank you very much.
So I cannot thank the organizer because I'm a non-organizer, but I can thank you guys for being here. So many.
It's okay. No, he's going to introduce himself. It's good. It's better, actually. Go ahead.
Sorry. Yes, it was just a thank.
No, basically, Professor, however, I think everybody knows Professor Alimonti. I know him since 25 years here, so that's okay. I know this fact. So he's a director of the Institute of Oncology Research and the full professor at the University of Svizzera Italiana, ETH Zurich. Andrea owns a lot of experience in basic research, but also applied research, from cancer biology to senescence, tumor microenvironment, and also other different clinical indications, but specifically in very important in prostate cancer. So, thank you.
Thank you, Gianni. Thank you for the introduction. So today I'm going to discuss about the role played cellular senescence in prostate cancer. Why prostate cancer? So it's a simple fact, right? 1 in 9 men in this audience will develop prostate cancer. So you, particularly male, you will have this problem, right? So, and if you look at the total number of prostate cancer around the world, this is increasing, so it's really a huge problem. So the key activity of my lab is try to develop novel and better therapeutics in order to delay the onset of a lethal type of prostate cancer, which is called castration-resistant prostate cancer. In other words, a form of cancer that doesn't respond to standard anti-androgen therapy. So if you don't respond to this therapy, there is essentially very little to do, and so this patient unfortunately dies. So we really want to try to delay this onset of cancer, or either reverse castration-resistant prostate cancer in sensitive. So how do we do that? So I have a big team now, 25 people plus 2 or 3 people into the clinics. And I spend a lot of time by using however transgenic mouse models in order to understand 2 things. First, to understand the biology and the complexity of prostate cancer initiation and progression. And second, try to use this transgenic mouse model in order to run preclinical trials, right? And many years ago, we were lucky enough to discover essentially that cellular senescence is a key process involved in prostate cancer initiation. Indeed, we show that the senescence prostate tumor cell can oppose tumorigenesis. However, later on, we also discovered that at late stage senescent tumor cell can have an opposite effect. So instead to stop prostate cancer, they propel prostate cancer and drive metastatization. And the story starts with when I was a postdoc in the lab of Pierpaolo Pandolfi, that everyone should remember for his contribution to the field of cellular senescence, where we were inactivating this important tumor suppressor gene that is called PTEN. With our surprise, you know that PTEN controls PI3K pathway and mTOR, and normally if you touch PI3K, the cells start to grow, right? On the contrary, we observe a paradoxical effect. In other words, we observed that the complete inactivation of PTEN was setting in motion this senescence response. This was driven by the fact that TOR get activated, P53 get translated, and senescence happen. So people already mentioned what is cellular senescence. So it's a stable cell growth arrest that happens in a normal cell, in our tissue during aging, but also in cancer cell, for instance, for effect of PTEN inhibition. The cell have a different characteristic. They are arrested irreversibly, so it means that it's very difficult to let them regrow in vitro, but there are some conditions in vivo in which we observe a bypass, but essentially they're much more harder to let re-enter into the cell cycle. Plus, these cells are been defined zombie cells, so cells which are suspended in between life and death, so they are actively producing factor in the surrounding microenvironment, which influence the tumor immune response, right? And our work has been dedicated for many years on this intercross between cellular senescence and the tumor immune response, right? And so this is a cartoon that has been shown already by Jesus G. I think it's a very nice cartoon. So this represents the effect of a chemotherapy or radiotherapy or an anti-androgen therapy in a tumor of a patient that is treated in the hospital. So there is an accumulation of senescent cell. This senescent cell, through the SASP, which is this capability of cell to secrete factor, can recruit different immune components. And we discovered that, of course, cancer is not stupid. It starts to recruit a lot of myeloid array suppressor cell, in particular this population of cell, which we have discovered, they're called polymorphonuclear myeloid array suppressor cell. This cell essentially prevent the elimination of senescent tumor cell by the tumor by blocking the function of T cell and NK cell. And for effect of the fact that these senescent cells are not removed, essentially there is a chronic inflammation, an inflammatory storm that propel the prostate cancer. So we have done two things for more than 10 years. We have focused together with the immuno team of my lab in trying to block this polymorphonuclear myeloid array suppressor cell, which we demonstrate can, for instance, hinder the therapeutic efficacy of docetaxel. Docetaxel is another type of drug which is used in prostate cancer in hormone refractory, also hormone sensitive patients, but in general in late stage metastatic prostate cancer. So this drug works by inducing senescence, but also by recruiting myeloid array suppressor cell, which block the clearance of this senescent cell by T and NK cell. And by blocking this population of cells, we were able to reestablish senescent surveillance, so the capability of T cell and NK cell to kill essentially not only senescent tumor cell, but also normal prostate tumor cell. And why this is important? Because after 10 years, by asking around to people like Gianni, Mani, to develop clinical trial for this concept, we were able to publish this paper on Nature together with Johan de Bono and Silke Gillesen. So this has been really a crusade that demonstrates essentially two things, that if you treat a patient, these are clinical study, a phase 1-2 clinical study, with enzalutamide. Enzalutamide is an anti-androgen therapy. There is a senescence also in vivo, in the longitudinal slide section, we could see that. And more importantly, this is associated to a SASP, which recruit this polymorphonuclear myeloid array suppressor cell. So now if we use the blocker of polymorphonuclear myeloid array suppressor cell, which is a drug that has been developed by AstraZeneca, unfortunately we didn't patent it many years ago, which is an inhibitor of a 6ER2, a receptor present in MDSC, we observe that senescence disappear and that there is also response in this patients that are heavily pretreated with a lot of different line of therapy, right? This suggests essentially that if we can impact on the senescence secretory phenotype of in cancer, we could result in partial response, not complete response, right? And so now together with Silke Gillesen, which is there, and I have to thank, and Johan de Bono, we are launching a second study, which is called ASPIRE study, where we are going to work on a second generation better inhibitor for this myeloid derived suppressor cell in order to see whether there are more response and patients that can beneficially be treated with this compound. So, but what's happened if we cannot use this immunomodulator, right? So we have another way to eliminate senescent cell, not through the immune system, but through the use of senolytics, right? This has been mentioned. So this compound, which essentially eliminates senescent tumor cell. And so in this paper that we published now in 2021, we, I think, demonstrate something which was not demonstrated carefully before. So in a transgenic mouse model, we also show that use of ABT, which is called navitoclax, right, can actually eliminate senescent tumor cells, specifically in this type of tumor, which is called PTEN-null, TIMP-1-null tumors. And this can impact on the survival of these mice because the mice do not develop metastasis. So senolytic actually should be used, in our opinion, as a way to prevent metastatic spread, so in an adjuvant setting. And there are very few studies that are testing this hypothesis because they're very expensive studies, right, if you think about it. So, however, for many, many years, myself, Scott Lowe, also Clemens, has also worked on the positive anti-tumor effect of senescence in cancer, right? So, because there were evidence, essentially, that, as I told you, that senescence at the early stage of tumorigenesis can block tumor development, right? But what about late stage of tumorigenesis? Can we find a compound that induces a good senescence response, which is not associated to a detrimental SASP? This has been done in the lab for many, many years. We have discovered different compounds, but recently we show in a cancer cell paper that essentially retinoids seem to have this capability to induce a very strong senescence response specifically in prostate cancer for reasons that you can read in the paper, because the metabolism of this tumor cell rely essentially on factors which can be activated by retinoid. But more important, this senescence response, which works very efficiently, is depending by P21 and is associated to a good SASP. In other words, to a SASP that instead to push normal cell to proliferate to become metastatic, it renders them less capable to proliferate, and more importantly, it activates the immune response, in particular NK cell. It's been said before by Jesus Gil that NK cell play a very important role, right? But more importantly, I think if you start to combine docetaxel with retinoids, you see that the SASP of docetaxel, which is really a bad SASP because it propels metastasis in different mouse models that we have used, it's all of a sudden reprogrammed by the presence of retinoids with many negative factors, protumorigenic factors, which are listed here. I don't know how to point, but essentially here. And that goes down in adapalene and also combo-treated. Adapalene is a rare agonist. And there are different factors which are immunoactivator and are activator of the recruitment of NK cell, which are upregulated in this tumor. So indeed, if we look, we use adapalene in this mouse model. This is an allograft mouse model characterized by loss of P10 and P53. We see the presence of senescence. This is enhanced by the co-treatment with docetaxel, and this depends by the presence of NK cells. So if we eliminate NK cells, there is still a partial response from this retinoid, but there is a much better response if NK cells are there. And so Manuel Colucci, which is in the audience, had a very good idea. It started to look at the migration capability of NK cells which are extracted from the blood of patient or healthy subject. And he observed that if you co-culture this with the tumor cell treated with the combination, essentially this NK cell migrate much more versus the tumor. So they have an enhanced capability to migrate. However, they also have another incredible capability. So the tumor cell treated with the combination, so they become senescent, so they upregulate different activating ligand of receptor which are present on NK cell. So when the NK cells see this ligand, they start to be activated to degranulate. So we thought that we could use this in order to implement allogenic infusion of NK cell in mice treated with the combo. And this is an experiment, essentially, they show in vivo that if you treat the mice with docetaxel and you infuse NK cell, there are a lot of effort in developing novel NK CAR, for instance. Nothing happened. In other words, the tumor doesn't disappear. However, if you do this in presence of adapalene or in combo treated cells, so docetaxel plus essentially adapalene, all of a sudden the NK cell become capable to kill the tumor. So we think this could be a way to enhance the efficacy of NK CAR T-cell therapeutics in the future. However, today I also want to show you new data about another approach that we have discovered in the lab, which is another interesting approach in our opinion to eliminate essentially senescent tumor cell. The story starts when we discover in the mouse model that senescent tumor cells, which are sorted out from the tumor, express a lot of Nectin-2. You maybe are familiar with Nectin-4 because it's been the first target of a successful antibody drug conjugated which work very well in bladder cancer. But we found a lot of Nectin-2. And so we look at the primary versus metastatic prostate cancer. We found that essentially Nectin-2 is the most upregulated immunocheckpoint target between a list of different immunocheckpoints, including PD-L1, PD-L2, etc., etc. And more importantly, we look at the receptor of this Nectin-2 on NK cell and T cell. In other words, Nectin-2 is expressed by the tumor, and the NK cell have a receptor which is called essentially TIGIT, PURG. So when there is this binding, NK cell do not work any longer. The same is also for the T cell, okay? And if we look at what happened in primary and metastatic data set of patients affected by prostate cancer, you see that there is a correlation between Nectin-2 and the activity of NK cell and T cell in this tumor biopsy. So more Nectin-2, less activity of T cell and NK cell. But very, very importantly, if we take longitudinal sample from patients, these are patients from Padua, in which we essentially have primary tumor and metastatic tumor from the same patient, right? You look at the presence of Nectin-2, Nectin-2 increase with the tumor progression, so there is much more Nectin in metastasis. And the increase of Nectin-2, it's increased even further with exposure to therapy, which are either enzalutamide or docetaxel. So there is a further increase in patients treated with this compound. If we take cell line, you try to drive senescence by using enzalutamide, docetaxel, or palbociclib, the senescence induction is accompanied to an increase in Nectin-2. So there is a strong upregulation of Nectin-2 in senescent cell. This is also seen in terms of antigen density, which is really important. How many molecules are present on the surface of senescent tumor cell, right? So you sort out senescent tumor cell by FDG staining. You look at the presence of this antigen density. So we did a simple experiment. So we treat the tumor cell with the different senescence inducer. I told you that antigen density was increased. We co-cultured them with NK cell or T cell, and we found that only when the NK cell, the T cell, see the senescent tumor cell, there is an inhibition of NK, thereby validating the important relevance of this immunocheckpoint, right? So in our opinion, senescent tumor cell, by overexpressing Nectin-2 can suppress the function of T and NK cell in prostate cancer. Well, the story became more exciting when all of a sudden we wanted to identify antibody that could target specifically Nectin-2, right? And we use a phage display library in which we screen for the first time normal cell or tumor cell treated or not with senescence induction. And we found that essentially there were like two antibodies. So these are SCFV, FC, so it's almost an entire antibody. So I would refer as an antibody. One that binds to a specific region of nectin, another one to another region of nectin, that essentially can have different results, right, in terms of binding. And so there is this antibody, it's called RB722, which is also a commercial one, which bind to equally to normal cell, up, you see the normal cell, and also the cancer cell. However, there is another antibody which is cancer specific, it's called RB725, that it doesn't bind to normal cell, but it binds only to cancer cell, and in particular to senescent tumor cell, where the antigen nectin-2 is particularly exposed. This is true also in human prostate biopsy. So this is a slide done from Andrea Cavalli that demonstrates that there is a differential binding of the two antibody. And so how can it be that an antibody which binds to different regions can recognize also nectin-2 in normal cell, and the other antibody doesn't do it? So we come up with this model. In other words, this is what's happening in a normal cell with these two antibodies. So one antibody binds to this Ig1 domain. However, antibody binds to the Ig2 domain, which is closed by the fact that in physiological condition, nectin-2 doesn't work as an immunocheckpoint, but work by binding other nectin-2 and to creating like this gap junction, which tied the cell, right, together. You can see that there are different models, but anyhow, our antibody doesn't bind. However, in cancer cell, which have this characteristic hallmark, so cancer cell lose the capability to essentially to adhere between one cell to the other because they start to migrate. All of a sudden, there is an exposure of all of this Ig2 domain. So Ig1 can still bind the other antibody, but we now see a specific binding of RB725 in nectin-2. So only in cancer cell, we are able to expose this antigen, and particularly in senescent tumor cell, because you know better than me, the senescent tumor cell are even more pro-migratory and less adherent than normal tumor cell because they produce a lot of metalloproteinase, right, through the SASP. So this antigen is massively exposed essentially in senescent tumor cell. If you do an experiment in which you take normal cell and you essentially create a nectin-2 which cannot form homo or heterodimer because these are mutated, all of a sudden now our antibody can also recognize essentially a normal cell, thereby demonstrating that if you lose this interaction between cell to cell, the antigen is exposed and the antibody is able to bind, right? So now why this story is interesting, because we wanted to now create CAR T cell against this new target, right? Because we didn't have money. Johnny still didn't give us money to create an antibody drug conjugated that we would like to do. And so we look at the prostate cancer together with Johan de Bono, and we saw that, you know that in prostate cancer, there are a lot of people that are developing PSMA CAR T cell. For instance, CAR-June is working a lot on this PSMA and CAR T. However, PSMA, it's really not a good target because sometimes it's heterogeneous, right? On the contrary, we found that essentially there are a lot of cells in this complex tumor microenvironment that are nectin-2 positive, PSMA negative, and also a double population of cells. So if you target nectin-2, I think it's better that if you target essentially anti-PSMA. And so we did that. So we created different CAR T. We created an anti-nectin CAR T with the support of Dennis Migliorini and CAR-June, and a double anti-PSMA anti-nectin CAR T. And what we found essentially that this worked very efficiently in vivo. You can see like that there is a really nice decay of the tumor when we use the anti-nectin on the double CAR T in combination with enzalutamide, which essentially increase senescence and led an exposure of this antigen. And this impact on the survival of the mice, right? In this mouse model, which is an LNCAP mouse model, which is a relevant mouse model for people who work in the field of prostate cancer. And so what happened to senescence, right? If you look at now at the senescence by looking at the immunofluorescence staining, you see essentially that the reason why the anti-PSMA CAR-T, in our opinion, do not work very efficiently, this is also true in patients, is because senescence tumor cells are still there, right? And I think Carl has also this data in vivo, in human. So because they don't remove essentially senescent tumor cell. Why? Because they express nectin, and nectin block the function of T cell, including the function of CAR-T. So you are creating an anti-PSMA CAR-T, which go against a target, but the cell that is PSMA positive is also nectin positive. So it's blocked. So, on the contrary, if you use the anti-nectin-2 CAR or the combo CAR, you eliminate all the senescent cell and essentially have a much better response. Okay, now, for reason we still don't know, when you use docetaxel instead of enzalutamide, the anti-nectin CAR work a little bit less, but the combo CAR still work very, very efficiently. So we believe that really the combo CAR, so the anti-nectin, anti-PSMA, could be a way to implement standard of therapy for CAR T cell, which is now try to use this anti-PSMA CAR T in the clinic. However, very importantly, this CAR T cell work better than other type of CAR T cell which are under development. For instance, this TGF-beta dominant negative CAR T cell, whose results have been published in Nature Medicine few years ago. The reason why this is happening is because it's associated to an increased toxicity, even in mouse. We don't see a toxic effect of our combo CAR T cell. So I hope that I convinced that there are pros and cons of senescence in cancer and cancer therapy. Senescent tumor cell can block tumorigenesis in early stage tumor. Failure to eliminate senescence in late stage tumor can contribute to tumor progression. That there are still ways to activate a positive anti-cancer senescence program by using, for instance, RAR agonists, and that we can generate effective anti-nectin-2 senolytic CAR T cell, which have this advantage that can recognize specifically a tumor cell without altering the essentially normal cell. And with this, I want to thank everyone. So it took a village to do this project. So I mean, my lab is big. I didn't have space to thank everyone. I also want to thank the collaborators, so Saul Prisman and Carl June and Dennis Migliorini for the implementation of CAR T cell, Johan De Bono and Silke Gillesen for the clinical implementation of the 6ER2 inhibitors and the new trial that we have already supported, that we are now continue to try to push into the clinic, and of course Andrea Cavalli for the antibody model generation. And with this, I'm happy to take your question.
Okay. Open to questions? Yeah, please.
Andrea, very nice talk. Question is, why should we think that this nectin-2 is important only for prostate cancer cells? And do you have any way, reason to think that maybe we can apply the same concept to other?
Yeah, no, exactly. So, for instance, in ovarian cancer, nectin-2 has been well known as a way to block a very well-known immunocheckpoint inhibitor. Immunocheckpoint, sorry. Ovarian cancer, for instance, is another type of tumors in which nectin is expressed, and we don't work in ovarian cancer, but I mean, this could be useful also there. And breast cancer is another tumor.
Yeah. Yes.
So far, we focus on the target identification and validation in prostate cancer.
Thank you, Jesus.
Hi, Andrea. Very nice talk. So you saw at the very beginning that there are like multiple immune checkpoint ligands that are expressed on prostate cancer. Do you know, for example, whether the Nectin-2 is expressed in the same cells that are expressing B7H3, or is a different subpopulation? Can you tell anything about heterogeneity?
Yeah, so we did this analysis with the PSMA, where we saw that there are double positive population or single positive population. We also done that with B7H3, and we see that there are different type of population. We also have a paper on that with Jon about B7. But more importantly, Nectin-2 is the only immunocheckpoint, right, that immunocheckpoint that increase in senescent tumor cell in prostate cancer, right? So there is and I think there is another one which I forgot now. Silvia is there, and yeah. So we focus our attention on this because it gets very elevated.
Thank you. There is time for another couple of questions. I will continue while you're... I want to take one from the previous question, Andrea, so talking about the retinoic acid and the TIP1. Actually, those kind of molecules are super important for the liver. So specifically in differentiation of hepatic stellate cells. I don't know if you have seen, which is fibrosis. So have you seen, which is called, cancer. So have you seen if there is any correlation in the liver as well?
No, we didn't look into that, but we know that RAR can, in our case, can induce a cellular senescence. It's also a program in which the cell cannot re-enter, right, in the cell cycle. But we didn't check in what is the effect of this in normal tissue.
Cancer, liver cancer. So.
Oh, liver cancer, no. We didn't.
Okay, next question.
Andrea, great talk. Can you disentangle for us a little more? What is the composition of advanced prostate cancer exposed to anti-androgens, exposed to taxanes? There are senescent cells and there are non-senescent cells. What is the actual target of the double CAR T attack? Is that just the senescent population? And is the rest of the tumor dependent on the replenishment of senescent cells in their whole composition? How do we need to understand this?
So if we look at longitudinal tumor sample, we see that enzalutamide or docetaxel essentially increase all these senescent signature like Seno, whatever, etc., etc. And of course, we see the Nectin-2 get elevated with the exposure, right? So we believe that essentially Nectin is a key immunocheckpoint that block T and NK cell to eliminate efficiently senescent cell, right? So if we target essentially all this senescent cell expressing Nectin-2, we can reactivate immunoclearance mediated by any type of drugs, like it could be immunocheckpoint, CAR T, etc., etc.
So if you would not treat with a taxane, docetaxel, or enzalutamide, is the CAR still effective, or does it lose its efficacy?
No. It's less effective, but it's still working because Nectin is still present also in tumor cell despite the senescence.
Okay.
So, yeah.
Thank you. There is time for another question. Yeah, please.
Thank you, Andrea. Great work. I also have a question that is connecting the two stories. The fact that you identified this myelosuppressive population, and then the fact that with the CAR, you seem to clear the cell. So it looks like the CAR, in terms of localization in the tumor, they're able to go, and they're not blocked by the myelosuppressive cells. So did you think of combining the two strategies? Because I was suspecting, being a solid tumors, that the immune suppressive microenvironment would prevail on the CAR while you see an activity.
Yeah, no, that's a very good question. So thank you. So our idea is in the future to try to combine the 6ER2 antagonist with this type of CAR T in order to further potentiate the efficacy, right? But of course, we did all this experiment now in immunodeficient mice, right? Because these are human prostate tumor cell and human CAR T, right? So we also have now ongoing a study where we have to switch from the human to the murine CAR T. And I will tell you whether I see an inhibition of myeloid cell. But we don't expect that because essentially CAR T should be really very potent, right, in eradicating tumors. despite the presence of myeloid cell. But this experiment is ongoing. We have to recreate a different type of murine CAR-T. It's, as I told you, to identify now this tumor-specific CAR-T in the mouse. It's other 2 years of work, right? So we are trying to do this with a classical sequence for the mouse and to see if myeloid can block it.
Okay, thank you. Andrea, one quick from me before closing. In terms of clinical development and what actually is an effect therapy for those prostate cancer patients, do you see a long-term treating with some senolytics or other kind of senotherapeutics, or it's just a short-term and then you have this adaptation?
No, I think that I believe that it depends, Gianni. So I don't understand why drug industry still doesn't believe in the field of senescence, right? Because I think we were not able as scientists to communicate efficiently to the industry the potential of this. So it's very much depend from the type of senolytics or intervention you could do. But there are like also natural compounds, right? Or plant extract, which could be used to essentially to eliminate senescent cell, right? And this should be used in an adjuvant setting, in my opinion, and try to run long study in order to see whether there is an effect, for instance, in aging-related symptom, in cancer. So this kind of trial need to be supported by industry. We cannot do that. So particularly us, to do this phase 1/2 clinical study, we have to fund 3 million. And the money don't go to the lab, they go to the clinic, and people in the lab start to think I'm crazy if I continue to diverge all this money to the clinics, right? So either we have the support of industry, new startup, and the people like you that believe it, or we will never see the clinical application of Senotherapeutics.
Okay, thank you very much for the pitching. So yeah, next.
So thank you, Andrea. We move now perfectly on time to our next speaker, who is Marco Demaria, who will refer about heterogeneity of cellular senescence from mechanisms to interventions. Marco is a professor of cellular aging and the medical faculty of the University of Groningen and the group leader of Laboratory of Cellular Senescence and Age-Related Pathologies at the European Research Institute for the Biology of Aging in the Netherlands. Thank you, Marco.
Thank you, Alessandro. Thank you, of course, the organizers, Fabrizio, Andrea, for... I just realized Andrea is very tall because microphone like this, or I'm very short. So thank you, the organizer, for putting together this wonderful meeting. And if you are not tired about senescence, 16-19 September, we are organizing the 10th International Cell Senescence Association Conference in Rome. Registrations are open, so please, if you're interested, join us in September, and it will be also the first day a session dedicated to Senotherapeutics. So it's going to be another nice forum to discuss, indeed, maybe some advances in the field. So as a disclaimer, I'm not going to actually talk about much about clinical trials. I'll go a little bit back about going from biology of senescence, maybe some new biology of senescence, to also maybe tell how we can exploit some new biology to develop therapeutics against senescent cells. We have heard, of course, a lot about the SASP. I just wanted to point one thing, that while the original discovery of the SASP, we go back almost 20 years ago, even longer, was based on these pro-inflammatory proteins that are indeed part of the secretome of senescent cells, mainly mediated by NF-κB, but then additionally by other transcription factors. We have also realized in the last decade or so that there are many, many other molecules that go beyond proteins that are secreted by senescent cells. We have lipids, we have extracellular vesicles with various content, and we have also nucleotides. And it is quite important for also what I will tell later. But also another important point is that we have heard a lot about the paracrine effect of the SASP. So the fact that the SASP can highly influence the behavior of the neighboring cells. However, there is also the autocrine component of the SASP. So certain SASP factors can potentiate other senescence-associated features. And again, this is a point that I will go back in a few slides. So we have heard about the detrimental function of senescence, so I'm not going to spend too much time on just summarizing again everything here, and the fact that we want to exploit the detrimental function of senescence to develop potential pro-health therapeutics. But also, we have heard about, especially in the cancer context, about the beneficial function of the SASP. And you see here on the top some of the various functions that beneficially the senescent cells can regulate via the SASP in the cancer environment. We have heard much less about potential beneficial function of senescent cells in tissue regeneration and also in the formation of certain structures, even at the embryonic level, which you can see here. One example of these actually was done by us now several years ago by using this reporter mouse that can selectively eliminate p16 positive cells, but it can also label and track the presence of p16 positive cells in mice. We have demonstrated that senescent p16 positive cells, at least, are induced very early upon injury. In this case, it's a skin wound healing, so you simply punch the dorsal back of the mouse, and then you simply follow. Normally, mice are very good at repairing this type of lesions, so in a few days they will be normal. But during the time of repair, you clearly see an induction of senescence that here indeed can be reported because of the luminescent component of this transgene that we have in the mouse. And then the cells actually are cleared very efficiently, so they disappear very efficiently. However, if you force the elimination of these cells by using, in this case, a prodrug, ganciclovir, that can activate a suicide gene, specifically in this transgenic animal, so this would not work in a wild-type environment, you actually see that the ability to close the wound is somehow delayed. So mice take longer to now close the wound if they don't have this induction of p16 positive cells. Or, even better, they might induce the p16 positive cells, but the cells are eliminated immediately after, and this is causing a problem to regulate optimal tissue repair. Later on, we also tried the same trick now by using maybe some of the most common senolytics at the moment, which is either ABT drug, not 263, not navitoclax, but 737. But the concept is the same, targeting BCL2 anti-apoptotic proteins, or the cocktail that Jim Kirkland and his team developed, dasatinib plus quercetin. And we have tried to apply the same these drugs in the same model of wound healing, and we have seen that actually we achieve a similar problem that we have when we eliminate genetically the p16 positive cells. So we see that indeed we suppress the induction of these p16 positive cells if we apply the compounds or feed the mice with the compounds, and also we have a problem with wound healing. So now also we have a sort of like delayed kinetic of wound closure if we are eliminating these p16 positive cells, or at least there is a correlation between lower level of p16 positive cells and delayed wound healing. So here, the idea is that, of course, we have heard about why we have detrimental cells. I mean, why senescent cells might become detrimental is mainly because they seem to accumulate, right? While, as I just mentioned, beneficial senescent cells are very different because they're kind of transiently present in tissues. They come and go very quickly. The kinetic of removal are very fast. And of course, at the moment, we have talked about senotherapeutics, and I will go back to the idea of senotherapeutics, so elimination or targeting certain detriments of senescent cells. But on the other side, when we talk about beneficial senescent cells, we might actually think that senescence might be a superior cell fate than, for example, cell death, because I showed you the example before that if I force a senescent cell to become apoptotic in that case, so dying, now I have a problem. So perhaps we can even exploit the other way around, contexts where you have a massive amount of cell death and trying to switch the fate towards senescence and see whether you can generate actually artificially these pro-regenerative senescent cells. And this is something that we have worked in the past few years, and in particular this is work that was pioneered by Jamil, a postdoc in the lab. So Jamil was really interested to understand a particular type of tissue injury, which is ischemia reperfusion in the kidneys. So ischemia reperfusion is an event that can happen unfortunately, quite regularly in the human population, which is due to a temporary blockage of blood flow to the kidneys. So I say temporary because only it happens for a few minutes, but this is enough to, of course, block the flow of also oxygen in the tissue. And then when the blockage is removed, for whatever reason, it can be surgically or it can be natural, there is a massive now flow of blood and oxygen, which create a lot of reactive oxygen species. And reactive oxygen species, especially at high level in a very small area, can create massive regulated necrosis. So many cells would actually commit to necrosis via different pathways. If we are confused about senescence and the type of senescence, we can become even more confused when we think about the type of cell death that you can have in your body, in particular necrosis. So there are many, many ways that cells can enter this type of death, this necrotic type of death. But in particular, it seems that one important one is also relevant for us is spartanotose, which is a type of regulated necrosis which is mediated by PARP1, which is an important regulator of DNA repair, but it also has a massive impact on metabolism cells. And eventually what happens is that when you have this massive necrosis, somehow you recover from the acute injury, but in the long term, your tissue will never repair completely, and it will start to degenerate. And this will create essentially chronic kidney failure. So then Jamil was really interested to understand whether certain cells that might enter this programmed necrosis because of high-level oxidative stress could be rescued and potentially pushed into senescence. So he did a screening using different inhibitors of various types of regulated necrosis. And what he found was indeed that if he exposed a normal fibroblast to lethal doses of hydrogen peroxide, but together with it, he also blocks the activity of PARP by using this compound, which is very well known in the cancer field, which is PGA34. It could actually suppress the cell death. So the cells remain viable. And this was the only type of death that could inhibitor that could rescue, indeed, the death of these cells. So by using PGA34, it was essentially able to block the execution of this death, this type of necrosis. And then he went on and he actually found that the cells that were rescued, they were viable, but they actually entered senescence. So they became, irreversibly growth arrested, beta-gal positive, and they started to have a classical signature of senescence. And this is something that we defined CODIS as conversion of cell death into senescence. So I'm not going to go much into the detail, but what we found was that essentially by inhibiting PARP1 activity during high doses of oxidative stress, we were able to reduce the mitochondrial calcium overload that normally happens. And because of this reduction of calcium overload in the mitochondria, cells did not really enter necrosis. But they entered senescence because actually they still had a lot of damage in their DNA, nuclear DNA. So that's the reason probably that they still enter senescence. So we couldn't rescue completely the cell, but they entered this senescence state. But then eventually what we did was we went in vivo. So we used a model of ischemia reperfusion in kidneys in vivo. And this is quite simple because you can simply surgically block by ligation blood flow to the kidney for a certain amount of time. Normally we do between 26 and 30 minutes. And then you release the blockage. And again, you create this massive reperfusion that comes with a lot of oxidative stress and a lot of necrosis. And what we observe is that, as we have seen in culture, also in vivo, there was an association between the feeding of PGA34 to mice and a switch between necrosis and senescence. So when we measure the necrotic state of the kidney from mice that were after ischemia treated with PGA34, we could observe a lower burden of necrosis. But on the other side, we actually saw a higher burden of senescence. So we saw P21 positivity, we saw P16, and we saw other markers. Suggested that also in vivo, perhaps we can achieve this CODIS. Interestingly enough, we then looked a little bit longer term, so after a few weeks, 5 weeks, where the kidneys start to actually degenerate and start to be fibrotic, we could see that the mice that received after the ischemic event PGA34 had much less marker of kidney damage, which is here represented by this marker called KIM1, and they also had less fibrosis. So we could prevent excessive fibrosis simply by exposing the mice to this PGA34. And this is a very short treatment because we only do the first couple of days after the ischemic event. It's not that the mice will be on drug. It's just this small switch that we have at the beginning, having more senescence and less necrosis, that help to recover better from the initial acute injury and then eventually help to have the tissue to degenerate in at least at a slower pace. We haven't gone longer than this, but we believe that this can be a very interesting approach to limit indeed the chronic and long-term consequences of having problem with your acute repair. So eventually, this is just what we've seen, and this is published. So if you're interested, you can look at it. So now going back, so this is the idea that we have probably different type of senescence, but we still have the problem of the detrimental senescent cells. But now, as I said, in tissue repair, you really don't want to mess up with your senescent, at least P16 positive cells, because they might be beneficial. So the idea would be to try to find something that is a little bit more selective for detrimental senescent cells. And I wanted to give you an example that we are developing in the lab. This is, again, indeed a story that started by understanding a little bit more about biology of senescence. And it is pioneered by Marta, postdoc in the lab, and Meng Ling, a PhD student in the lab. So actually, this work started with Marta coming to the lab to understand connexin 43. So she, during her PhD, studied connexin 43 in the context of osteoarthritis, and she started to be very intrigued by the fact that potentially connexin 43 was associated to senescence as well. And this connexin 43, as a main function, is a gap junction protein, so regulate gap junction activity. So she found that mouse, human, so here is mice and here is human, different type of cells. When they enter senescence, they start to overexpress this connexin 43. So during senescence, connexin 43 is overexpressed, and is actually localizing where you are when it is supposed to localize. So it is on the plasma membrane of the senescent cells. So it's there, potentially doing something. So then, as I said, major role of connexin 43 and other connexins is to regulate gut junction activity. However, when we look at the gut junction activity of senescent versus non-senescent cells, we did not find any difference. So they don't seem to be different in how indeed they can regulate these gut junctions. However, connexins, and in particular connexin 43, can also form, instead of these gap junctions, what are called hemichannels. So these are channels that serve the cell to release in a non-selective way a number of molecules, normally small molecules. And in particular, there is one that is non-selectively eliminated by cells, which is extracellular ATP. So ATP going out in the extracellular environment. So of course, we know a lot about ATP as the energy that we use in the cells for a number of functions. But ATP can also be secreted by cells, and normally is an alarming protein. So it can stimulate, for example, immune cells to come and fix some problem that happens. It's a danger molecule. Interestingly enough, senescent cells seem to release more ATP in the extracellular environment. And this phenomenon seems to be mediated by connexin 43, because if we knock down connexin 43 from senescent cells, now we had a sort of like normalization of the amount of ATP that was released by the senescent cells. So another interesting function of ATP and biological function of ATP is the fact that it also works mainly autocrinally. So it's actually picked up by the same cell that releases it. So it's very unstable outside the cell. And normally the same cell that secretes it can use it in a way, either directly, so as ATP, or as derivative. So many cells express also enzyme ectonucleotidases, which are able to degrade ATP in derivatives, ADP, AMP, and adenosine. And all these molecules, ATP or ATP derivatives, have their own receptor on the plasma membrane of cells. So we decided to look at whether senescent cells also has some differences in the expression level of all these type of receptor for ATP or derivatives. And interestingly enough, there was also some ideas that especially the receptor for ATP can activate NF-κB. And of course, here we became very intrigued because we know that the SASP and the pro-inflammatory proteins part of the SASP are also dependent on NF-κB activity. So we thought that maybe there was some links here between the classical SASP and a non-classical mechanism regulating and reinforcing the SASP. So then we went to look a little bit about the expression level of these receptors, and we found that actually in one of there were different receptors that were differentially regulated as senescence, but in particular one, P2X1, which is one of the P2XR receptor, so receptors that can bind directly ATP, was highly overexpressed in senescent cells. And we validated this in various ways, so I'm not going to spend too much time on this, but we found that it was indeed expressed in various types of senescent cells, and again, we found that it was on the surface, so also localizing where it's supposed to be. So potentially there to bind ATP. Then we look at the upregulation of this P2X1 in data sets of either skin wound healing or aging skin. And what we did there was to isolate P16 positive cells and look at the transcriptome of these P16 positive cells to fish for the expression of P2X1 and also of connexin 43, the protein that I described before. So, and what we found was very interesting because while both the wound-associated P16 positive cells and the age-associated P16 positive cells were expressing higher level of connexin 43 compared to the P16 negative populations, suggesting that both beneficial and detrimental senescence, or at least this type of beneficial and detrimental senescent cells, are able to secrete ATP. Only the age-associated P16 positive cells overexpress the P2X1 protein, so the receptor for ATP. So only these detrimental age-associated senescent cells were somehow supposedly using this ATP for some signaling that was autocranally happening. And we thought immediately about NF-κB because, of course, one of the things that we know about wound healing associated and aging associated is that the NF-κB activity is not the same. Actually, in wound healing, it seems that the NF-κB is much lower than it is in aging associated senescent cells. So we tried to look into NF-κB activity, and what we found was that when we knock down P2X1, we actually suppress the production of most of the classical NF-κB regulated SASP factors. So you can see, in the various interleukins that we normally associate to NF-κB activity, were all down in senescent cells that were knocked down for P2X1. So then we decided to also look a little bit more translationally and trying to look for compounds that could block this receptor, this P2X1 receptor. And there are a number of molecules that have more or less selectivity against these receptors. We tried all the ones that were supposedly blocking the P2XR receptors. And with the exception of one of these compounds, all the others were able to suppress the SASP from senescent cells. So then we decided to select this compound here, which is called an NF157, a decision that I'm regretting because this compound is extremely expensive. So when we went to use this in vivo, we spent a fortune. Some others might be cheaper, but this was already quite well tested in vivo as well. It's not approved, but it was tested quite extensively in vivo. So that's the reason why we selected this compound. So at first, we just trying to use this compound to look at the SASP in various contexts and in culture, and we found that independent of the model, we could see that these NF157 were able to suppress the SASP. Importantly, we didn't find any senolysis, so it's not that applying this compound to senescent cells was inducing death. There was nothing. Actually, it was very well tolerated by other proliferating or senescent cells. We could go up to 100 micromolar. Nothing happened in both type of cells. But then we decided also to look a little bit more complex structures first. These are actually tissue explant from actually osteochondral explant. And we did this in collaboration with the laboratory of Ingrid Mullebelt at the Leiden University, and in particular Ilya, which is a PhD student in our lab, did these experiments for us. So they took these osteochondral explants, and then they have a model where they can induce these explants into senescence by a combination of radiation and mechanical stress. And then we exposed these cultures to NF157, and essentially what we found, also here you don't see much, but essentially we didn't see any senolysis, so there was no elimination of these senescent chondrocytes that are in these explants. But again, what we saw was that the application of NF157 on these cultures was sufficient to reduce the SASP that was generated by the exposure to this double radiation and mechanical load stress. So then we decided to indeed move to an in vivo context, and we did a number of different models. The first one is a very simple model of chemotoxicity. So we exposed the mice to high doses of doxorubicin, and then after a few days, we treated the mice with NF-157. So normally, because of the exposure to chemotherapy, mice get kind of sick. They have a lot of physical issues, and they also start to have a pro-inflammatory phenotype in various tissues. Indeed, what we found was that indeed liver and kidney, we have the upregulation of various SASP factors, but the exposure on NF-157 somehow normalized the expression of these. We found that there was hyperactivity of NF-kappa B, especially in kidneys, and this was also normalized by exposure to NF-157. And eventually also when we look at the physical performance of these mice, we could see a very nice amelioration of different parameters when we applied NF-157, when we fed the mice with NF-157. So there was an improved strength and an improved endurance in these mice, suggesting that indeed by reducing the inflammatory profiling potentially of the senescent cells in this context, we were able to improve their health. So then we decided to move, and as I mentioned before, one of the reasons why we started the whole study was that Marta, our postdoc, was actually interested in studying connexin 43 in osteoarthritis in the past. So we decided to look at whether this was something interesting also in terms of osteoarthritis. And of course, senescence has been described as a modulator of osteoarthritis, can accelerate the pathogenesis of osteoarthritis. So at first, we used a model of LPS-induced osteoarthritis, very dirty models, but quite simple. So we simply inject high doses of LPS in the knee joint of mice, and this is enough to create these OA-like symptoms. We found that indeed this can generate a lot of senescence and also induce knee inflammation, and actually make the ability of the mouse to use that joint much worse. So if you ask the mouse to respond to a hot plate, it will take much longer to the mouse to do that because it feels pain and it doesn't want to move the joint. However, when we exposed the mice to NF-157, first of all, we found that there was a suppression of the SASP also in this context, in these mice. But in particular, we also saw improvement in OA-like symptoms. So there was reduced inflammation in the knee, and also now the response time to the hot plate was somehow normalized. So the mice that received NF-157, even with osteoarthritis, could respond quite well. So then we repeated a similar experiment, but with a completely different model. This is in collaboration with Jennifer Elisef, John Hopkins. So Jennifer is an expert on osteoarthritis, and in particular on mechanical osteoarthritis, which in this case in mice we do by surgical removal or cut of the joint of the knee ligament, which is enough to create a cascade that lead to osteoarthritis because of cartilage deterioration. So Jennifer did this experiment for us, and she indeed treated the mice with NF-157 during this OA-like model. And she found that also in this case, we found that there is a reduction of the SASP in the knees that are treated with NF-157. And again, there is a better response to the hot plate. So actually the knee joint seems to function better when the mice are exposed to NF-157. And then at last, all these experiments motivated us to try the aging experiment, trying to see whether we could do something for an aging mouse. So we started with middle-aged mice, 18 months of age. We treated the mice with various cycles of NF-157. So now we give 3 injections every other week. And then we did for a few months, and we monitor essentially just frailty and a few physical parameters in the mice. And to make a long story short, what we could see was that the endurance of these mice was actually much better. So the mice that received NF-157 was much better than the mice that didn't receive. So rotor latency and cumulative distance on a running wheel was significantly improved in the mice that received NF-157. And also the strength in these mice was significantly improved when we look at hanging test and grip strength, suggesting that also during aging, we could potentially consider this compound to reduce the inflammation. And potentially also what we can think about is that potentially we are also reducing a little bit the accumulation of senescence, as what we are seeing in certain tissues, because by blocking the detrimental SASP, we might also reduce a little bit paracrine senescence effects. And in the long term, this might serve as also a way to prevent senescence in these animals, and that's why potentially they benefit from the treatment. So this, in summary, what we think is that senescent cells seem to secrete higher level of ATP in the extracellular environment. However, they can also use potentially more the ATP that is available in the surroundings to signal via P2X1 to activate NF-κB and potentially to sustain the SASP and the detrimental SASP, what we call the NASP for NF-κB associated secretory phenotype, which is related to age-related dysfunctions. And potentially by blocking this signaling cascade, we are able to reduce the burden of age-related dysfunctions. And with this, just a general conclusion, so I hope that I convey the message that, of course, senescent cells and the SAS can cover both beneficial and detrimental function. Of course, there are many other beneficial functions and many other detrimental functions. I just show you a couple of examples, but there is much more to learn about this. I mentioned that some of the current therapies might interfere with at least a pool of beneficial senescent cells, so we need also to be a little bit careful about this. And I show you the potentially purinergic signaling, so the ATP signaling seems to be associated with detrimental senescence, and its inhibition could ameliorate senescence-associated dysfunctions. And I would like to stress on the idea that it is true, it's fantastic, we're moving into so many clinical trials and so many translational efforts, but we have so much to learn about the basic biology of senescence, which we hope that besides having venture firms and pharma interested in investing money, we also have funding agencies that want to fund basic research on senescence, because there is a lot to learn. And we have now very nice tools that help us to actually really understand the biology of senescence, and we need more investments there to use these tools and to understand this better. So I think I mentioned everybody that, and one slide after, but everybody that participated in the experiments I showed you. And I just wanted to emphasize that at Aging we have this call for papers for a commemorative collection honoring Judy Campisi, which I think many of you in this audience knew, which just passed away last year. So if you have an interesting paper on senescence, senotherapeutics, on anything, please submit to Aging, because it would be nice to indeed collect nice biology, nice interesting ideas on senescence for this collection. Thank you very much.
Thank you very much, Marco, for this very interesting results and inspiring talk. There are certainly some questions. I'll start very briefly with one short question. You mentioned the importance of basic research, and I fully agree, but anyhow, how far away are we from a clinical trial with an F157? And if we go in a clinical trial, you mentioned that this compound is quite well described in terms of safety. Also, should we expect some adverse effects, and which one?
Yeah, so unfortunately, these compounds have never been gone through any type of clinical trial, despite aberrant extracellular ATP signaling seems to be associated to a number of diseases. The reason is not clear to me. I couldn't really understand the reason why. One of the major issues that we might have, though, is that, of course, immune cells also use this, and it's important to activate certain immune cells. So we need to be, trying to be a little bit more pragmatic here, trying to also understand what is the long-term effect of applying these compounds systemically, or, on the other side, trying to find something that we can directly target two senescent cells instead of hitting also the immune cells. That's where I see the problem, that the immune system might also suffer from long-term application of these compounds.
Okay, thank you.
So I think on one of your slides, it seemed to show that NF157 also downregulates expression of P16. Was that right?
Slightly.
In the LPS model?
Very slightly, it does.
Oh,
yeah.
Okay. I was only... Maybe if it's only slightly, maybe it's not relevant. But I was going to ask whether you think perhaps in vivo could it be senolytic and/or actually be bypassing the cell cycle arrest, in which case could either of those activities be contributing to the effects?
Actually, we have two potential mechanisms happening. One, as I mentioned, it could be the prevention of paracrine senescence, and that's the reason why we see that if you apply for a few times this compound, you see less P16 positive cells. And the second one is that we believe that actually this ATP signaling is used by senescent cells to protect themselves from immune-mediated clearance. In particular, potentially macrophages and NK cells. So maybe because we are now blocking this signaling from senescent cells, then they are cleared more efficiently. And that's the reason why also in the longer term, we might see that we have senolysis, natural senolysis.
Right, makes sense. Thanks.
Marco, thank you very much for the talk. As you know, connexin 43 is a very important protein in the heart for the gap junction. Have you checked in your model, in doxorubicin 1, but also in the aging model, if you have overexpression of connexin and the formation of these hemi channels?
Yeah, not really. I mean, the whole tissue is always complicated. We wanted to go a little bit more single cell level, in particular for senescent or senescent-like cells in tissues. And I show you the aging skin as an example, but we've looked at different disease models and trying to look at the signature of a P16 positive cell. And most of the time we see connexin 43 being high. So it seems to be quite correlating with P16 positive cells, that's for sure. However, of course, there might be other cells that also have it. In the bulk is always tricky though, but we are doing more studies on single cell to see whether, this can be a mechanism, general mechanisms for P16 or generally senescence-like cells.
So thank you, Marco, for the talk. Really interesting. I'm curious about the level of the intracellular ATP and the state of the mitochondria of the cells, no? Because in our hands that we are studying senescence in cardiomyocytes, the mitochondria are quite compromised, and we observe this huge decrease of intracellular ATP. So just curious. Andrew?
Yeah, but it might be that they also release more, and that's the reason why they have less intracellular ATP, right?
For that.
So it's an excellent question. That's actually our next step to understand why they have all this ATP or why they release all this ATP. We don't understand it. We know that indeed it's connected to having more connexin 43, but why it doesn't stay in the cell, we don't know. But at the beginning, we thought indeed to look into whether there was, but we actually end up with the same conclusion that we see less intracellular ATP in senescent cells, but maybe one of the reasons is that they secrete more. It's a little bit difficult, we realize, to study the fluxes and trying to, track live cells and how they are, producing and releasing. So we're struggling a little bit with that, but I think that that's something that we should do. Yeah.
Thank you.
Yeah. Here.
Yes, please.
Thank you for the talk. Outstanding. Is there any information on the downstream mechanism of P2X1? I mean, if it is only a sodium channel or there are some phosphorylation events that may be important also for targeting and preventing ATP effects?
Well, I mean, actually P2X1 seems to be regulating two important potential mechanisms also in senescence. One is calcium, and the other one is the inflammasome. So both of them seem to be affected directly by P2X1. So we are working on that side at the moment. We know, for instance, that calcium signal is extremely important regulator of this transition from P2X1 to, for example, pro-inflammatory and FKB activity, etc. We don't know yet the inflammasome itself, but we believe there is something there as well. So these are the mechanisms where we are focusing on.
Yes, we have a last question here.
Nice talk, thank you. I have a question about the story of PARP inhibition that move necrotic cells to senescence. PARP inhibition is used in the clinic for cancer patients affected by DNA damage repair gene. And there are new evidences showing that also patients unselected by this mutation can benefit. Do you have any clue about the impact of PARP inhibition in DNA damage repair deficient and proficient cancer cells in terms of senescence?
No, I don't. The only thing I know is that, well, I mean, it's an important point to note that what we normally use, we do with PARP1 inhibitors is that we just do 1 or 2 treatments. So it's probably not enough to affect anything. And at least we never look at proficient and deficient cancer cells. But what I can tell you, if I look at the DNA damage in a senescent cell, that is generated by this, or where we apply the same amount of PARP1 inhibitor we use to switch necrosis to senescence, we don't see any difference in terms of DNA damage. So we are not affecting that much the DNA repair activity of PARP1 with just this dose. That's the only thing. But of course, maybe there are differences if a cell is proficient or deficient, and in particular in the cancer context, this might be interesting. Yeah.
Any other questions? Doesn't seem the case. So thank you again, Marco.
The next one is Maria Antonietta D'Ambrosio. She's postdoctoral research at the MRC, London Institute of Medical Science in the UK. She got the PhD from the molecular oncology lab from Professor Alimonti and was in Imperial College. And I hope we can say something new about these new drugs, Senolytics drugs. Thank you.
Hi, good afternoon. I am Maria Antonietta and I work in Vassilis G lab. And today I will present you my project that is titled Screening a Library of Covalent Inhibitor Identify Novel Senolytics. Covalent inhibitors are very old inhibitors that have the characteristic to bind cysteine or other amino acid inside the protein. There are a lot of common drugs that are covalent inhibitors, like for example the aspirin, and in the last 20 years a lot of cancer drugs are covalent inhibitors, like for example inhibitor of BTK. They can have several advantages, such as the possibility to target protein without an enzymatic domain, and they are able to bind the target that are very considered undruggable, like for example different RAS mutation, but they can have unexpected toxicity. To start our screening, we use a model of oncogene-induced senescence that are IMR90 RAS, in which we induce senescence with the treatment, with the tamoxifen treatment for 7 days. And then we screen our 10,000 compound at a one single concentration, 10 micromolar. And then after 3 days, we use our as a readout DAPI staining that we acquire and analyze with the InCell. Once we have the first senolytic candidate, we tested not only in IMR90 RAS, but also in 2 models of therapy-induced senescence that are A549 and SKML. Once we have the senolytic kits, we wanted to deconvolute the target, and we did it using a pull-down experiment thanks to clickable probes. And once we have the target, we wanted to validate both in vitro and in vivo. This screening approach allows us not only to find a new target, but also new chemical species. These are the results from the first screening in IMR90 RAS. The pie chart represents, summarize all the warhead of the covalent inhibitor we have in our library. And among 10,536 compounds, we found 72 senolytic kits, and the majority were chloroacetamide and cyanoacrylamide. Once we have the 72 senolytic kits, we decided to retest them at different concentration in ERAS, SKML, and A549, and we found 47 senolytic kits that were working in at least one of the 3 cell type in which we tested. And since in SKML we got a lot of positive hits, we decided to continue our experiment in this cell type. SKML are melanoma cells in which we induce the senescence with etoposide and tozasertib, and we went to check for senescence response, and we saw that the cells stop proliferating, express P21, and become beta-gal positive. In SKML, we found 4 chloroacetamide that have a senolytic activity that is comparable to the ABT263. So we decided to see if our compound were senolytic also in different systems, and we tested in IMR90, in which we induce senescence with etoposide, a model of ovarian cancer in which we induce senescence with cisplatin, and a model of breast cancer in which we induce senescence with etoposide and tozasertib. Since our compound were broad-spectrum senolytic, we want to understand the target. For this reason, some chemists developed for us two different probes for each compound. These probes have the same chemical structure of the original compound, but they have attached an alkene group that will allow us to deconvolute the target through pull-down experiment. The first thing we did was to double-check that the probe retained the senolytic activity, and this was the case. Actually, they had a senolytic activity that was comparable to the original compound. So we perform a pull-down experiment, and this heat map summarizes our results. These compounds were really reactive, so a lot of protein were bound, but there was a protein that was bound from 3 out of 4 chloroacetamide, and this protein was GPX4. This is more clear in this Vulcano plot, in which we compare the compound that bind GPX4 to the one that doesn't bind. What is GPX4? GPX4 is an antioxidant enzyme that helps to maintain redox homeostasis in the cells. So basically is a defense mechanism that the cells have against ferroptosis, that is a cell death characterized by accumulation of iron, ROS, and lipid peroxidation. Once GPX4 is deactivated or inhibited, we have accumulation of iron and ROS in the cells, lipid peroxidation, and ferroptosis. So basically, GPX4 can be compared to BCL, XL, or BCL2 in apoptosis as a defense mechanism. So to see in which way our compound were having the senolytic activity, we decided to use different cell death inhibitor, and what we saw was that the 3 out of 4 compounds were rescuing cell death through ferroptosis and only one through apoptosis. And the one that was rescuing through apoptosis was the only one that was not binding GPX4. To see if our cells were prone to ferroptosis, we stained them for different markers, and what we saw was that in senescence condition, we have more lipid oxidation, more accumulation of iron, and more reactive oxygen species. And at the same time, we have also the overexpression in senescence condition of GPX4. Ferroptosis involve different pathways, so we went to check if there were other ferroptosis inducers that were having this senolytic activity. And what we found was that only GPX4 inhibition and erastin was having in our cells senolytic activity. So we tested already available GPX4 inhibitor in our SKML, and we saw that they were having a strong senolytic effect, also a very low concentration. And we did a few more testing with RSL3 in different cancer cell line, because RSL3 is a covalent inhibitor and we want to use for in vivo experiment. And we saw that it was senolytic also in ovarian cancer and in IMR90 treated with etoposide. So to conclude, our screen allowed us to find different covalent inhibitor with the senolytic activity. Few of these chloroacetamide have a common target, that is GPX4. Senescent cells are primed for ferroptosis because they have a high level of iron, high level of lipid oxidation, and high level of ROS. Induction of GPX4 prevents ferroptosis, but its inhibition induces senolysis through ferroptosis. I want to acknowledge all my lab, in particular Jesus for the opportunity to work to this project, Laura and Jody that helped me with different experiments, and at the Tate lab, in particular Matt that worked with me for all the pull-down experiment, and Eftimios that synthesized for us the probes. And thank you all for the attention.
Thank you very much. Session open for questions.
No question. Okay.
Move to the next
Let's go to the
speaker.
next one. Yeah.
Yes, to Benjamin Le Calvé, who is the Chief Scientific Officer at Therapeutics in Lille, France, who will speak about DPP-4 as a new convincing target to eliminate cancer and senescent cancer cells. Thank you.
So firstly, I would like to thank the organizer to give me the opportunity to present this data. That's the first time I present this research program in front of scientists. Normally, I present in front of investors in 5 minutes. So 8 minutes, it's almost paradise for me. So our project is focusing on DPP-4, and we are developing targeted therapy able to eliminate cancer and senescent cancer cells. This is my disclosure statement. So, as you know, probably better than me, so conventional cancer therapies such as radio, chemo, immunotherapy, or targeted therapy are able to eliminate most of cancer cells on tumor site, but some of them resist and acquire a senescence phenotype. And this senescence phenotype is directly related to deleterious effects for the patient, like EMT induction, immunosuppressive environment, or remodeling of extracellular matrix. So at Starcage Therapeutics, we would like to develop a new approach in which we would like to develop antibody drug conjugated able to eliminate cancer and senescent cancer cells. So the first step of the project was, okay, if we want to develop an ADC, we need a target. So we perform a screen by surfaceome analysis, and we discover more than 200 potential targets expressed at the plasma membrane of senescent cells compared to the normal cells. So we apply several filters to determine a short list of potential targets. And finally, we selected one of them to design the first ADC against senescence. And this target is DPP-4. So you probably know DPP-4 because it's a protein expressed as a plasma membrane of cells. This protein is involved in enzymatic activity. It is already known to be implicated into the binding of protein of extracellular matrix. This is recognized as a good senescence marker, such as other potential targets like UPAR protein or CD36. The expression of DPP-4 is induced by therapy-induced senescence, and also DPP-4 is known to be overexpressed in a wide range of pathology related to aging. So the most interesting, so we highlight that DPP-4 is involved in senescence, but also when we test and we analyze the expression of DPP-4 in cancer, so we discover that this protein is overexpressed in a wide range of cancer. So we decide to focus on 2 main indications, so on liver cancer and pancreatic cancer, and also in liver metastasis, because we found that this protein is clearly overexpressed in this kind of indication. But the most important, we perform and we create some models in which we induce senescence in liver cancer and pancreatic cancer. And by inducing senescence, by radiation, or by hypocyclic, cisplatin, and toposide, we validate all these models by analyzing the level of beta-galactosidase activity, or the level of expression of Ki67, or other markers such as P63, P21, or P16. But in all these kind of models, when we induce senescence, we induce an overexpression of DPP-4. So clearly, the senescence phenotype is able to boost expression of DPP-4. So if you have a good expression in the cancer, we are able to induce more expression by inducing senescence in this kind of cancer. So, okay, the second step, we have a target. So now we have to develop a drug candidate. So we develop some specific antibody against DPP-4, and also we need a payload, but a payload effective on cancer and senescent cancer cells. So we perform a screen with potential payloads, so already known to be used in ADC field. So as you can see, we screen the MMAE, PARP inhibitor, gemcitabine, NMT inhibitor, or topo-1 inhibitor. And we screen the activity of each payload on cancer and senescent cancer cells. So, for example, with the MMAE, so we have a very good activity on cancer cells, but we have no activity on senescent cancer cells. So, probably, yeah. Okay, just here with the MMAE. So, the only one molecule which displays a great activity on cancer and senescent cancer cells is the topo-1 inhibitor, exatecan. So based on this result, we decide to develop the third generation of ADC, which targeting DPP-4. So we associated our antibody against DPP-4 with the exatecan DAR8, and we validate in vitro the activity of this third generation of ADC. So as you can see on this graph, so we have the activity in vitro on different models of cancer and senescent cancer cells. So we have a huge activity in both populations. We also validate the STX1, this ADC, with internalization assay, ADCC activity, and bystander effect, so everything is okay. Okay, the last step. So we need to validate the activity of this ADC in vivo model, but also the safety associated to this treatment. So the first step, so we use classical model of cancer. So we use the HG2 model, which is a human hepatocarcinoma, which expresses a high level of DPP-4. And the second model is the HPAF model, which is a pancreatic model, which expresses a low level of DPP-4 compared to the HG2 model. So as you can see with the HG2, when we treat the mice, in which we have grafted the tumor in the subcutaneous models. So at 5 milligrams per kilogram or at 10 milligrams per kilogram, we observe a strong anti-cancer activity. We are able to stop tumor progression, and we observe a strong regression of the tumor size. And this effect perdures into the time. So for the HPAF model, so in which the expression of DPP-4 is low, as you can see, when we treat with the ADC, we are able to delay tumor progression, but not stop the tumor progression. So after 50 days that we observe a strong relapse of the tumor. So keep in mind this result, because after that, we try to develop cancer in vivo models in which we induce senescence. And we use a model developed by the Scott Law Lab in which they treat the mice after the tumor injection with a combination of trametinib and palbociclib. So during 4 days in a row, you treat your mice with this combination, and clearly this combination of treatment induces a large amount of senescent cells into the tumor. And after that, we are starting to treat with the ADC. So for the HG2 model, so what we observe the same activity even we induce senescence into the tumor models. But for the HPAF model, so clearly after 50 days, that we observe is a good activity of the ADC. So clearly the senescence phenotype are able to potentiate the activity of our ADC when we induce senescence. So probably this effect is due to the overexpression of DPP-4, but not also the expression of DPP-4, but probably a higher bystander effect into the senescence tumor cells. So the last point is the safety. So it's very important because we need to be sure that our treatment is safe. So we use a model of mice in which we deplete the mice form of DPP-4 and we replace the gene by the human form of DPP-4. And we treat the mice at high dose of the ADC at 25, 50, and 100 milligrams per kilogram. And we analyze the body weight and also some blood samples, for example, the liver activity and also the kidney activity. So we observe any side effects related to the ADC treatment. So clearly, we have a treatment efficient, but also safe in the relevant mice model. So to conclude, so STX1 is the first in-class ADC which target DPP-4 protein in cancer and also in senescence. Clearly, this first generation of ADC is efficient on cancer and senescent cancer cells. So we are developing some other models in vivo, particularly with the radiotherapy and the PARP inhibitor. We have a large therapeutic window with any side effect observed at high doses of treatment. And also, we need to test this ADC in PDX and organoid models. So we have a partnership with the Institut Gustave Roussy, and now we have an access to the bank of PDX to test the efficacy of our ADC. And also, we would like to extend the research program because probably DPP-4 is involved in other pathology, and we would like to focus on other diseases such as fibrosis or osteoarthritis. And we have a second target into the pipe, and we would like to generate bispecific approach because we have some evidence that this second target colocalized with DPP-4 during the senescence phenotype. So I would like to thank all the team, especially Dr. Thierry Mathieu, which is the founder of the company, and also some specific advisor of the company. So Eric Angevin, which is an oncologist at Gustave Roussy, and Florence Lospice, which is a specialist of ADC development. So sometimes I heard something on Nectin-2. So Florence is one of the co-founders of Emergence, which develops the Nectin-4 antibody acquired by Eli Lilly. So thank you.
So the last speaker is Nathan Lebrasser. It's director of the Robert and Arlene Kogod Center on Aging on Mayo Clinic in USA. Well, he has different positions, so let's see what he's going to tell us. Thank you.
All right, well, thank you very much. It's fantastic to be here, and really want to take a moment to echo the comments of others and congratulating IPSA Foundation on putting together such a remarkable conference. Kudos, it's been outstanding. I'm exhausted. It's been so good, but I'm also inspired, and kudos to Fabrizio and Andrea for doing such a stellar job, and certainly Alessandro and Giovanni. You probably need a neck massage after chairing this session with the neck twisting, but it's great to be here, and I have to confess before I get started, I've changed my talk a little bit. I haven't changed my title, but recognizing that I'm the last speaker in the translational session of this conference, I'm going to push a little bit more on the human side of our research, which is only about 25% of my laboratory's program. But I think it's a good way to end as we think about taking these emerging technologies from bench to bedside. It's really been an inspiring day, and I think it's time to have serious discussions about this. I'm fortunate to have a remarkable research program. I'm going to highlight the work of some early stage investigators in my lab, Xu Zhang, and some postdocs, including a very fine Italian, Zaira Aversa, who trained in Roma at Sapienza University, and Sarah Ashik-Calais. And I just have to say, too, that most of my knowledge and understanding of senescence is because I'm at a remarkable institution with an outstanding group of investigators. You've heard several of them mentioned today: Joao Passos, Diana Yerk, Darren Baker, Sandeep Khosla, and others listed here. I should mention Tamir Chandra and Christina Kirshner, who are friends of many of yours as well. So, and thanks to many of you in the audience that have really inspired my thinking in this space. So to that end, I want to talk a little bit about this idea of biomarkers. And it's hard to not open the newspaper and see an advertisement of getting your biological age measured, or to go to a fitness center and people telling you what your biological age is. And we've been thinking a lot about this over the years in the concept of biomarkers of aging from the sidelines of senescence. And I'll get into that in just a second. But I want to emphasize that even before we have a tool to bring to the clinic for targeting senescent cells, biomarkers of aging have incredible utility today. And that's because all of us, whether you're in Italy or the USA or in Switzerland, are dealing with a very high percentage of older adults taking our slots in the clinical setting, right? We have a very high percentage of payers who are of an advanced age. And when it comes down to it, it can be challenging to decide whether that individual should undergo an invasive medical procedure or should be managed medically in a much more conservative way, right? We understand that a very high percentage of our resources in healthcare go to a very small percentage of patients. And sometimes we just have to acknowledge that the eyeball test saying that this person, this individual is frail because of their chronological age or because of how they look or how they're moving in the room isn't sufficient to really guide that decision-making. So really, I think biomarkers of aging can be of incredible utility for choosing the right treatment for the right patient at the right time. And it doesn't mean those individuals should not undergo that more aggressive procedure, but perhaps they'd benefit from some prehabilitation strategies to improve their health and function and their resilience to counter the biology of aging before they go into the clinic for that procedure, right? And for me, we think exercise is the magic bullet. It may be nutrition. I'm sure Walter Longo will talk about that later today. Or it could be these emerging drugs that we're all talking about that could be part of this regimen that we use to take better care of older adults. And of course, some of those who don't tolerate those procedures well need a lot of resources after they're discharged from our facilities. So we think about more robust transitional care planning for those older adults as well. So that's a pitch for how we can use biomarkers today. And of course, as we kind of bolster research in the space of interventions targeting the biology of aging, we think a lot about, well, who should receive what intervention? Today, you've heard a lot about Senotherapeutics, but I can promise you that there are conferences going on simultaneously about the other hallmarks of aging and people engineering drugs, devices, therapeutics, cells, et cetera, to target those other hallmarks. So how do we decide who gets what, right? That's a big deal. In addition to that, many of the outcomes that we're most interested in, right? Changing cognitive function, changing physical function, changing pulmonary function, changing metabolic function, those changes take time. And as we bring new ideas into the clinical setting, we'd like to know if they're moving the needle much sooner than it takes a year or two to change cognition or function or those other outcomes that I mentioned. So the perfect biomarkers are, number one, they're predictive of future health, right? And number two, they're responsive to an intervention. And we kind of now acknowledge those biomarkers as surrogate outcomes or surrogate biomarkers that can be very useful in the clinical setting because, one, they're responsive, so we know if the drug is working or not, having the biological activity we hope it has, as well as they're predictive of future outcomes that the patients care about, right? So this the other pitch there. And then, of course, thinking about how we move these ideas from bench to bedside means that we have to have a very good understanding of their predictive ability, but also of their underlying biology. And we've talked a lot today about how senescent cells drive disease and dysfunction, and without question, we're excited about the SASP in my lab because we're really concerned about why does the SASP compromise skeletal muscle regeneration in the context of injury and aging? How does it drive progressive deterioration and degeneration of bone and muscle and other joints? How does it drive aberrant growth and remodeling in the case of cancer and other conditions? We talk a lot about inflammation in the context of aging. Inflammation is a hallmark of so many different age-related conditions, but what's the source of that inflammation? And we would argue that senescent cells are a very plausible and significant source of that inflammation. And we've heard a lot today, too, about how the SASP can convey secondary senescence into other cells and tissues. And as much as I remark at the biology and the function of the SASP, we should also recognize that there is a unique element of this hallmark of aging. And potentially, components of that SASP could be used as biomarkers of senescent cell burden in individuals. And I'll walk through some examples of that and also share with you at the end some new ideas that we have in this space. So we took a very simple approach several years ago, thinking about if we exploit the literature, talk to our colleagues in the field, whether it's Marco or Judy Campisi or Brigitte Schilling or others, what are common elements that are core components of the SASP that we see across tissues in mice and in humans that we can sometimes detect in the circulation? And we put together a very simple list of cytokines, chemokines, matrix remodeling proteins, and growth factors that are common elements that we see in the SASP. And the point of this slide is to show you a couple of things. Number one, on the top three rows in each box there are control cells, C1, C2, C3. And in the next three boxes are senescent cells, so SNC1, 2, and 3. And you can see just how remarkably robust the secretome of these cells are, at least in vitro, right? This is not an artifact of transcript numbers of a low CT value going to an even lower CT, or a high CT value going to a lower CT value, but this is protein abundance in the media. You see up to 750-fold changes in the protein concentrations in the media by what these cells are producing. It's really quite remarkable. In addition to that, here are 5 different human cell types, and you can see how diverse their SASP appears, even with this very simple kind of heat map that I'm showing you here, right? Showing the complexity and the diversity, the heterogeneity of senescent cells. So we really moved forward with this very simple platform. It really combines kind of a multiplexing on the Luminex, using the Luminex technology or MagPix technology that some of you are familiar with. We also coupled in ELA, which is a more high-sensitive way to detect lower abundance proteins. And then we use an ELISA for a couple of the proteins as well. And I won't go into the details here, but this just shows you how we're trying to capture more than just inflammation, but trying to capture multiple hallmarks or features of this senescence-associated secretory phenotype. And what's cool about this is that we've now used this simple platform in probably too many studies to show that, yes, surprise, surprise, as you get older and you have a higher senescent cell burden, most of these proteins change in the circulation in their abundance with advancing chronological age. In the context of disease and disability, if we compare those with disease, or those with functional limitations, or those with cognitive decline, or those with other elements of advanced chronological age or biological age, we see a higher abundance of these proteins in the circulation of those who are affected versus those who are unaffected. And we've been able to utilize these now for the purpose that I said earlier of identifying individuals who are undergoing surgery for heart disease or for cancer, and we can predict those who are going to have consequences like rehospitalizations, surgical complications, and other things after the procedure, just based on the abundance of these biomarkers of senescence in their circulation. Importantly, I won't highlight it today, but we've been able to show as well that caloric restriction and exercise are able to lower many of these markers in the circulation as well. And I just want to give you a couple of examples. So Here are two very important outcomes related to skeletal muscle health and function. The short physical performance battery was mentioned earlier. This is a measure of standing balance, your ability to get out of a chair 5 times quickly, and your gait speed. It's a scale from 0 to 12, and what I'm showing you here is that those in the highest quartiles of these biomarkers of senescence demonstrate deterioration in their score over time, over a 2-year period, right? This is in about 1,300 individuals over the age of 70 with multimorbidities. These are very representative individuals that we studied here. And you can see, again, many of these factors change in a stepwise fashion with increasing concentrations of those biomarkers, predicting worse function over time. In addition to that, we looked at major mobility disability. So this is a very important outcome in older adults, your ability to walk 400 meters. Okay? So at baseline, all these individuals could walk 400 meters, but over the course of 2 years, a couple hundred of them lost that ability. And again, you can see here, individuals in the highest quartile of those biomarkers of senescence had the greatest risk for losing the ability to walk 400 meters independently. And I'll just show you one more example of this. In a completely separate cohort, we had another lab measure these values. Part of this is because I run a lab like many of you. I don't have robotics to analyze 2,000 samples, so I get kind of a dirty look from my lab when I ask them to run another batch. But this is just showing in 1,600 individuals in a completely separate cohort, the same biomarkers emerge as predicting this very important outcome in older adults. So that's that. In addition to that, we did a very cool study where you would say, well, maybe it's really not, in a 2-year study, the predictive ability isn't that strong. So maybe it's the fact that these individuals were already on the kind of downward trend, that their biomarkers were associated with these outcomes, right? Cross-sectional studies are very easy to show associations, but how do you start to pick away at causation? So we did a pretty cool thing that we could do at Mayo Clinic because we have this incredible biobank of about 57,000 participants. Almost half of them are over the age of 65. And we coupled that with an epidemiology project that kind of monitors and marks all the events that happen in these individuals in terms of their health care, their health needs, as well as their survival. And we then asked, can we identify 2,000 individuals in that cohort that at the age of 65 have only 0 or 1 medical condition, meaning hypertension or hyperlipidemia. So incredibly healthy individuals, right? If we can all be so lucky to make it to 65 and not have a chronic condition. And we asked, whether or not biomarkers of senescence could predict death in those individuals. So just here's a look at those individuals. We had a nice kind of segmentation of folks from 65 to 69, 70 to 74, and over the age of 75. And surprise, surprise, if at the time of study entry, you were over 75 versus you were being 65 to 69, you had a significantly higher risk of death in the next 7 years. Exactly what you'd predict, right? That makes total sense. And I'm sorry, gentlemen, the women lived much healthier and longer than the men. So women had half the risk of death over that 7-year follow-up period than the men. And this is what it looks like in terms of our biomarkers. And this maybe seems a little bit underwhelming in terms of hazard ratios, but you can see here that biomarkers of senescence compared to being in that oldest age group in terms of their strength of predicting death over that follow-up period. And most importantly is in the bottom right, where you can see that the senescence biomarkers, independent of age, sex, race, and the history of a medical condition, were just as strong predictors of death. And when you combine them together with those covariates that we all know, we have the strongest prediction for death. So for those of you who don't know C statistics, if I flip a coin in the air, I have a 50% chance of predicting whether you're going to die or not. When we add these variables to the equation, we now have over an 80% ability to predict risk of death in the next 7 years. So it's really an exciting space. So I don't do much in the cancer space. I have to disclose that up front. But we're fortunate at Mayo Clinic, again, to work with clinical colleagues who have taken great interest in this space and understanding how well their patients are going to do after invasive procedures. And a very talented surgical oncologist, Monica Kumar, takes care of women with advanced ovarian cancer. So here we have a cohort of 280 women in a 10-year follow-up period, which is pretty remarkable. And in these individuals, when we looked at hazard ratios for death, we also looked at hazard ratios for surgical complications in response to surgery, their ability to tolerate full cycles of chemotherapy. And we could again show that these biomarkers of senescence were highly predictive of adverse outcomes in those individuals. And again, it's interesting when we go from indication to indication, we see some new biomarkers pop in and some other biomarkers move out. I'm just showing you the top hits here. But then we can put those biomarkers into a gradient boosting machine learning model, which is really a black box of complex math that I can't understand or explain to you. We can then see what's the relative influence of those individual biomarkers or everything that you put into that equation. And what really jumps out to me here is the fact that several biomarkers, and I just did a short list here, are much stronger than chronological age in predicting the adverse outcomes in these patients, right? So again, chronological age does not compare to the strength of biological age. And you could say, okay, those relative influences look pretty impressive, but what does that really mean? What does it mean that I have a relative influence of 25, 22, 17, et cetera? So then we can then do modeling and say, okay, if you are in quartile 1, meaning that you're a woman with the lowest concentrations of those top biomarkers, your risk of dying over the next 10 period is dramatically different than those in the highest quartile of those biomarkers. And when we look at the 5-year survival rate, we're going from 70% survival in those with the lowest biomarker concentrations to those in the 10th percentile. And this is just using biomarkers alone. This isn't using other things like minimal residual disease or other things that you find out after surgery. This is baseline biomarkers. So pretty exciting to see the predictive strength of these tools. So, Andrea, you mentioned zombie cells. I figured someone else would have mentioned it by now, but we're of course interested in what can you do about some of these things. We've heard a lot about senolytics, and we've been thinking a lot about intervening, particularly for age-related muscle loss, which is sarcopenia, as well as this cancer-associated wasting that we see in many different cases of cancer, where you see not just loss of muscle, but you see loss of fat and extreme loss of body weight, right? And this is incredibly predictive of adverse health outcomes in those individuals suffering from cancer. I want to note that oftentimes the muscle field is guilty in pushing everything into the same bucket, whether it's a childhood myopathy or sarcopenia or cachexia or bed rest-induced muscle loss. But cachexia is really this different condition that has this very high inflammatory component that drives catabolism. So I want to mention just one project that we did in this space. We thought a little bit more about which cancers we were going to be interested in and pursuing first. So we did a model of colorectal cancer, and this is just showing you that in response to tumor or tumor plus chemotherapy, the frontline defense for colon cancers is fulfuri, which is a mixture of folinic acid, fluorouracil, and irinotecan. This is a toxic combination that robustly induces many of the markers that I showed you in the human populations. We don't have the same breadth of coverage with this simple platform in mice as we do in humans, but just showing you that the chemotherapy on top of the tumor really drives these levels up. And you can see it even drives some factors that we associate with the SASP up independently of there being a tumor on board. And of course, thinking about the target of action of these chemotherapies, many of them hit the liver incredibly hard. So we've been interested in what's happening in the liver that may be driving systemic consequences in response to these drugs. And what I'm just showing you here is if we take mouse hepatocytes in vitro, no different than if you use etoposide or many of the other chemotherapeutic agents that have been talked about today for inducing senescence in vitro. Fulfuri very nicely can induce senescence. I'm just showing you a snapshot of a couple of the markers here, as well as increasing the production of some of these factors in the media that I showed you previously that are components of the SASP. So Davis England, who is now an assistant professor at the University of Alabama, Birmingham, took on this project, and he treated mice independent of cancer, I need to acknowledge here, with fulfuri, this chemotherapy. And what you can see is it's a really beautiful model of cachexia with over the course of 9 weeks, you see profound decreases in body weight in these animals, a marked reduction in lean mass, a reduction in fat mass, or a failure of fat mass accretion in these animals over the course of time. But what's slightly different than traditional models of cachexia is they did not have anorexia. So their food intake was normal, but you can see significant effects on their physical function if you look at exercise capacity on a treadmill. And then consistent with what many people have shown today, we see markers of senescence markedly increased in the liver, whether you look at P16, P21, or components of the SASP. And then the circulation of these animals, again, with this chemotherapy regimen, we can replicate kind of the circulatory milieu that we described in the human populations with upregulation of many of the cytokines, chemokines, matrix remodeling proteins, and growth factors that I've been hinting at. So also note that, I'm a muscle guy, but we were kind of pushed to think about the liver because the liver is getting exposed to so much of this compound. And I'm just showing you early stages of, we, like everybody else, want to get into the spatial proteomics. We're big fans of the Xenium platform. Before we had access to that, we used Visium. And I'm just showing you two features here of how significantly upregulated the NF-kappa B pathway is in the response to FOLFIRI. So Marco showed beautiful data earlier on targeting this pathway, but we can see significant upregulation of, I can't even read my own screen here, IK-B, as well as P65, P50 looks the same, where we see this really turned up in the liver of these animals. And in response to that, we had the unique opportunity to work with our friends and colleagues just north of Mayo Clinic in Minnesota at the University of Minnesota, Laura Niederhofer and Paul Robbins. And Paul had published a story working with Ted Kamenica at University of Florida Scripps Research Institute using an IKK beta inhibitor to block NF-kappa B signaling in another story. So we asked them if they'd be willing to collaborate on this project to see if there are therapeutic effects of this intervention. And what I'm showing you here is kind of the molecular profile of the liver, and I'm sorry, this is probably quite small font, but again, just showing that the usual suspects associated with the senescence program and the SASP at a transcript level were markedly suppressed in these animals that received this IK-kappa B inhibitor in parallel with their FOLFIRI treatment regimen. And that significant reduction in the liver was reflected by a significant reduction as well in many of these factors that we were most concerned about in the circulation, these biomarkers of senescence. And while that was all fine and good, what we're really interested in, whether there were therapeutic effects at these clinical readouts and endpoints that I've been pushing on you. So again, here you can see, just by suppressing the SASP in the context of this toxic chemotherapy, we had a pretty remarkable maintenance of body weight throughout the treatment period. That's the middle line there on the top left in the green bars. And compared to those animals receiving FOLFIRI alone, who had marked reductions in body weight, lean mass, and fat mass throughout the study period. Our maintenance of lean mass was significant, but it was maybe a little bit more marketable effects on the degradation of fat mass. And then just in the other panels here, I'm just showing you how muscle weights were preserved uniquely. So when we measure lean mass overall, there's other organs that are calculated with those measures, but there was really no meaningful effects on heart weight, kidney weights, or liver weights. And importantly, we kind of prevented this marked reduction in exercise capacity and physical function in these animals as measured by the treadmill test to exhaustion and a grip strength measure as well. And then importantly, I'll just show you at a molecular level, or more of a morphological level, I should say, is that we preserved muscle cross-sectional area. We prevented the central nucleization of myofibers, and that's typically a very robust signal for degeneration, or denervation. So by preventing that, we think we're really improving muscle quality, and we see much less fibrosis in the muscle of these animals as well. And now I'll just confess that I've been selling this to you as a liver-driven process, but even in muscle, we see that the NF-kappa B inhibitor was sufficient to reduce senescent cell markers in skeletal muscle as well. So what's kind of direct and what's indirect remains to be determined, and that's really hard to tease out in vivo, but really quite profound effects. Reductions in P21 in muscle, which we're really quite excited about, as well as reduced DNA damage markers in muscle. So to summarize this, we're really quite excited about biomarkers of senescence as being predictive of major health outcomes in older adults. And we think even today, before we have the perfect senolytic to bring into the clinical setting, or a therapeutic, I should say, in the clinical setting, we think they have utility for decision-making processes. I've kind of showcased a couple examples of senescence biomarkers being predictive of adverse health outcomes in the context of cancer and cancer-related surgery. And then we're really quite excited about understanding how targeting the SASP, of course, can improve kind of these concepts of body composition, but we have to be thoughtful, right? That this first study, we're now doing it with tumor on board, but how is that influencing chemotoxicity, chemoefficacy, intolerance, et cetera? So more work to be done there. And then I think there's a lot of opportunities now for advancing these biomarkers of senescence. I've admitted that we've used a very biased approach to start. We haven't really done the traditional discovery science. A couple minutes. I'll just end with a couple of slides, just kind of sharing what we're doing next. And this has been a really fun approach, bringing our center together and asking individuals is, hey, Sandeep Khosla, you're interested in bone. What would be the bone cell that you'd want to target? Or Paul Tang, who is a heart transplant surgeon, what's the cell that you would want to target that you think might become senescent and you'd want to target and understand its biomarkers in the context of aging, so on and so forth. So now we've expanded this repertoire of cells that we're using. We've done a very nice job of establishing a similar etoposide-induced senescence protocol, so the stressor is the same across the different cell types. And what you can see on the right-hand side is just a very simple transcriptional profile of those different cells when they are senescent. And again, you see this heterogeneity, right? Some are high in P16, some are high in P21, and SAS factors are a little bit all over the place when you go from cell to cell. And we've had the great opportunity to work with a newer company in the proteomics space called Seer. They've been outstanding technical partners. This is not a plug for them. I'm just kind of telling you the facts. But they use these nanoparticles that allow you to enrich for low abundance proteins and also try to address the overwhelming features of high abundance protein when you try to do mass spec analysis of proteomic samples. And we've been doing this on the cells in vitro, and what I'm showing you here are these beautiful heat maps that allow us to go from non-senescent to senescent cell across the 8 different cell types, and you can already see this kind of incredible variability between cells. And really the idea in this reverse engineering experiment is to understand, are there unique markers in the cells that can be targeted therapeutically? And we can learn that from the lysates on the far right-hand side of the screen, right? What are the unique proteins in a senescent versus a non-senescent cell? What's a membrane protein that could potentially be targeted with a cell-based therapy in a senescent hepatocyte versus a senescent endothelial cell? So that's part of it. But the other part is leveraging the media to identify unique biomarkers of senescence. So I can say, you know, Fabrizio, I'm sorry, you have high markers of hepatocyte senescence. Andrea, you have high markers of endothelial cell senescence. That's the idea, right? And as we kind of develop these interventions that may be more targeted, could this be helpful for guiding clinical decision making? And this is a very complex analytical process, right? You're culturing human cells, but they're in bovine serum, so then you have to kind of deal with addressing components of proteins coming from cows, not humans, so you rule those out. But then what we did is we're really interested not just what's in the media, but what in that media can be detected in human samples. And this is very expensive, so we've limited it to 20 young, healthy individuals, 20 older, healthy individuals, 65 plus, and 20 older individuals with at least 4 chronic conditions, all right? Just to kind of get a sense of how these are moving in space. And I won't go into the details here, I just wanted to share this idea with you. But what I'm going to show you here is that how remarkable it is that you know, these cells are producing so many proteins and spitting them out into the media. These are just the human proteins that I'm showing you here, where we see as high as 1,575 proteins being spit out by renal epithelial cells, and as few as 3 proteins in the THP1 cells, which is a little bit of a head scratcher. Nate Bassisti just published a beautiful paper in Nature Aging using this technology, and with irradiation, he sees a lot more proteins, so we have to figure that out. But again, number one, we can show these are in the media of the cells, but then when we look in the human samples run on that same platform, you can see we're getting pretty good capture. It actually surprised me of how many of those proteins are detectable in the human circulation. I'm sure many of them are produced and function locally, not globally, but you can see many of them here are detectable in the human circulation, and as high as 77% of proteins secreted by preadipocytes are detectable, and as low as 61% of proteins secreted by hepatocytes are detectable. So just something to share there. And then it's interesting that when we look across the different cell types, we see a lot of proteins uniquely produced by one cell, fewer proteins being produced by many cells. So, you know, we get to 6 cell types, we're down to 54 proteins out of the 1,300 that we've started with across all cell types. And more details here, and I won't go into it, but it's just kind of an interesting way to look at common and unique features of the secretome within and across cell types as we start to think about biomarkers that may provide us more insights into senescent cell burden in humans, and then be responsive to interventions as we move forward. So with that, thank you very much. I appreciate your attention, being last in the day, and happy to take questions.
Thank you. Session open for questions. One here.
So very beautiful talk, right? So I have 2 questions. So the first question is if you have a look at the post-translational modification in some of these SAS pathways, that could be really a key biomarker. And second, all these studies are retrospective, right? So if you are launching a longitudinal study associated with some intervention in order to validate this signature.
Yeah, so the first question is about post-translational modifications, and that's near and dear to our heart, but we just haven't done it, and we need to find someone to pay for it. So Giovanni, if you could write a check, that would be great.
Of course.
So, but that's such an important question that's so often overlooked, and we don't talk about enough. Number two is kind of getting samples before interventions, and I have to highlight the beautiful clinical trial that Sandeep Khosla recently published in Nature Medicine, going from kind of a mouse study in 2017 to the human trial in 2024, 30 treated and 30 control participants. And, some people could look at that study and say the effects were kind of underwhelming, but I think we should stop getting so hung up on the outcome and look at what we've learned through the process. And he's published 2 subsequent papers to that study showing who the responders were. And that's really based on these biomarkers that would have predicted who was most responsive to that intervention. So we'd love to get engaged in more prospective studies and analyze samples to see how they're changing and moving the needle on our historical panel, but then in our new panel as well.
Thank you. Other questions? Yeah, there's another one there. Yeah.
And it's a great talk. So, general question. Now, of course, going to tissue level, cell type level for senescence mapping. Same effort is done by, for example, biomarker consortium trying to do the same for epigenetic age and other biomarkers of aging. So should we connect more, all these efforts? Because Jim, for example, mentioned before that some intervention seems to, for example, go the other way around in terms of epigenetic age. So a senolytics or senolytic intervention seems to accelerate the epigenetic age clock. And we don't integrate all these tools we have, and now that we go at the tissue level, would it be important? So are you trying to connect with them more and do more work on this?
Yeah, that's really a great recommendation, and I think we're all trying that. You also can see how things are evolving so fast, right? So the clocks are evolving, and I'm showing you an evolving proteomic signature, and you mentioned how the SASP is composed of so many other biologically active components. So yes, we should connect more. I will comment that one thing that's really improved our approach is having Tamir Chandra as part of our team, who's very invested in the epigenetic space. And he's actually been able to predict, as has Riccardo Marioni, predict some of these factors just based on methylation profiles. And some of the predictors, which is really interesting, is that the methylation predictors of the actual protein are stronger predictors of health outcomes than the actual proteins themselves. So we've been talking about that a bit too.
Any other question? Yep.
Here.
Okay.
Hi. Thanks for the talk. I had a question regarding the biomarkers of senescence across tissues. I was wondering if you have explored the dynamics across tissues, the dynamics of senescence.
Well, there's a large effort underway through the SENNET consortium, which Peter Adams nicely articulated and showed some examples of that. This has really just been a cell-based approach for us, which is, not fair and it's not representative of entire tissue, but it's given us this flavor that there is incredible heterogeneity and both again, just at a cell lysate level, we see remarkable differences in the proteomics profile, the cell surface profiles. And we think that could be very informative for again, developing interventions, particularly, right now of going a disease-specific approach as opposed to what was just mentioned earlier of developing an intervention just to live longer, healthier, which would be really tough to move forward with regulatory agencies on today.
Yeah. No more questions. So I have a last one, Nathan. Thank you very much for that. It was really amazing. So, when I did develop a drug in my life, and it was quite complicated. But when I think about senescence, I have headache because, it's very complicated, much more complicated. So, all the effort for what was saying before, the effort to find the right biomarker, because as far as I know, there is not a clinical validated biomarker for the burden of senescence and a therapeutic window for patients, but also for selecting patients. Mm-hmm. Which one? So where do we stand today? And what is your suggestion and challenges that you see for the next design of the clinical trials?
Yeah, well, I think Marco hinted at it, and that's, and Andrea, were, being very open-minded to sharing samples from your trials and your studies for groups with emerging technologies to test them to see, again, how are biomarkers changing in response to the interventions that are being tested? And then also, who is responding to those interventions, and biomarkers help predict that. So I think there's a lot of room for growth and opportunity here, and it's through meetings like this that bring people together to have these conversations and initiate collaborations that's so helpful.
Okay, thank you very much. So, yes. And now there are the closing remarks from the main actors. Thank you.
Okay. I ask Professor Musaro, Professor Dada, and Professor Alimonti to come here.
Shall I start?
Yes.
Yeah, so
Go ahead.
it has been a fantastic day, and I was given the daunting task to summarize it. Pretty sure that I will offend most of the speakers by not having captured in full the impact of their findings. But again, it was really an overwhelming day, and again, quite dense and quite rich in information. So we started the day with Peter Adams, who reminded us that aging is modifiable, and indeed he highlighted the value of repurposing approaches, his approach with an MDM2 inhibitor. I think that was really an opportunity for all of us to realize that cancer studies and cancer research, and therefore cancer drugs, may benefit the aging field and vice versa. And I think this was also one of the highlights that I think that emerged during the day. Vassilis Gorgoulis provided us with some really exciting advancements on his approach in identifying drugs that can highlight the presence of senescent cells. They can also be drugs, they can be senolytics, and I found particularly exciting the potential for the in vivo detection, including in humans. So we are really looking forward to see the results first in mice, as you were saying, and then in humans, perhaps, hopefully. Jesus reminded us about the relevance of the immune system, in particular the involvement of NK cells in clearing senescent cells, different kind of senescent cells. He reported us on his siRNA screens. He identified initially these 76 modulators, and eventually boiling down to SMARCA4, and suggesting this provocative approach that, yes, as we were saying, xenomorphics may be an approach, but also the opposite approach, so super-inducing SASP may be an opportunity to explore, especially in cancer settings, and he provided us with some evidence of efficacy of the value of this approach in ovarian cancers. You may have heard also something about telomeres, but then we have also heard two very short presentations selected by the posters, more of which later on. We heard from Federica Manasca from the Bodega's lab about the relevance of line 1 and its broad impact in immune cells, maybe not also in non-immune cells. And Marco Bollis reminded us about the epigenetic level of aging and cellular senescence, and epigenetic modulators, in particular miR29. I found particularly interesting its impact that it can have on the overall the epigenetic machinery. We heard about the DNA methylases and chromatin demethylases. Raffaella Di Micco reminded us about the complexity of cancers. AML is an heterogeneous disease. It's an elderly disease, but again, by selecting upon therapy-induced senescence, the more senescence-inducing and the less senescence-induces, this provided her to find novel opportunities for interventions, and again, even to reaching the eradication of leukemic conditions by combining therapy-induced senescence, this with immunotherapy, and then highlighting the potential value also of polycomb inhibitors, as we have heard. After lunch, we heard the first presentation was from Jim Kirkland, that standing on his long experience provided us a very broad view of the many opportunities for senolytic interventions. He reminded us that, again, few hundred, really few I think 500,000 cells injected in a mouse were sufficient to induce a senescent syndrome, as I think he called it, and that is really once more reminding us how a relatively small number of cells, which of course are hard to detect because small, and certainly we heard how heterogeneous can be, how again powerful again this cell extrinsic capabilities of senescent cell can have more in general to an entire system. And again, from this historical perspective, he reminded us of his quercetin and dasatinib approach and his very many uses. And again, multiple clinical trials in parallel, as he mentioned, is the really way to move fast rather than sequential ones. Andrea Alimonti, the other co-organizers, his prostate cancer is his turf, and he reminded us that, of course, senescence is initially is a cell intrinsic tumor suppressive mechanism, but later on may be pretty bad, especially again in the context of the therapy-induced senescence. And he reminded us about his work, his insights about the impact of immune suppressors, these PMN MDCs that are recruited in cancer cells, inhibit the functions in clearing immune cells, and updated us on the impact of retinoids on inducing good cells and activate these anti-tumor immune cells. And he concluded his presentation with his very recent, and that was the first time that I heard it, about the work on Nectin-2, and a fully developed story down to the CAR-T development that really was, I think, was quite impressive. Marco Demaria once again reminded us about the difference between a transient, good senescence and a chronic, bad senescence, and told us briefly about the two stories, how to turn necrosis into a senescence, and this has a positive impact on kidney functions. And then this fascinating new story about ATP that is secreted by senescent cells. So it's honestly, I did not know about its role as an alarming molecule, and the fact that it is sustaining a SASP itself through NF-kappa-B, and most importantly, the fact that he showed that it can have efficacy in wild-type animals studied for long terms. I think really this is the golden standard that we as a community should adopt. So transgenic mice, acute settings are convenient, but the hard way is probably proving efficacy in natural settings, meaning wild-type animals, and of course, in the long terms. Clemens Schmidt told us many things, and again, Andrea has a prostate cancer, Clemens has a lymphoma as his area of expertise. He reminded us about cell plasticity, cross-differentiation, cancer senolysis, but possibly the most impressive, certainly the most impressive, at least to me, part of his presentation is this pan-senolytic that was tested. I've never seen such a broad testing for a drug, only maybe Martian-induced senescence was not tested. Clemens, you should consider that everything else was really considered and tested. So that was, again, quite impressive. And again, we then had two short presentations, one from Jesus lab by Maria Antonietta D'Ambrosio, that expanded what Jesus hinted in the morning about the covalent binders and the senolytic search for drugs, for senolytics, and boiling down eventually to GPX4, while Benjamin Lecalvé by Starcage provided, I think, compelling evidence of DPP4 as a senescence target. And then we concluded the day with Nathan Leberser from Mayo Clinic that reminded us about the value of biomarkers and senescence biomarkers, of course, as predictors of functional decline and risk of death and their impact on cancer. And again, quite interesting, the model model of cachexia induced by porphyry, and how the NF-κB inhibitors can improve muscle mass and muscle fitness, and the new proteomic technology that very likely will provide us with more biomarkers. That means more work for all of us. So that was my attempt to synthesize and summarize the day today, but I think now we have some official awards to assign. So there were 4 posters that were selected for the short talks, so that we want to acknowledge now. And so Silvia Bisiti will, yep, exactly. Yep. So
we call Federica Marasca. Stay here. Marco Bolis. Maria Antonietta D'Ambrosio. Marco. E Benjamin Le Calvé. Facciamo qui la foto qui.
So the winner for the best poster go to Simona Fusca from the group of Professor Di Micco for the important work about EML. So I think Simona will also receive an award of 500,000 Swiss francs. No, 500 francs, and they will be invited at dinner, probably. We don't know. Okay. So she go to dinner with a good award.
So this was the end of the official scientific day, but it's not over. As most of you, we are going to have a lecture by Professor Vartelongo that will be in Italian. So for the foreign speakers, there will be a translation. And so we'll reconvene at 6:30 here. Am I correct? I think so. Okay, 6:30 here. See you.
Buonasera a tutti. Mi sentite? Credo di sì. Ben arrivati. Come avrete notato, we switch on Italian perché abbiamo concluso una giornata intensa, ricca di contenuti, di stimoli, e quindi per quelli che sono stati tutto il giorno con noi, adesso passiamo all'italiano per sentire la lecture del professor Longo. Intanto un grazie molto sincero a tutte le persone che ci hanno raggiunto adesso e un complimenti e congratulazioni a quelli che restano, che hanno passato tutto il giorno qui. Abbiamo scelto di passare alla lingua italiana per diversi motivi, ma il principale è che la Fondazione IPSA per la ricerca scientifica ha una vocazione molto dedicata alla divulgazione e quindi dopo una giornata dedicata principalmente a esperti del settore dell'aging, senescenza, eccetera, Questa lecture è più dedicata al fuori dal mondo accademico, quindi al pubblico, quello che siete voi. Quindi intanto grazie, ringrazio di cuore anche i colleghi di IPSA, ce ne sono tantissimi stasera, e quindi IPSA è l'azienda farmaceutica svizzera che, come sapete, nel 2012 ha dato vita alla IPSA Foundation per la ricerca scientifica e la continua a sostenere, quindi la vostra presenza per noi è motivo di orgoglio e di ringraziamento per il supporto costante che ci permette di lavorare con entusiasmo a tutte le nostre attività. Con questo spirito abbiamo deciso, insieme al comitato scientifico del forum, che vi ricordo, il professor Andrea Alimonti, il professor Fabrizio Dadda, il professor Antonio Musarò, del board scientifico della Fondazione, con una lecture d'eccezione. Un grande ricercatore, io adesso non dico altro perché lascio al professor Fabrizio Dadda di darvi il suo profilo scientifico e poi lo ascoltiamo, e intanto io lo ringrazio per essere qui. Buona serata, a dopo.
Buonasera a tutti e benvenuti. Giusto pochissime parole di introduzione al professor Walter Longo, è il professore Edna Jones presso l'Università della California del Sud, è professore di biologia ed è il direttore dell'Istituto di Gerontologia di quell'università, che è uno degli istituti più antichi su questo tema. Come ha detto giustamente Silvia, Walter è eccezionale sotto diversi punti di vista, perché Walter è un scienziato che fa ricerca di base ed è partito da qualcosa che forse molti di voi non sospetterebbero, il lievito. Molti di noi magari lo associano alla birra o alla pizza, ma lui ha iniziato studiando un organismo unicellulare, starvandolo, nel senso affamandolo, e verificando cosa potesse succedere a questo organismo così semplice quando veniva affamato. E così facendo, da questa osservazione che sembrerebbe molto semplice, quasi banale, ha sviluppato una ricerca scientifica fondamentale, scoprendo che i processi che lui aveva scoperto, aveva osservato in lievito, sono conservati evolutivamente fino a noi. E perciò adesso ci sta affamando tutti, e lui sicuramente ci spiegherà meglio di come posso fare io, dimostrando come i pathways molecolari, permettetemi questo termine magari un poco criptico, però diciamo tutti quei meccanismi cellulari che possono controllare la longevità degli organismi sono appunto conservati dagli organismi molto semplici all'uomo. Perciò grazie Walter di essere qui tra di noi.
Grazie Fabrizio e grazie alla Fondazione IPSA. Happy to be here, che sono contento di essere qua. E niente, quindi non parlerò di lieviti, parlerò principalmente di studi clinici e parlerò, per il tempo che ho, di quelli più recenti. Diciamo che la maggior parte degli studi clinici su quello che noi facciamo sono stati fatti negli ultimi 7-8 anni, e quindi mi concentrerò su quello. Intanto parto spesso da questo studio fantastico fatto da qualcuno che era nel mio laboratorio, del laboratorio di Roy Walford da UCLA, prima di me, negli anni 80, che si chiamava Richard Weinberg. E Richard si trasferisce, diventa professore all'Università del Wisconsin e decide di fare uno studio, diciamo, un po' pazzo. Perché questo? Perché hanno studiato le scimmie e quindi la durata della vita nelle scimmie, quindi uno studio intero che è durato 30 anni e è finito penso quest'anno. E quindi queste scimmie, un gruppo di scimmie veniva data una dieta normale, una western diet un po', diciamo, americanizzata, e all'altro gruppo di scimmie veniva data la stessa dieta ma ridotta con un 25% di calorie in meno, nient'altro, quindi diete identiche, 25% di calorie in meno. E vedete intanto gli effetti straordinari per le malattie, quindi quelli che sono in restrizione calorica, solo il 5% sviluppano diabete o insulinoresistenza, quindi diciamo uno stato di prediabete. E per i tumori c'è una riduzione del 50%, quindi metà dei tumori, e malattie cardiovascolari 20-30% in meno, e in questo caso si considera tutta la vita di queste scimmie, quindi 20-30% in meno delle scimmie che mangiavano questa western diet uguale, ma 25% di calorie in più. Però se vedete poi la A e B, la mortalità dovuta alle malattie dell'invecchiamento è chiaramente quelle calorie restriction è la curva in nero, vedete che vivono molto di più di quella in rosso, che è la dieta western normale, però se guardiamo tutte le cause di mortalità, di morte, tutte le cause di morte, vedete che quelle due curve di sopravvivenza sono quasi uguali, no? Quindi cosa succede? Probabilmente succede che ci sono dei trade-off, per usare un termine inglese, cioè c'è un controbilanciamento, ci sono dei problemi creati da questa restrizione calorica e dei vantaggi. Vantaggi e problemi, sì, hai un effetto straordinario sulle malattie dell'invecchiamento, ma poi alla fine vivi quasi uguale, no? Quindi probabilmente, per esempio, il sistema immunitario, la muscolatura, ci sono una serie di problemi che questa restrizione calorica a lungo termine può causare, e probabilmente ha causato. Quindi queste scimmie, però diciamo che la lezione è straordinaria. Che farmaco potrebbe mai ambire a questo tipo di risultato? Penso che sia inesistente, e probabilmente lo rimarrà per tanti anni, cioè riuscire a far questo con un farmaco o anche con un cocktail di farmaci. Ok, quindi Walford, che è il medico pelato sotto, decide di fare su se stesso e altre 7 persone questo studio di restrizione calorica. Quindi ha un altro, diciamo, esperimento da matti, si rinchiude dentro questa biosfera 2, cioè questo è il mio primo anno di dottorato a UCLA a Los Angeles. Walford decide di rinchiudersi dentro questa biosfera 2 che era stata costruita probabilmente per come futura base lunare, no? Quindi avevano costruito questa base nel deserto pensando poi alla Luna o a Marte. Però poi Walford ha visto bene di entrare dentro, fare questo studio umano, erano sigillati dentro per 2 anni, e poi, siccome non riuscivano a far crescere abbastanza cibo per usufruirne, Walford ha detto: perché non facciamo uno studio sulla restrizione calorica? E quindi ha messo tutti a restrizione calorica, queste 7 persone. Allora, io poi sono andato, quando sono usciti, dopo 2 anni, e sono stato impressionato da come, dallo pessimo stato apparentemente di salute che avevano. Anche se poi, se andiamo a vedere ad esempio sotto la pressione sistolica, vedete che entrano, la curva inizia quando entrano con una pressione sistolica di 110, scendono a 90, entrano con una glicemia di 90, scendono a 70, e entrano con un colesterolo di 185 e scendono durante la restrizione calorica a 125 colesterolo totale. Quindi di nuovo fantastico, molto simile agli effetti delle scimmie. Quindi probabilmente queste sono persone che non svilupperebbero mai diabete, non svilupperebbero mai, cioè non mai, ma sicuramente avrebbero probabilmente una riduzione delle malattie cardiovascolari e probabilmente anche dei tumori. Però questo è lo stato di Walford, come vi ho detto quando è uscito. Sulla sinistra questo è Walford, quindi ovviamente spinto al limite, se non oltre al limite. Infatti Walford morirà nel 2004, mi pare, relativamente ancora giovane, mi pare 74 anni, di SLA, e quindi probabilmente o la restrizione calorica o altre condizioni in questa biosfera 2 lo spingono oltre al limite, e diciamo, per me queste sono state tutte lezioni, perché ovviamente da quel momento in poi il mio obiettivo è diventato: com'è possibile mantenere tutti questi effetti della restrizione calorica senza tutti questi problemi della restrizione calorica. E il problema principale è proprio la compliance, cioè chi è che riesce a fare una dieta che ti porta, adesso la maggior parte di voi non saranno necessariamente familiar con il termine BMI, Body Mass Index, però il Body Mass Index in queste persone era molto basso, quindi per un uomo 19, che sarebbe meno circa 20 kg, no? Quindi immaginatevi quanto, non dovete immaginarlo perché lo state guardando Walford all'uscita, e poi Walford invece un anno e mezzo dopo l'uscita dalla biosfera 2, quindi Walford comunque era una persona che aveva una dieta ideale, eppure guardate la differenza. Quindi insomma la restrizione calorica e risultati fantastici, e penso che la maggior parte di chi lavora sull'invecchiamento è d'accordo che probabilmente è l'intervento storico che ha avuto più successo di tutti per adesso sull'invecchiamento, però viene con un costo enorme, e ovviamente poi addirittura poi un gruppo al National Institute of Aging ha rifatto l'esperimento su un altro set di scimmie, non vedendo nessun effetto sulla longevità, anche se in questo secondo studio, sempre sulle scimmie, la dieta di controllo, cioè non confrontavano più il 25% in meno con una dieta western, quindi una dieta diciamo occidentale, ma una dieta un po' più sana, e quindi in confronto a una dieta più sana questa restrizione calorica non serviva più a molto, serviva sempre a ridurre il tasso di cancro, ma non molto altro. Ok, quindi un'osservazione, questo torniamo indietro, a parte i lieviti, quindi sono partito dai batteri in effetti, quindi mettevamo i batteri a digiuno e vivevano molto di più, mettevamo i lieviti a digiuno, e vivevano molto di più, anche se tutti quelli con cui parlavo pensavano che questa fosse una cosa diciamo non molto interessante, per me mi è sempre interessata molto, e poi eventualmente ho iniziato a capire che comunque c'erano tante specie che digiunavano non solo per qualche giorno, ma che potevano digiunare, ad esempio questi pinguini imperatori del Polo Sud, che digiunano per mesi, e digiunano per mesi per covare, i maschi tra l'altro, per covare le uova. E quindi questo mi ha iniziato a far pensare: è possibile che invece di fare queste restrizioni caloriche fare dei brevi periodi di digiuno, e infatti siamo partiti con questo. Vedevamo che nei topi brevi periodi di digiuno di 2 o 3 giorni, cioè solo acqua, rendevano i topi più resistenti alla chemioterapia. E quindi siamo passati da questo a uno studio clinico a USC Norris Cancer Center già 15 anni fa, con digiuno ad acqua per i pazienti che avevano vari tipi di tumore e che ricevevano chemioterapia. L'idea era la protezione del paziente dagli effetti collaterali della chemioterapia. Oggi abbiamo sentito parlare tanto di questo con gli agenti che intervengono sulla senescenza, sulle cellule senescenti, diciamo, probabilmente parte degli effetti che vedevamo anche nei topi sono dovuti proprio alla modulazione delle cellule senescenti. E comunque l'abbiamo portato questo agli uomini per capire che nessuno voleva fare il digiuno d'acqua, quindi il paziente oncologico ha praticamente detto: questo non è una cosa che vogliamo fare. L'oncologo non era entusiasta di farlo fare ai pazienti, e il paziente si sentiva un po' truffato, diciamo, dal dover fare digiuno d'acqua. Un paziente col cancro che riceve la chemioterapia. Questa è una cosa che ci ha molto sorpreso, perché noi eravamo convinti che il paziente oncologico sarebbe stato molto motivato, ma in effetti ci siamo resi conto, almeno a Los Angeles, che questo non era fattibile. Quindi la dieta mime a digiuno parte proprio da questo fallimento, di questo trial di 15 anni fa, e quindi noi poi ci rivolgiamo al National Cancer Institute, al National Institute on Aging, e entrambi ci danno fondi per sviluppare la dieta mime a digiuno, che ha lo scopo di, adesso senza entrare nei dettagli, di cambiare questi 4 marcatori nel sangue nello stesso modo che viene fatto dal digiuno GF1 con sola acqua, quindi GF1, GFPP1, corpi chetonici e glicemia. Questi sono i 4 marcatori che cambiano in maniera molto, cioè gli zuccheri si abbassano, questo growth factor, fattore di crescita, si abbassa, e questi altri 2 marcatori, i corpi chetonici e il GFBP1, si alzano, no? Quindi questa dieta che cos'è? È una dieta con basse calorie, basse proteine, bassi zuccheri, alti grassi, e poi al tempo mi era venuto in mente di farla diventare una dieta composta solo da ingredienti vegani e composta solo da ingredienti vegani molto comuni nelle zone di longevità record, no? Questo non era necessario, l'avrei potuto fare anche con ingredienti non vegani e non di longevità, non di zone con longevità record, ma probabilmente è stata una buona idea partire già con la dieta ideale, che era anche mima digiuno, cioè una dieta sanissima ma mima digiuno. Ok, quindi uno degli studi che mi piace sempre presentare è questo, cioè che abbiamo sempre voluto fare, finalmente qualche anno fa l'abbiamo pubblicato, ed è cosa succede se diamo a un topo, a tanti topi, una dieta western, un po' come quella delle scimmie controllo, e gli diamo da mangiare quanto vogliono, no? Quindi più o meno, adesso se consideriamo che il 60% degli europei sono sovrappeso obesi e il 75% degli americani sono sovrappeso obesi, questo tipo di dieta è abbastanza comune nella popolazione mondiale e non solo di americani e europei. Quindi cosa succede? Succede che ovviamente questi topi diventano obesi. Poi abbiamo dato la stessa dieta a un altro gruppo di topi, dandogli però la dieta minima digiuno per 5 giorni al mese, no? Quindi tutti e 2 questi gruppi avevano questa dieta tremenda, ma uno dei 2 gruppi per 5 giorni al mese andava alla dieta minima digiuno per poi tornare alla questa dieta western. E i risultati sono ovvi, cioè quel topo obeso è quello che mangia questa dieta, però siamo rimasti sorpresi dal fatto che ci aspettavamo un miglioramento del peso, non ci aspettavamo che il peso tornasse al normale, a quello dei topi che hanno una dieta healthy, una dieta sana. E la cosa anche interessante era che non solo il grasso rimaneva normale, ma che la massa muscolare e la massa ossea rimanevano normali, e quindi non c'era una prevenzione dell'accumulo di grassi, ma anche una riduzione di massa muscolare, quindi c'era solo un effetto sul grasso e non sulla massa muscolare. Ok. Ovvio che questi interventi, come per la restrizione calorica, devono agire in maniera multisistemica, quindi l'idea non era di abbassare il grasso, ma era di rendere il topo più giovane in un certo senso, nonostante questa dieta, 25 giorni al mese di questa dieta molto negativa. E infatti, se vedete anche il colesterolo, il colesterolo aumenta, quella dieta HFCD e la dieta high fat, alti livelli di grassi, alti livelli di calorie e di zuccheri. Vedete che il colesterolo nei topi, mentre i topi invecchiano, aumenta, e poi eventualmente nei topi vecchi aumenta tantissimo quella barra molto alta. Di nuovo, i 5 giorni al mese di dieta minima digiuno sono in grado di riportare il colesterolo al normale o comunque vicino al normale. E poi infine la sopravvivenza, che è poi la cosa più importante. E quindi vedete, intanto è incredibile l'effetto della dieta western sulla longevità, no? Quindi la dieta western, vedete in viola, riesce quasi a dimezzare la vita di questi topi, no? E solo 5 giorni al mese di dieta minima digiuno non riportano la sopravvivenza al normale, ma molto vicina al normale. Ok, quindi eventualmente poi siamo passati agli studi clinici umani, e il primo studio l'abbiamo fatto proprio a USC su pazienti che andavano dai 20 a 70 anni, e l'idea era proprio quella di dargli questi cicli di 5 giorni di dieta minima digiuno, che la dieta minima digiuno che contiene tra le 800 e le 1100 kilocalorie al giorno, Quindi a questi pazienti veniva dato una scatola essenzialmente, e questa scatola era tutto quello che mangiavano una volta al mese, e poi potevano tornare a esattamente quello che facevano prima dell'intervento. Questi cicli, quindi le misurazioni venivano fatte all'inizio, prima dell'inizio dei 3 cicli della dieta mia a digiuno, e poi una settimana dopo la fine del terzo ciclo. Intanto il peso corporeo diminuisce il grasso addominale, proprio come nei topi che vi ho appena mostrato, la circonferenza addominale, e proprio come nei topi lean body mass, quindi la massa muscolare, massa ossea, non diminuisce, addirittura quella relativa aumenta, però quella assoluta rimane uguale, quindi mantengono la massa muscolare pur perdendo la massa grassa. Prima vi ho fatto vedere gli effetti della restrizione calorica, quindi anche chi iniziava con dei livelli di colesterolo, glicemia, eccetera, pressione sanguigna bassi, scendeva, e questo probabilmente ha contribuito ai problemi di Walford, cioè parti da una pressione sanguigna di 110 e sano scendere a 90? Probabilmente no. E quindi adesso torno indietro. E quindi la differenza qui con nella dieta, nei cicli di dieta mia a digiuno, è che per esempio la glicemia, quelli che partono con una glicemia normale rimangono a quella glicemia. Ricordate che i pazienti col diabete partivano i pazienti nello studio di restrizione calorica partivano da una glicemia normale ma scendevano a glicemia 65-70. In questo caso non lo vediamo, non so perché sta andando avanti da solo. Comunque, morale, tutti i marcatori nelle persone in cui i marcatori sono normali, quindi pressione sanguigna, glicemia e colesterolo, se partono con livelli normali rimangono lì, se partono con livelli elevati scendono, e scendono spesso, per esempio per la glicemia spesso scendono nel range normale, quindi ritornano a un range di normalità. Ok, poi il secondo studio invece è stato un confronto con la dieta mediterranea. Cosa succede se un gruppo di pazienti del Tennessee, a questo gruppo di pazienti del Tennessee che partono già da una situazione di sovrappeso e obesità, vengono assegnati o a 4 mesi di dieta mediterranea o a 4 cicli di dieta a digiuno di 5 giorni. Quindi insomma 5 giorni al mese contro 30 giorni, cioè tutto il mese in dieta mediterranea. E intanto tutte e due funzionano. Effettivamente la dieta mediterranea riesce a far perdere peso a questo gruppo, anche se la dieta a digiuno, i soli 5 giorni hanno un effetto superiore sul grasso, diciamo principalmente il grasso addominale, quindi agiscono più specificamente sulle riserve di grasso addominale. Però iniziamo a vedere già delle differenze, quindi mentre la leptina scende drasticamente in tutti e due i gruppi, sia la dieta mediterranea che i 5 giorni di dieta a digiuno, l'adiponectin, che è un sensibilizzatore dell'insulina, quindi fa funzionare l'insulina meglio, e nella dieta mediterranea c'è in effetti una trend, cioè non è significativo, ma una trend, una riduzione potenziale dei livelli di questo sensibilizzatore dell'insulina, mentre nella mia digiuno, adesso ricordatevi che questo è stato misurato una settimana dopo che hanno smesso di fare la mia digiuno, quindi questi effetti sull'insulina sono long lasting. Cosa significa? Durano anche dopo che i pazienti in questo caso sono tornati a mangiare quello che hanno sempre mangiato, che li ha resi sovrappeso, obesi, e gli ha dato una serie di condizioni. Ok, l'altra cosa molto interessante che non ci aspettavamo è questo: dopo 4 mesi di dieta mediterranea questi pazienti del Tennessee perdono 3 libbre, cioè quindi circa 1,5 kg di massa magra, di muscoli principalmente, e dopo, questa sta andando avanti da sola comunque, e dopo 7 mesi ne hanno persi circa 2,5 kg, comunque 2,5 kg di massa magra, e di nuovo nella mima digiuno non vediamo questi effetti. Quindi questi interventi brevi riescono a riprogrammare metabolicamente questi pazienti, però senza ad andare a intaccare la massa muscolare, la massa ossea. Ok, e qui iniziamo a vedere, diciamo a preparare adesso quello che faccio vedere sui pazienti diabetici, iniziamo a vedere gli effetti differenziati in base di nuovo allo stadio di partenza, cioè chi parte con una glicemia normale rimane lì, chi parte prediabetico, di nuovo qua, chi parte prediabetico scende, l'emoglobina glicata in questo gruppo scende, ma chi parte diabetico scende di molto, no? Quindi effetti che sono simili a quelli dei farmaci per il diabete, tra l'altro. E questo ha motivato diverse università, ma prima di tutto Heidelberg, e quindi un gruppo di Heidelberg decide di fare uno studio sui pazienti diabetici, quindi li randomizza e li randomizza o a una dieta mediterranea. Probabilmente la loro ipotesi era che bastava una dieta sana per fare quello che fa la dieta mia digiuno, e quindi li randomizzano a una dieta mediterranea, oppure 5 giorni di dieta mediterranea, oppure 5 giorni di dieta mia digiuno, e vedete in cima a destra che la dieta mediterranea non ha nessun effetto né sull'emoglobina glicata né sull'insulino resistenza, lì OMA-ER l'insulino resistenza, e invece i 6 cicli, in questo caso sono 6 cicli di dieta mia digiuno una volta al mese per 6 mesi in questo gruppo di pazienti diabetici. 6 cicli di dieta mia digiuno riescono a causare una grossa diminuzione dell'insulino resistenza. La cosa più importante è l'effetto sui farmaci, quindi le 6 cicli di dieta mia digiuno, mentre la dieta mediterranea, nel gruppo di dieta mediterranea, il 20% dei pazienti aumentano l'uso di farmaci durante questi 6 mesi, il 50% non hanno nessun cambiamento, il 30% riducono i farmaci. Nella dieta mia digiuno nessuno aumenta l'uso di farmaci, il 70% riduce l'uso di farmaci. La cosa ancora più importante, se pensiamo, diciamo, quello che secondo noi dovrebbe essere il futuro degli interventi di medicina, cioè di ringiovanire il paziente. Oggi tutta la giornata è stata basata proprio su quello, e quindi vediamo l'effetto anche sull'ipertensione. Quindi anche i pazienti che prendono farmaci per l'ipertensione, nella dieta mediterranea vediamo 10% aumentano l'uso di questi farmaci durante i 6 mesi, e una piccola percentuale li diminuiscono. Nella mia digiuno nessun aumento, e una percentuale di 2-3 volte più grande riduce anche gli antiipertensivi. Quindi insomma effetti sistemici che vanno a toccare, come abbiamo visto, la restrizione calorica, il colesterolo, la pressione sanguigna, la glicemia, probabilmente vanno a toccare proprio la funzione sistemica. Adesso Leiden in Olanda decide di fare uno studio più lungo, più ambizioso. Allora vanno a 12 cicli di mia digiuno, il professor Hanno del Dipartimento di Endocrinologia all'Università di Leiden, 12 cicli una volta al mese, di nuovo pazienti diabetici. Questo è uno studio più grosso di 100 pazienti, e quindi in questo caso sono pazienti diabetici, ma selezionano solo quelli che usano metformina e non altri farmaci. Quindi sono pazienti diabetici che usano la metformina. E di nuovo la percentuale dei pazienti che riduce l'emoglobina glicata nel gruppo che prende solo la metformina è di 3 volte inferiore a quella al gruppo che prende metformina insieme ai 12 cicli di mia digiuno. Adesso non tutti hanno finito tutti i 12 cicli, però diciamo che la maggior parte ha finito la maggior parte dei cicli. Quindi un forte miglioramento contro i farmaci da soli, e poi 8 volte una differenza di 8 volte nella riduzione dell'uso di farmaci. Quindi chi faceva i 12 cicli di mia digiuno dopo un anno era 8 volte più likely di aver ridotto proprio l'uso di metformina, o smesso la metformina o ridotto la dose della metformina. Ok, questo è solo per far vedere che adesso questa cosa è stata testata in 5 studi clinici diversi, i risultati sono molto consistenti e quindi iniziamo a vedere dati consistenti senza il bisogno, come nel caso della restrizione calorica, di cambiare stile di vita, ma solo potenzialmente. Adesso infatti abbiamo appena finito, stiamo per finire uno studio di 500 pazienti in Calabria, abbiamo scelto una delle regioni più problematiche d'Italia e in Calabria siamo partiti con persone sovrappeso, obese, con una serie di problemi come ipercolesterolemia, la glicemia alta, ipertensione, eccetera, eccetera. Adesso stiamo per completare questo studio, però questa volta una cosa più ambiziosa. Cosa succede se facciamo la mima digiuno una volta ogni 3 mesi? Adesso vedremo, non lo sappiamo ancora, però ci stiamo spostando verso una modalità, diciamo, basata su una dieta vegana che però non pretende un cambiamento di stile di vita. Quindi queste persone, la maggior parte, anzi la stragrande maggioranza di queste persone in Calabria hanno finito i 3 cicli di dieta mia digiuno senza problemi, quindi insomma è molto fattibile. Adesso vedremo se riesce a avere questo tipo di impatto o meno. Ok, quindi però quali sono i meccanismi? Perché funziona su così tante cose? Quindi una delle possibilità adesso di cui si parla tanto, e secondo me è la cosa forse più entusiasmante, una delle cose dei subfields del cambio di invecchiamento più entusiasmante, la riprogrammazione cellulare, quindi la riprogrammazione epigenetica. Quindi è possibile riportare le cellule a essere non solo più giovani, ma giovani, attivando questi fattori di riprogrammazione, i famosi Yamanaka factors. Quindi già un po' di anni fa avevamo fatto uno studio su questi topi che sono geneticamente condannati ad avere il diabete, quindi sono DBDB mice, hanno delle mutazioni nei recettori della leptina e quindi hanno sempre fame, mangiano sempre, diventano obesi e poi sviluppano, come vedete sulla destra, iperglicemia e poi muoiono di questa iperglicemia molto rapidamente. Intanto, se partiamo in questi topi, diciamo, condannati geneticamente ad avere il diabete, ad essere obesi, avere il diabete, se partiamo, quando siamo partiti con la dieta a digiuno, vediamo una riduzione, un blocco della mortalità e una forte riduzione dell'iperglicemia, della glicemia. E questo già, insomma, era importante, ma ce l'aspettavamo in un certo senso. Quello che non ci aspettavamo è il modello di diabete tipo 1. Quindi quando siamo andati a vedere il diabete tipo 1 sono le cellule del pancreas, delle beta cells che non funzionano più e quindi siamo andati a vedere cosa succedeva in un modello, questo streptosotosi, quindi danneggiamo il pancreas in maniera irreversibile e poi questi topi diventano praticamente come dei topi di diabete tipo 1, non riescono più a produrre l'insulina. E l'insulina qui la vedete con quel colore rosso, queste sono islet, cellule del pancreas, il rosso indica produzione di insulina. Dopo, quando dice STZ-BL-D5, quello è il 5° giorno dopo l'uso di questa tossina che distrugge le cellule del pancreas. Quindi vedete che non ci sono più cellule rosse e D50 è 50 giorni dopo. Le beta cell del pancreas vengono distrutte e vengono distrutte in maniera permanente, quindi sono permanentemente topi di diabete tipo 1. Se però iniziamo la dieta a digiuno e la facciamo 5 cicli e poi ci fermiamo dopo 30 giorni, dopo 50 giorni vedete in fondo che il pancreas è ripopolato con beta cells che producono insulina, quindi ritornano a produrre insulina in maniera normale e questo poi è un effetto permanente. Infatti la mima digiuno viene bloccata al giorno 30 e questa misurazione viene fatta 20 giorni dopo e ancora e poi comunque a lungo termine questi topi producono normali livelli di insulina. Quindi cosa succede? Quello che succede è proprio questo, il pancreas viene riprogrammato, quindi una serie di geni, inclusi diversi Yamanaka factors, questi fattori di riprogrammazione vengono attivati, ma il programma è ben più complesso degli Yamanaka factors, ce ne sono decine di questi geni e se vedete sulla sinistra quella linea verde è il topo a cui viene data una dieta normale, quindi verde significa che questi fattori di riprogrammazione sono spenti, non sono attivi. Poi al centro vedete FMD, quello è alla fine dei 4 giorni, mi pare in questo caso, di dieta mio a digiuno. Tutti questi riprogrammi, infatti questi fattori di riprogrammazione sono attivati, quindi il pancreas si sta preparando a riprogrammarsi, o si sta riprogrammando in effetti. E poi RF1D è un giorno di rifeeding, un giorno dove essere tornati alla dieta normale, e vedete che molti di questi geni continuano a essere rossi, significa che sono altamente espressi. Quindi il pancreas si riprogramma, cioè capisce durante il digiuno che c'è un problema, riprogramma, attiva questi Yamanaka factors, attiva tutti questi geni della riprogrammazione e poi inizia il processo di riportare il pancreas al normale, no? E quindi diciamo che se pensiamo alla restrizione calorica ci rendiamo conto che cosa manca nella restrizione calorica. Qual è la cosa più importante? È proprio il refeeding, cioè non è tanto la restrizione calorica. La restrizione calorica inizia il processo, poi lo conclude quando torni, cioè l'IGF-1 stranamente, che è molto basso, questi fattori di crescita che sono molto bassi durante la restrizione calorica e durante il digiuno, hanno bisogno di rialzarsi magari anche a dei livelli alti per poi ricostruire quello che è stato danneggiato. Quindi la restrizione calorica in un certo senso fa solo la prima parte, ma gli manca la ricostruzione, è per quello che potenzialmente ha tutti questi problemi. Ok, quindi perché funziona questa mimia digiuno? Per tante ragioni, ma adesso senza andare nei dettagli perché molti di voi non hanno un background di biologia, però diciamo che agisce sull'insulina, sulla sensibilizzazione dell'insulina, sull'obesità. Tra l'altro gli effetti sull'insulino resistenza nei diabetici non è associata, cioè non è correlata alla perdita di peso, perché molti hanno detto ah sì vabbè, perdono peso, è ovvio che il diabete, che diminuiscono l'uso di farmaci e l'insulino resistenza diminuisce. Non è così, non c'è correlazione, quindi non è che chi ha più perso peso beneficia di più, ma sono proprio i meccanismi di riprogrammazione e meccanismi anche di riprogrammazione metabolica, quindi l'insulino resistenza non necessariamente è influenzata da questi Yamanaka factors, ma una riprogrammazione che passa da uno stato di accumulo insulin dependent, accumulo di grassi, a uno stato di utilizzazione del grasso che è stato messo da parte, no? Quindi pensiamo a estate-inverno, estate nel nostro passato 10.000 anni fa, abbondanza di cibo, stessa cosa per i pinguini del Polo Sud, abbondanza di cibo, e quindi si mangia di più del normale, soprattutto frutta e altri high fructose, quindi cibo con alto fruttosio. Questo porta all'obesità, poi eventualmente arriva l'inverno o comunque il periodo dove il cibo non c'è più, e questo periodo di digiuno poi sblocca questa riprogrammazione e porta alla perdita di peso. Quindi da un lato è quello, dall'altro lato una cosa abbastanza sorprendente per la maggior parte delle persone, perché tutti pensano che mangiando tante proteine si perde peso. In effetti quello che noi stiamo facendo vedere, stiamo anche per pubblicare, che è esattamente il contrario, cioè le proteine, soprattutto certi aminoacidi contenuti nelle proteine animali, di origine animali, attivano queste vie di segnalazione, questo axis growth hormone IGF-1, che sembra essere centrale per il blocco dell'insulino resistenza. Quindi finché le proteine sono alte, questi fattori di crescita sono alti, non si riesce a sbloccare l'insulino resistenza, si rimane bloccati in quella fase di accumulo di grasso, diciamo. Poi la rigenerazione del beta cells, questo però l'abbiamo solo dimostrato nei topi, però c'è molto interesse ovviamente a capire, è possibile nelle persone con diabete tipo 1 intervenire. Adesso avevamo iniziato uno studio al Gaslini di Genova, però con bambini, però questo studio sta andando in maniera molto lenta per ovvie ragioni, quindi questo studio non è ancora stato fatto sugli adulti. Ok, poi recentemente abbiamo fatto un follow up in collaborazione con Laura Perrin del Los Angeles Children's Hospital, andando a vedere se questi effetti che avevamo già adesso dimostrato per l'intestino, per il fegato, muscoli eccetera eccetera, se questo succedeva e come succedeva anche nei reni. Quindi un modello, questo puromycin dependent tossina, nefrotossina, quindi abbiamo usato nei ratti questa tossina che distrugge la funzionalità renale. E quindi nella parte di sotto c'è quello che si chiama spatial transcriptomics, quindi andiamo a vedere in maniera spatial, quindi l'architettura dell'espressione genica, quindi andiamo a vedere i diversi tipi di cellule e i diversi tipi di geni che vengono espressi, come sono influenzati da questa tossina, e vedete che al centro questi sono i controlli, quindi i topi che hanno un rene normale, questo rappresenta un rene tagliato, no? un rene sezionato, quindi normale, poi gli viene data la tossina, vedete che tutta questa espressione genica viene distrutta, no? E in questo stato centrale il rene non funziona, il ratto non funziona più. Poi da questo stato qua, che è diciamo un danno a lungo termine, iniziamo i cicli di dieta mia digiuno, 6 cicli di dieta mia digiuno, e vedete che dopo i 6 cicli questa architettura dell'espressione genica ritorna al normale, quindi il rene sa come ritornare durante questi cicli di digiuno e ritorno alla dieta normale, fatto 6 volte, quindi digiuno, dieta, ritorna, sa esattamente dove ritornare, no? Quindi ha un programma, cioè un programma di riparazione molto sofisticato, tra l'altro, perché vedete ogni cellula è ritornata al suo posto e esprime i geni giusti, no? Quindi, e infatti questi topi, questi ratti, non topi, tornano a una funzionalità del rene normale dopo i 6 cicli, mentre ovviamente i controlli sono ancora non in grado di usare in maniera normale il rene. Poi di nuovo Yamanaka factors adesso cambiano, in questo caso il MYC viene espresso altamente, però comunque vediamo di nuovo questi fattori di riprogrammazione elevati alla fine della dieta, la seconda colonna, ma anche dopo 24 ore e molti ancora dopo 72 ore. Quindi questo programma di riparazione multisistemico continua a funzionare per potenzialmente, ad esempio nel pancreas avevamo visto addirittura settimane dopo continuavano almeno parte di questa riprogrammazione che portava alla rigenerazione delle parti danneggiate del pancreas. E stessa cosa qua. Poi i nostri colleghi Alessandro Laviano, Silvia Lai dell'Università di Roma, della Sapienza, hanno fatto uno studio, diciamo, pilota, però su 13 pazienti, quello che si chiama randomized crossover, quindi prima 7 pazienti fanno 3 cicli della dieta mia digiuno, questi sono pazienti con malattie renali croniche, quindi questi pazienti con malattie renali croniche fanno 3 cicli della mia digiuno, contro 6 pazienti che sono di controllo. Poi i 6 pazienti fanno anche loro, si chiama randomized crossover, quindi anche loro seguono 3 cicli di dieta mia digiuno. Intanto vedete che la proteinuria, quindi uno dei marcatori del danno ai reni, la proteinuria dopo i 3 cicli di mia digiuno scende quasi ai valori normali in questi 13 pazienti. Ma la cosa più interessante è che un anno dopo 12 mesi, dove ci aspettavamo tutti che sarebbero ritornati ad avere una proteinuria elevata e quindi disfunzione renale, rimane bassa. Quindi questo effetto succede rapidamente in qualche mese e un anno dopo continua a essere presente. E se vedete la cosa interessante, le progenitor cells, quindi queste cellule diciamo semistaminali, sono in circolazione, queste progenitor cell che danno vita proprio alle cellule renali, ai podociti del rene, triplicano in circolo. Quindi non sappiamo, adesso mentre nei ratti possiamo dimostrare cosa sta succedendo e nei topi, in questo caso non lo sappiamo, è una correlazione, un'associazione. Quindi vediamo che queste cellule progenitor cells triplicano e queste progenitor cells però potrebbero essere in teoria responsabili per questi effetti a lungo termine, quindi potrebbe esserci sia una riprogrammazione delle cellule del rene come abbiamo visto nei ratti e nei topi, ma anche potenzialmente un recruitment delle cellule staminali da altre, queste sono in circolo, quindi potenzialmente partivano dal rene e poi vanno in circolo, ma potenzialmente partivano da qualche altra parte del corpo e andavano ai reni per riparare il danno renale. Ok, quindi l'altro studio di quest'anno, qualche mese fa, e questo in collaborazione con Tor Vergata, quindi il professor Alessandrini e Micharelli, e con loro dopo Covid osservavamo che molti pazienti dicevano: beh, avevo perso l'olfatto, avevo perso il gusto, poi ho fatto il ciclo di mio digiuno e mi è tornato normale. Quindi abbiamo detto: ma è coincidenze potenzialmente. Però abbiamo deciso di fare uno studio, tra l'altro grande, 100 pazienti, randomized crossover, quindi 50 e 50, poi 50 di nuovo nel controllo, fanno anche loro, in questo caso 6 cicli di dieta mio digiuno, e quindi i risultati molto consistenti, soprattutto per quanto riguarda l'olfatto. Circa oltre il 30% dei pazienti, questi erano pazienti sovrappeso o obesi, oltre il 30% avevano difetti, quindi quello che si chiama iposmia, difetti nell'olfatto, e sia nel gruppo iniziale di 6 cicli di mio digiuno, sia nel secondo gruppo, quindi i controlli che poi dopo 6 mesi iniziano a fare 6 cicli di mio digiuno, si vedono effetti quasi uguali. Quindi l'iposmia, l'inabilità di avere un olfatto normale, quindi un olfatto disfunzionale, questi pazienti tornano al normale, quasi tutti. E l'altra cosa interessante è che quelli tra di loro che erano diabetici, come abbiamo visto prima, circa oltre il 60% diminuiscono l'uso di farmaci in tutti e due i gruppi, sia il gruppo originale che il gruppo di crossover. E di nuovo l'uso di farmaci per l'ipertensione viene ridotto in tutti e due i gruppi, in oltre il 20% dei pazienti in tutti e due i gruppi. Quindi iposmia, glicemia, ipertensione, di nuovo l'effetto è chiaramente multisistemico, non sta agendo su un particolare target, ma sta agendo su tanti tipi di obiettivi. Ok, anche qua vediamo addirittura in questo caso i pazienti che fanno 6 cicli nel controllo perdono 9 kg di massa grassa, e addirittura questo studio faceva vedere aumento di massa magra. Probabilmente non è vero, perché è stata usata l'impedenziometria e quindi non DEXA, quindi non raggi X, ma impedenziometria, quindi probabilmente un dato non vero. Quello che è vero però sono i 9 kg, questo lo sappiamo, di perdita di grasso. Quello che probabilmente non è vero è addirittura questo studio faceva vedere 5-6 kg di aumento di massa magra, che probabilmente è esagerato, però se fosse anche che massa muscolare è rimasta uguale, questo comunque già è un grande traguardo. Ok, Miami ha appena pubblicato sulla colite ulcerosa, quindi questo uno studio randomizzato dove confrontavano in 32 pazienti o i farmaci da solo, questi sono JAK inhibitors della Pfizer, o i farmaci da solo o i farmaci più la dieta mio digiuno, e in questo caso hanno fatto solo 2 cicli di dieta mio digiuno, quindi anche noi non pensavamo che questo sarebbe stato sufficiente per fare la differenza. Ma questi pazienti con colite ulcerosa dal moderato al severo, in effetti la risposta va dal 35% nei pazienti, la risposta clinica, cioè quelli che migliorano, sono il 35% di quelli che ricevono solo il farmaco, e va al 57% in quelli che ricevono farmaco più dieta mio digiuno. Non è significativo perché il gruppo era piccolo, però diventa significativo quando qualsiasi aumento di diminuzione dell'attività clinica, quindi dei sintomi, viene considerata. Allora in questo caso vediamo già la mio digiuno che è significativamente migliore che il farmaco da solo, quindi la mio digiuno aiuta questi pazienti a fare meglio che il farmaco da solo. E lo vediamo con vari tipi di test. Senza andare nei dettagli, comunque è molto consistente l'effetto in tutto quello che hanno guardato. E particolarmente interessante sono i marcatori nel sangue, no? Quindi poi noi siamo anche adesso dopo questa giornata, devo dire che ho iniziato a pensare potrebbero essere parte di questi effetti dovuti alle cellule senescenti, che sono molto interessanti andare a vedere. Tra l'altro diciamo siamo molto interessati a collaborazioni, soprattutto col sangue dei pazienti, e vedete nel gruppo di controllo controllo che sia la proteina C reattiva, che sia la calprotectin, che è un marcatore della leakiness del gut, che sia l'amiloid alfa, che sia l'uso di steroidi, non cambia niente. Vede, la control diet, sì, è un gruppo piccolo questo, diciamo, però non cambia niente. Invece dopo i 2 cicli solo di mio digiuno vedete riduzione della proteina C reattiva, anche se è solo una trend, però forte riduzione della calprotectin, quindi da 16.000 va a 847, amiloid A da 2.200 a 1.500, e l'uso di prednisone da 78% a 22%. Quindi di nuovo vediamo questi pazienti che dipendono, sono più sani, diciamo, e dipendono meno dai farmaci. Adesso Stanford ha finito e sottomesso uno studio sulla Crohn's disease, e adesso non posso parlare dei dati, però dico è uno studio con 84 pazienti, quindi più grande, randomizzato, e insomma a breve sarà pubblicato, quindi vedremo anche l'effetto sul morbo di Crohn's. Ok, quindi finisco con lo studio almeno quella parte, poi c'è la parte del cancro, se ho tempo di finirla. Questo è uno studio sulla età biologica, quindi cosa succede? In questi abbiamo preso i dati del sangue in 2 studi clinici diversi, quindi in questi 2 studi clinici i pazienti di nuovo facevano 3 cicli di ditamina digiuno, e la domanda era cosa succede all'età biologica. In questo caso abbiamo usato questo BioAge method di Morgan Levine, e Morgan ha generato questa formula basata su marcatori che possono essere facilmente misurati nel sangue, per esempio proteina C reattiva, emoglobina glicata, pressione sistolica, colesterolo. Quindi adesso stiamo testando anche questi CLAC che sono epigenetici, però questo mi piace molto perché è basato su fattori di rischio che sono accettati da qualsiasi medico e persona del mondo come fattori pericolosi, e quindi anche se uno potrebbe contestare l'età biologica, il loro ruolo nel segnalare l'età biologica, comunque è difficile contestare che avere livelli ridotti di questi marcatori sia positivo, no? E usando questo metodo di Morgan Levine vediamo l'età biologica ridotta di 2 anni e mezzo. Quindi questi pazienti, almeno in base a questi marcatori, dopo 3 cicli di mio digiuno perdono 2 anni e mezzo di età biologica. Adesso vedremo l'effetto usando diversi metodi che sapremo a breve. Ok. Quindi cancro, rapidamente, tanto tempo fa avevamo iniziato con questi studi sulla chemioterapia, e faccio vedere sempre questo slide dove dico se andiamo a vedere non tanto la progressione del tumore ma cancer free survival, cioè quindi nei topi, quanti topi vengono curati da queste terapie. Quindi se usi la chemioterapia, nel caso di lung cancer 0, se usi i cicli di digiuno 0, se lo fai per triple negative breast cancer, quindi cancro alla mammella triple negativo, bassi 0 o bassi livelli di cura, cioè quindi dopo un anno il topo è ancora senza tumore, in remissione. La cosa ovviamente interessante è che se unisci la chemioterapia o al digiuno o alla ditamina digiuno, ottieni questo, non in tutti i tumori, ma in tanti tumori o in diversi tumori, ottieni questa sinergia. Questa, diciamo, era la prima fase nei nostri studi sul cancro e il digiuno e la mima digiuno. La seconda fase, invece, fatta a svolta all'IFOM, aveva a che fare con andiamo a capire qual è la riprogrammazione differenziata delle cellule normali e delle cellule del tumore. Quindi cosa succede a una cellula tumorale quando viene messa a digiuno? E quello che ci aspettavamo e che abbiamo visto poi, che vanno in direzioni opposte delle cellule normali. Le cellule normali si proteggono, attivano questi fattori di trascrizione della resistenza cellulare e riducono la divisione cellulare. Le cellule tumorali invece continuano, e però diventano anche disperate, quindi iniziano ad attivare quello che noi chiamiamo starvation escape pathways, quindi attivano delle vie di segnalazione che sono necessarie per la sopravvivenza, e Giulia Salvadori nel laboratorio all'IFOM ha fatto proprio questo, usando questo RNA-seq, quindi prima metti i topi a digiuno con un tumore, in questo caso alla mammella triple negative, e poi vai a vedere l'RNA come viene modificato, cioè l'espressione genica come viene modificata, cercando di identificare queste vie di fuga. Quindi quali sono le vie di fuga? Com'è che il cancro riesce a fuggire al digiuno? Vi ho fatto appena vedere che questi tumori se ne fregano del digiuno, e questo è anche interessante perché di nuovo molte persone pensano faccio digiuno e questo può avere un effetto sul tumore. Sì, ha un effetto sul tumore, ma un effetto in effetti minimo. Allora fai questo RNA-seq, vai a vedere come che il tumore si riprogramma e poi puoi intervenire su questi target, no? Su questi geni. Quindi in questo caso Giulia vede che il tumore aumenta questo TOR, PI3-kinase, IKT, adesso senza preoccuparvi del significato, ma usa 3 farmaci per bloccare queste tre vie di fuga, e il tumore che stava crescendo rapidamente scende e tra l'altro adesso rimane giù solo se, adesso è una cosa un po' complicata per questa audience, però solo perché la mimia digiuno agisce sia sulle cellule tumorali staminali con un meccanismo diverso da quello delle cellule tumorali differenziate. Adesso non c'è bisogno di andare nei dettagli, però diciamo che è questa abilità di questa dieta mimia digiuno di mettere le cellule tumorali in una situazione di alto confusione. Perché? Perché le cellule tumorali sono abituate a eccesso di glucosio, eccesso di aminoacidi, eccesso di fattori di crescita, quindi questo gli viene elevato e entrano in una fase disperata. Non sono abituati e spesso entrano in quello che noi chiamiamo anti-Warburg effect, cioè cercano di ritornare alla glicolisi e cercano di ritornare a usare i zuccheri per rimanere in vita, ma non lo possono più fare perché gli zuccheri sono ridotti o altamente ridotti in questi 5 giorni di dieta mimia digiuno, e quindi spesso attivano questa fortissima produzione di radicali liberi, quindi free oxygen species, specie di ossigeno reattive, scusate, e muoiono proprio di questo suicidio dovuto a questo anti-Warburg effect. Ok, quindi il gruppo di Debro e Vernieri all'Istituto Nazionale Tumori ha seguito i pazienti adesso per tanti anni. Uno dei primi studi relativamente piccolo, quindi uno studio piccolo ancora non del tipo che sarà conclusivo, speriamo un giorno, però confronta un piccolo gruppo di pazienti che fanno la mima digiuno insieme alla chemioterapia, questo di nuovo triple negative breast cancer, quindi la tipologia più aggressiva del cancro alla mammella, quindi confronta mima digiuno più chemioterapia con solo chemioterapia. Vedete che dopo 4 anni la sopravvivenza è quasi raddoppiata nei pazienti che avevano cancro metastatico e che hanno ricevuto sia chemioterapia che dieta mimia digiuno. Adesso un altro studio interessante fatto sempre da Debro e Vernieri è questa pubblicazione che in uno studio di 100 pazienti ce n'erano 5 che avevano varie tipologie di tumori in stadio avanzato e i risultati erano così sorprendenti che Debro e Vernieri decidono di pubblicare quello che loro chiamano risposte eccezionali dei pazienti, in questo caso pazienti che ricevevano immunoterapia, chemioterapia, una serie di terapie standard insieme alla mimia digiuno. Di nuovo studi da ripetere, ovviamente da allargare a centinaia di pazienti in maniera randomizzata, ma promettente, cioè un inizio promettente, come lo è. Adesso questo fa solo vedere che una delle critiche principali è che i pazienti non riescano a fare la mimia digiuno perché perdono peso, perdono massa muscolare. In effetti il gruppo di Nencioni in questo studio dove c'è scritto phase angle and fat free mass fa vedere che non solo non diminuisce la massa muscolare in questi pazienti anche dopo 8 cicli di dieta mimia digiuno, ma addirittura aumenta la massa muscolare e quindi questo questo può ovviamente essere un problema per una porzione dei pazienti che tendono a perdere peso, ma in questo studio non l'è stato. E poi finalmente di nuovo Vernier e Debro pubblicano quest'anno questo studio che è randomizzato in questo caso, in cui i pazienti ricevono o di nuovo triple negative breast cancer, quindi cancro alla mammella, ricevono solo la terapia standard o la terapia standard più dietamina a digiuno o la terapia standard più dietamina a digiuno più metformina, e in effetti la mima a digiuno aumenta di tanto quello che si chiama pathological complete response, quindi la risposta del tumore alla terapia, ma la metformina non aggiunge niente, quindi mima a digiuno più terapia standard o mima a digiuno più terapia standard più metformina hanno ottenuto risultati molto simili. Ok, quindi adesso non vado nei dettagli perché questo è uno slide molto più dedicato ai lavori, però diciamo che la mima a digiuno nel caso del cancro crea un ambiente molto ostile, come vi ho detto prima, quindi riduzione della glicemia, dei fattori di crescita e effetti sull'autofagia, riprogrammazione obbligazione o obbligare le cellule del tumore a fare delle riprogrammazioni che non sono più in grado di fare, insomma tutte queste cose insieme. E poi non ho parlato dell'effetto del sistema immunitario, che chiaramente sembra che la mima a digiuno e il danno autonomo che viene fatto dalla mima a digiuno alla cellula tumorale rende comunque la cellula tumorale più visibile dal sistema immunitario, e quindi c'è un effetto doppio sia nella cellula tumorale sia nei confronti del sistema immunitario, che adesso riesce a identificare e attaccare la cellula tumorale. E con questo concludo semplicemente dicendo che questi cicli di dietamina a digiuno hanno una serie di effetti sia metabolici, riprogrammazione metabolica, quindi insulinoresistenza eccetera, sia rigenerativi, quindi autofagia, di cui non ho parlato, cellule staminali, riprogrammazione di questi fattori cellulari. Quindi tutto questo insieme porta molti sistemi almeno a essere più funzionali, più giovani, e quindi da questo gli effetti pensiamo su tutte queste malattie che ovviamente sono difficili da ottenere con un singolo intervento. Grazie.
Hi, Walter. Thank you very much for this amazing presentation. So just one question on, I was expecting to see something maybe provocative compared to the GLP-1, and all the aspect of the GLP-1, the production of insulin and the insulin resistance. What do you think about this GLP-1 that is going on and everything compared to the fasting and to your diet?
Yeah, can I answer in English or Italian?
Oh, scusa.
Ah, in italiano, ok.
No, possiamo parlare in italiano.
Sì. La domanda è sul GLP-1 e cosa ne penso. Penso che dovrebbero essere, che hanno un ruolo, ma solo dopo che questo tipo di intervento, cioè adesso questo è uno, poi ce ne saranno anche degli altri, questo tipo di intervento insieme ai professionisti di questo tipo di intervento falliscono. Secondo me questo deve essere il futuro, cioè di dire lo riesco a fare senza danno, come abbiamo sentito prima dal professor Kirkland. Ci sono una serie di danni agli occhi, perdita di massa muscolare con i GLP-1 receptor agonist, eccetera. Ne è appena venuto fuori uno per macular degeneration, raddoppio del rischio di macular degeneration. Poi cioè è chiaro che anche questo potrebbe avere effetti collaterali, ma è molto più difficile che questo tipo di intervento con una dieta vegana fatta per 5 giorni ogni 2 o 3 mesi abbia questo tipo di range di effetti collaterali. Poi è ovvio che se ci fosse il team, che non esiste né in America e non è rimborsato, né in Europa e non è rimborsato, cioè quindi l'alternativa non c'è, perché questo non viene rimborsato, mentre il GLP-1 sì. Quindi dico, una volta creata quell'alternativa e una volta che quell'alternativa fallisce, come adesso per la chirurgia bariatrica, allora si può anche considerare, secondo me il GLP-1 ha un ruolo, perché a quel punto è chiaro che non c'è modo di riportare il paziente alla normalità. Comunque noi vediamo questi pazienti e la maggior parte entro 2 o 3 anni ritornano a non prendere farmaci e tra l'altro anche i pazienti che hanno ipertensione e diabete ritornano a peso normale, niente ipertensione, niente diabete, niente farmaci, no? Questo è il modo di andare avanti, no?
Era proprio sul fatto di dire provocativo, è proprio in quel senso. Però dal punto di vista del meccanismo d'azione, che io poi voglio dire, da un lato si spinge la produzione di insulina che poi potrebbe anche causare insulin resistance long term, e dall'altra parte invece andando verso la parte del fasting dovrebbe essere l'opposto.
No, è l'opposto.
Esattamente, quindi è l'opposto, per cui anche lì ho un po' è un po' puzzling, no? Come si riesce...
Sì, ma la cosa più importante è proprio la storia, cioè che questo digiuno rappresenta qualcosa che cioè si è evoluto questo programma di riverso dell'insulinoresistenza milioni di anni fa in specie che ci precedevano, no? Quindi questa è una cosa molto antica. È ovvio che, come abbiamo visto con la restrizione calorica, anche una cosa molto antica può essere dannosa, no? E quindi bisogna, in questo caso, assicurarsi che non c'è niente di quello che vediamo per questi GLP-1 receptor agonist.
C'è una domanda qui?
Buonasera, innanzitutto complimenti per il talk. Conoscevo gli effetti della dieta mima digiuno, però vedere nero su bianco gli effetti sono davvero sorprendenti. Ho solo una domanda. Sono mai stati comparati gli effetti della dieta mima digiuno con, per esempio, una dieta mediterranea in caloric restriction per un periodo di tempo, per esempio?
Sì, diciamo che questa è la domanda che ci fanno tutti. È chiaro che noi siamo partiti dal digiuno acqua, no? Quindi se fai una restrizione calorica anche con la dieta, cioè se fai lo studio di Heidelberg faceva quello ma senza la restrizione calorica. È ovvio che se fai una dieta mediterranea e la metti anche a restrizione calorica, probabilmente parzialmente funzionerebbe anche quella. Noi ci aspettiamo esattamente questo. È ovvio che la mima digiuno l'ha fatto De Cabo, l'aveva fatto sui topi e vedi comunque delle grosse differenze, diciamo, nella dieta isocalorica. Quindi lui fa il match nei topi, una dieta di contenuto normale e la dieta mima digiuno, quindi vedi le differenze, però si vede già la potenza, diciamo, del fasting mimicking in confronto a una dieta semplicemente calorie restricted o severely calorie restricted.
Grazie.
C'era una domanda qui?
Qui c'è una domanda. Questa è da addetti ai lavori, nel senso che non ho capito se si attivano le sirtuine durante questi cicli, perché nei pathways non mi è sembrato di vederle, però dovrebbero.
Probabilmente sì, non è una cosa che è mai emersa, perché ovviamente abbiamo fatto tanta RNA-seq, metabolomica, diciamo che non è mai emerso qualcosa di eclatante che aveva a che fare con le sirtuine, anche in questo multi-omic approach, no? Però sono emerse dei SIRT6, mi pare, non mi ricordo adesso quali. Ogni tanto vediamo delle sirtuine che sono elevate l'abbiamo fatto sia nell'uomo che nel topo, però per ora non sono le sirtuine che sono il top dell'espressione né a livello di metabolico né a livello di RNA.
Buonasera, grazie davvero per questo speech, insomma, bellissimo, ci ha dato anche tanta speranza, credo, per il futuro. Sto cercando di portarmi a casa qualcosa poi per me, la mia famiglia, mio figlio, il mio futuro. Spesso alle donne viene anche detto a una certa età di mangiare molte più proteine per non perdere massa muscolare e mi è sembrato di capire che la proteina animale, per quanto invece ci viene detto da tantissime cose che si leggono e si dicono, anche a livello divulgativo, c'è veramente di tutto, per cui per portarci a casa veramente dei take away molto utili per la sopravvivenza, per la longevità, per il futuro. Quali sono le scelte fondamentali che dobbiamo fare?
La scelta più fondamentale è di contattare la mia fondazione e farsi seguire da un nutrizionista esperto, perché poi diciamo, la tipica persona che noi convertiamo da diabetica ipertesa al normale ci vogliono 2 o 3 anni. Quindi, inclusi i medici, abbiamo una serie di medici che vengono da noi con malattie e ci vogliono 2 o 3 anni, no? Quindi le proteine animali vanno benissimo, siamo anche noi che raccomandiamo ad esempio dopo i 65 anni di aumentare di un 20%, però partiamo da 0,8 grammi per chilogrammo di proteine principalmente vegetali prima dei 65 anni per poi arrivare a 1 grammo per chilogrammo di un mix più vario, perché certi aminoacidi sono importanti sia nel topo anziano sia nella persona anziana. Questa è una cosa che abbiamo già dimostrato e pubblicato, quindi però di nuovo è complessa e va personalizzata, quindi siccome comunque il costo di un nutrizionista è anche abbastanza basso, diciamo, e poi volendo una persona può vedere un nutrizionista un paio di volte all'anno, non c'è bisogno, una persona che sta relativamente bene, stiamo parlando di un investimento di 200 euro all'anno, no? Questo secondo me è la cosa più importante. Purtroppo i professionisti di questo tipo quelli che abbiamo noi in fondazione, che abbiamo una ventina adesso, sia negli Stati Uniti che in Italia, questo training che noi facciamo è inesistente, cioè quindi o c'è il medico dietologo che comunque ha un training di alto livello, o non c'è una figura adesso diciamo certificata che sia diciamo affiancabile a un medico. Questo noi sono anni che parliamo con l'ordine dei biologi per cercare di crearlo, però sia in Italia che negli Stati Uniti comunque è una cosa, negli Stati Uniti peggio, perché almeno qui c'è il nutrizionista che è una magistrale, in America non c'è neanche quello, quindi è più una specie di triennale, quindi il top esperto di nutrizione negli Stati Uniti ha fatto 3 anni di scuola, e quindi ovviamente non può essere affiancato a un medico come esperto di stile di vita, no?
Ci sono altre domande? Sì.
Ok. Davvero, grazie.
Grazie, solo... Scusi.
Il partito.
Scusate.
Allora.
Solo una domanda veloce, se avete verificato anche un effetto di questa dieta mima digiuno sui marcatori infiammatori dell'infiammazione.
Sì, quindi l'abbiamo fatto sulla proteina C reattiva, l'ho fatto anche vedere qua ovviamente non erano dati comunque su un gruppo, un piccolo gruppo, l'abbiamo fatto in diversi studi clinici randomizzati, e quindi chiaramente ci sono delle riduzioni di proteina C reattiva in chi parte con una proteina C reattiva elevata. E poi insomma, diciamo che adesso ci sono una serie di studi sulle autoimmunità che usciranno quest'anno, incluso uno sulla sclerosi multipla, quindi ci saranno opportunità di espandere diciamo la conoscenza sull'effetto della mima digiuno su malattie autoimmunitarie infiammatorie, vedremo, però diciamo che per adesso i risultati sono stati molto positivi.
C'era una domanda lì?
Sì, molto rapidamente volevo sapere se avete fatto grazie mille ovviamente per il talk, se avete fatto delle riflessioni e le possibili implicazioni sulla Barker hypothesis di tutto il lavoro vostro sulla dieta mima digiuno, cioè se cambia in un certo senso nella life course perspective cosa sappiamo del mismatch della prediction della vita post natale rispetto all'ambiente.
Dici la Barker hypothesis?
La Barker hypothesis, sì, se avete...
Questo è molto interessante perché in effetti la restrizione di certi aminoacidi in topi molto giovani cambia l'aspettativa di vita nei topi, no? Quindi è possibile che un intervento, probabilmente epigenetico, però nessuno lo sa, non so se qualcuno ha mai capito, la Barker ipotesi addirittura parte ancora prima della nascita, quindi va a vedere il feto, la posizione del feto, quindi insomma c'è la possibilità che l'esposizione, diciamo nelle early days of life o addirittura nell'embrio, che l'esposizione possa riprogrammare, ad esempio i dati sui topi che sono growth hormone receptor deficient, questi sono i topi che hanno il record di longevità. Se a questi topi però gli dai l'ormone, quindi è come se non avessero ormone della crescita, no? E vivono il 40% di più, sono molto più sani. Però quando a questi topi gli viene dato l'ormone della crescita all'inizio della vita, l'effetto sparisce quasi completamente, no? Però c'è anche da dire che invece l'intervento con la rapamicina a middle age, quindi a mezza età, funziona. Quindi insomma probabilmente c'è un effetto sia molto di riprogrammazione very early, e sia un effetto poi lifelong, dove tu ad esempio con la restrizione calorica puoi intervenire anche in un topo vecchio, già vecchio, e comunque gli estendi la vita. È ovvio che se parti dal topo giovane l'estensione è 2 o 3 volte maggiore. Quindi insomma, però io sono molto interessato, comunque tanti sono molto interessati proprio a questa riprogrammazione potenzialmente epigenetica in early life, per un controllo poi del programma di vita, no? Quindi questi piccoli topi sembrano entrare in un programma diverso da quello dei topi di dimensione normale. Sono molto più piccoli perché non hanno questo IGF, insulin-like growth factor 1.
Ci sono altre domande? Una, forse l'ultima.
Grazie. Avrei 2 domande. Una è se lei consiglierebbe questo fasting o questo mimic fasting a pazienti che hanno cancro cronico tipo il multiple myeloma, e la seconda domanda sarebbe pazienti con osteoporosi, visto che digiunare è uguale perdita di peso e normalmente è sconsigliato per la perdita anche di massa ossea. Quindi volevo sapere.
La prima malattia era?
Multiple myeloma.
Multiple myeloma. Multiple myeloma è interessante perché avevamo seguito un famoso scienziato che era a Southwestern Medical Center, che Fabrizio probabilmente conosceva, Irwin Wright, e che aveva il multiple myeloma, e lui per tanti anni aveva abbinato queste bortezumi, questi farmaci che vengono dati ai myeloma patient col digiuno anche prolungato. Aveva fatto digiuni di 10 giorni, 13 giorni. Questo è un caso, non significa niente, però lo vediamo con molte leucemie, linfoma, quindi vediamo che l'unico tipo di tumore in cui vediamo la mima digiuno o comunque forti cambiamenti di dieta avere un effetto da soli sono i blood cancer, quindi i tumori che coinvolgono le cellule del sangue. Ovvio che adesso bisognerebbe fare uno studio randomizzato che non è stato ancora fatto, insomma, soprattutto per le leucemie. Poi per l'osteoporosi l'unica cosa che abbiamo fatto, abbiamo visto che i cicli di mima digiuno non riducono la massa ossea, questo fatto con DEXA nelle persone in multiple trials. Nei topi, facendo mima digiuno lifelong, cioè tutta la vita, vediamo che i topi che hanno fatto la mima digiuno hanno una densità ossea superiore, arrivati alla vecchiaia. Questo è l'unico dato che abbiamo, però non abbiamo ancora i dati sulle persone, cioè non abbiamo mai fatto uno studio di 5 anni, cioè lo studio sulle persone sarebbe molto più lungo, mentre quello sulla massa muscolare lo possiamo vedere molto prima, sulla massa ossea probabilmente avremo bisogno di anni per vedere una differenza.
Grazie ancora.
Grazie.
Concludiamo perciò una giornata intensa, fruttuosa, crediamo, in tutte le sue fasi, la prima parte più scientifica, la seconda più divulgativa, e perciò concludiamo ufficialmente la giornata ringraziando di nuovo IPSA che ci ha permesso di passare questa giornata assieme, gli istituti IFOM e CNR che ci hanno accompagnato in questa giornata.
Grazie ancora, ringrazio soprattutto Lusi che ci ha dato la possibilità di essere accolti in questa bellissima aula, e l'Istituto Oncologico di Ricerca che è stato il co-organizer di questo evento. Grazie a tutti.